Progress.
Test, test, test one, test. अरे test, test one, check one, test.
Good morning, ladies and gentlemen. Prior to the official welcome, I would like to welcome you all for the Asda 2022. In order to inaugurate the Asda 2022 in a ceremonial manner, I would like to welcome distinguished to lighting the traditional oil lamp. Dr. Ajanta De Silva, Director General of Agriculture, Mrs. Janta Ilangakona, Additional Director General Development, Dr. Samanthi Vasala, Additional Director General Research, Professor Ogata Kazu, keynote speaker who is all the way from Japan to participate Asda 2022, Mrs. Disna Ratnasingha, Chairperson Asda 2022, and Mrs. HMT Kumari, one of the paper presenter today. So you all are welcome to, lighting the traditional oil lamp. In addition to that, I would like to welcome Dr. Sumit Abe Sirivardhana to join the traditional oil lamp. Thank you very much. Ladies and gentlemen, please raise for the national anthem of Sri Lanka.
Very good morning to you all. Director General of Agriculture, Additional Director Generals, Development and Research. We are very glad to see you all here, both physically and virtually, to participate at the Asda 2022. As the chairperson of Symposium of the Department of Agriculture, Asda 2022. Asda is the most prestigious annual event of the Department of Agriculture, which brings all the outputs of the all hard work and the dedicatedly carried out by you during the past few years. All your new research findings and other innovative development works will be displayed here today in a very ceremonial manner, in line with the theme of today's Asda, Feed the Land, Feed the Nation. First, I would like to welcome the Director General of Agriculture, Dr. Ajante de Silva, the personality behind all the success stories of the department. Your constant guidance and encouragement greatly helped us to make this event a success. It's my great pleasure to welcome Additional Director Generals and all the members of the Directorate and Provincial Directors of Agriculture to this event. Your presence here is an encouragement for us. Then I would like to welcome all the JICA officers who are joining with us today, including Professor Ogata Kasuo, a Chief Advisor from JICA, also from Kyushu University, Japan, the keynote speaker in the ceremonial session today. We are encouraged by all of your presence here. Then I am very pleased to welcome the chairpersons of technical sessions at NAICC, Dr. Sumit Abesirivardhana, and at PGIC, Dr. WMADB Vikram Singh. We are very pleased to see you here, sir. Actually, thank you very much for accepting our invitation to chair the session, even in the mid of your busy schedules. Finally, I warmly welcome all the officers of the Department of Agriculture who joined with us today, physically and virtually. Today, we are going to unveil all your new findings too, will reflect all your dedicated hard work during the past few years. So this year, Asda technical sessions are organized as 2 parallel sessions at PGRC and NAICC under 6 different themes. The program is enclosed in your docket for your reference. The ceremonial session will be starting at 3 p.m. in the evening at PGRC. We are expected to felicitate our best performing officers there. So once again, welcome all of you to the annual symposium of the Department of Agriculture, Asda 2022. Thank you very much.
Thank you very much, ma'am. So I cordially invite Dr. Ajante de Silva, Director General of Agriculture, to deliver the opening remarks.
Ayubowan and very good morning to all of you. Additional Director General Development, Ms. Jayanta Ilanko. Additional Director General Research, Dr. Samanthi Vasala. All the members of the Directorate of the Department of Agriculture, provincial directors, district directors, principal agriculturists, principal scientists, all who are involved in the Asda 22, from the chairman Asda 2022 and all the executive committee members, all who are involved, who are now logged into the sessions of today Asda technical sessions, ladies and gentlemen, and also Professor Ogata Kazuo from who is going to be the keynote speaker for today, and all the participants from JICA, Sri Lanka. It is indeed pleasure to address you at this moment in the ceremonial opening of the today Asda 2022, which is the 24th Asda consecutively carrying out by the Department of Agriculture. Starting from 1999, so we have been able to continuously have these sessions despite all the problems that we have in the country, as well as even with the other difficulties. So therefore, first of all, I would like to thank all the officers who are involved in this one, and of course, the previous, those who have immensely supported the Asda. So Asda, as we all know, is the forum for the scientists who are in the Department of Agriculture, the externals who are in the development stream, and the exchange, the economics who are in the Department of Agriculture, to deliver and to publish their research findings in a methodical manner to the scientific community in a glamorous way. So during the past also, we have gone through the similar exercise from 1999, and in fact have delivered so much of research and extension findings to the Sri Lankan agriculture community as a whole. So in that sense, I think the Department of Agriculture is really going forward to support the national initiative of the development of agriculture in Sri Lanka for the betterment of this country, people. Now, the Department of Agriculture is really has to come in big way, since we all know we are in a crisis because of different, I should say, the activities in global and nationally occurred during the last 1 or 2 years. So therefore, I think the theme that we have already selected, the seed the land and feed the nation, which is really timely concern, and of course, it is the right thing that we should do to restore the self-sufficiency and also to see that Sri Lankan people, that our motherland is really once again nourished with the food which is needed by the people, and also to see that the export potentials that we have to gain foreign exchange from other countries. So in that context, I think the Department of Agriculture has the capability of doing that one. And of course, the support given by all the agencies from private sector, and also the UN organizations, and the organizations like JICA, COICA, and all the other, those who are friendly to Sri Lanka is mostly welcome. And I hope that today's deliberations will give more insights what the department is doing for the people of this country and also the scientific community. Let me also thank Dr. Sumitabe Sirivardhana and Dr. WMADB Vikramasinghe, who is going to be the first session chairman at the invitation of the Department of Agriculture, and all the others who are going to be involved in the sessions by supporting as chairpersons and the judges during the course of the day. Also, finally, let me wish that the deliberations today will be a very fruitful and also very much supportive to the national agriculture development in Sri Lanka. Thank you very much.
Thank you very much. Thank you very much, sir. So the brief ceremonial opening of the Asda 2022 ends here, and we are moving to the technical sessions. So it is my duty to introduce the technical sessions. The 24th annual symposium of the Department of Agriculture comprises of 2 parallel sessions at Plant Genetic Resources Center and the National Agriculture Information and Communication Center. There will be 20 presentations organized on the 6 thematic areas. So the sessions are available virtually through the Zoom meeting as well. So kindly join us if those who cannot join on ground. Again, all are welcome to participate the technical sessions of the Asda 2022. Thank you, ladies and gentlemen. Thank you very much.
Good morning, all. Distinguished guests, ladies and gentlemen. I'm very pleased to welcome you all to the first technical session of
Good morning. Good morning, ladies and gentlemen. On behalf of the Executive Committee of the Asda 2022, I would like to welcome you all to the second technical session of the 24th Annual Symposium of the Department of Agriculture, Asda 2022. A special welcome goes to the chairpersons of the session, the panel of judges, presenters, and all the participants. Though this is the first technical session at PGRC, we numbered this as a second session since, as you all know, parallel session is happening at the same time at NICC. The technical session is Soil, a Challenge and an Opportunity to Sustainable Development. The session will be chaired by Dr. WMADB Vikramasinghe, one of the eminent scientists in the field of plant nutrient. Dr. Vikramasinghe was the former Additional Director General Research in the Department of Agriculture, retired after 36 years of service to the Department of Agriculture, and after his retirement, he joined with the UNDP and presently serving in the UNDP as a technical advisor on environment. So I would like to cordially invite Dr. Vikramasinghe to chair the session.
Right, good morning, everybody. And it's always pleased to be with you after several years. Always be happy to and feel more comfortable when you come to your Mahagedara. So I'm really glad to see you again. And yes, we'll start the usual business as usual. And before that, I think, thank you very much for your kind introduction. And also, I think we have 3 judging panelists sitting behind you, I think over there. Mr. Kandaragama, one of my colleagues at the NRMC, who is a very eminent scientists again in soil science. Then Professor Dr. Chammi Attanayaka from the University of Peradeniya, senior lecturer. And also, I came to know that Mr. Prasad Idamakorala from Export Agriculture Department, again, soil scientist. Welcome you, sir. And yes. And yes, we have 4 presentations in these sessions. So each presentation allocated 20 minutes. And but we'll allow only 15 minutes for presentation, and we'll go for 5-minute discussion if they have any questions. Then, of course, after 12 minutes? After 12 minutes, you will hear a ring sound, giving notice that you will have only 3 minutes left. And then again in 40 minutes, that you will to indicate that you will have 1 minute left. And then finally in 15 minutes, I think you need to stop. I think you should adhere into the time allocated. I think time management is very important. One of the objective of this exercise is to manage your time. I have seen in a lot of forums that, when people start talking, and they start talking, and ultimately, sometimes they just cover only the introduction and sometimes the methodology part. But most importantly, the results and also the discussion is very much important. Try to be brief on introduction and maybe a brief methodology, I think. So this is a little, this will be helpful for you to, make your time. Right, the press presentation is an invited presentation. It will be done by Miss or Dr. Miss Upul Kumari Ratnayaka, and also Mr. D.N. Sirisena is also co-author of this presentation. And Dr. Ms. Upul Kumari will talk on strategies recommended to minimize fertilizer usage in rice cultivation in Sri Lanka and overview. It's overview. I think just to introduce Ms. Upul Kumari, she is graduate from the Peradeniya University, BSc Agriculture graduate in 1993. She obtained her master's degree in natural resource conservation from Asian Institute of AIT, Thailand in 1991. And later, she obtained her PhD in geoinformatics from University of Peradeniya in 2019. So earlier, when she joined the department, she has served in Field Crop Research and Development Institute, Mahalupallama, and on the field of soil and water management. Then later, she got transferred into Rice Research and Development Institute, Batalgoda, and conducted research on field of water management, soil science, and geographic information systems. She was promoted to Principal Agriculture Scientist in soil science in 2017 and acted as an additional director at RRDI from 2018 to 2020. And she has a number of publications under her. And I cordially invite Dr. Ms. Upul Kumar Ratnayaka. I usually call her Upul, to make a presentation, and over to you, Upul. Thank you.
Thank you, sir. Good morning, Chairman, sir, my colleagues, and ladies and gentlemen. The topic of my presentation is strategies recommended to minimize fertilizer usage in rice cultivation of Sri Lanka. This is a review report. Rice is consumed about 110 kilograms per person per annum. To meet the rice requirement of the 22 million people in Sri Lanka, we need 2.4 million tons of rice. To give that quantity of rice, the 3.7 tons of paddy is needed. To provide the required quantity, paddy cultivation is practiced in all parts of Sri Lanka, covering various soil groups. And to maximum rice production, high-yielding shortage varieties have been introduced to most of the paddy fields, and these varieties are grown in 2 seasons per year, Yala and Maha. And the productivity of the above high-yielding rice grown in different soil groups varies from 2 tons per hectare to 7 tons per hectare. In addition to inherent soil productivity, cultural practices as well as climatic factors also affect on yield variability. The plant nutrient management is essential to achieve the yield potential of these rice varieties. The essential plant nutrients are mentioned here as macronutrients and micronutrients. The nutrient deficiency may appear when essential nutrients are not in sufficient quantities, and when there is the excess nutrient, it may be toxic to the plants. So it is important to maintain the nutrients at the critical limits of plant to obtain the normal plant growth. And yield levels are determined by the limiting nutrients, especially nitrogen, phosphorus, and potassium, and the other nutrients. If we have all nutrients, we can get more than 5 tons per hectare of rice. If there is no any nutrient in NOK also, at this moment also we can take about 2 tons per hectare of yield according to the soil's carrying capacity. When harvesting, nutrients will be removed as mentioned here, and the nutrient removal is increasing with the increasing of yield. The critical levels are here you can see the critical levels of nutrients in soil and plant tissues. These limits we have to consider when fertilizing. And with this general information, I move to our fertilizer recommendation for rice in Sri Lanka. First fertilizer recommendation for rice was initiated by the Department of Agriculture in 1956. After that, it renewed in several times based on the fertilizer availability, farmers' response, nutrient accumulation, fertilizer use efficiency, environmental pollution, yield levels, and water availability. Then the major nutrients NPK in both Yala and Maha season are recommended, and the micronutrients zinc only during Maha season is recommended in our fertilizer recommendation. The major component of the current fertilizer recommendation, for the chemical fertilizer use, urea triple superphosphate and muriate of potash and the zinc sulfate. And the application of organic manure has been encouraged in every recommendation. Now let us look the latest recommendations to 2001 and 2013. Fertilizer recommendation in 2001 was based on the target yield. The whole country was categorized into 8 ecological regions, considering the dry and intermediate zone, wet zone, major irrigated, minor irrigated, and rain-fed. Then 2013, fertilizer recommendation was based on the fertilizer use efficiency. Then the country was categorized into 4 major groups: dry and intermediate zone irrigated, dry and intermediate zone rain-fed, wet zone irrigated, and wet zone rain-fed paddy. The total use was cut down by 25% from 2001 to 2013 for the majority of majority of the paddy fields located in the dry and intermediate zone under irrigated condition. You can see the 2001 total fertilizer requirement and the 2013 requirement, and then it totally save about 24%. 27%. As well as urea, TSP and MOP usage in paddy fields also reduced from 2001 to 2013. That means when we are renewing the fertilizer recommendation, always scientists have identified measures to reduce the fertilizer usage and to increase the fertilizer use efficiency. The measures taken to minimize nitrogen fertilizer use in paddy fields from 2001 to 2013, the first one is introduction of leaf color chart for the dry and intermediate zone and wet zone paddy fields. It reduces 25% of the nitrogen applied to paddy fields. Then they identified and introduced the granular urea in place of prilled urea to reduce the nitrogen losses. And nitrogen application have been changed from 3 applications to 4 applications with lesser quantities to increase the nitrogen use efficiency. And the nitrogen supply from rice, straw and stubble and the other organic matter help to remove the nitrogen fertilizer from the basal application, and the nitrogen requirement was fulfilled by those organic matter and rice, straw and stubble for the first 2 weeks from the establishment. Then it helped to save 15 kilograms of urea per hectare. Then what are the measures taken to minimize P fertilizer use in paddy fields? In 2013, P fertilizer used in paddy fields was minimized by reducing the critical level of soil P from 10 ppm to 5 ppm. This was based on the phosphorus response field trials carried out island-wide and considered the phosphorus accumulation in the soil. And the measures taken to minimize potassium fertilizer use in paddy fields. Based on the potassium fertilizer response studies were conducted island-wide, and the rice straw and rice stubble introduced as the good source of potassium to maintain the exchangeable potassium. Therefore, the basal application of MOP fertilizer was removed from the rice fertilizer recommendation. To avoid the yellow color appearance of rice during the maximum growth rate and to improve the grain filling rice, K application was recommended at 2 times as 4 or 6 weeks after planting in the latest recommendation. And zinc fertilizer was recommended to apply as zinc sulfate once a year only in Maha season. And this is the current fertilizer recommendation. There are 4 ecological conditions categories, and chemical fertilizers, urea, TSP, MOP and zinc sulfate with organic matter or compost. And when at RRDI Batalagoda, they have conducted, they have started the trial in 2000 onwards, and now it is around about 34 seasons, continuous seasons. During this period, we could see the highest yield has given when we applied NPK plus organic matter, which is our DOA recommendation. Then it follows NPK only, then organic only, and no fertilizer has given the lowest yield. Those recommendations are mainly blanket fertilizer recommendations we call, because it is a single recommendation for thousands of rice growing farmers, and it is very broad basis as agroecological zones, yield level or water availability. But instead of that blanket recommendation, soil test-based fertilizer recommendation was introduced to provide the more specific recommendation for individual land units based on soil nutrient levels of particular fields. This was initiated by the Department of Agriculture in 1993 and updated in 2014 for rice by reducing the level of application of fertilizer based on soil fertility levels. This is the soil test-based fertilizer application for potassium fertilizer for rice, for dry and intermediate zone and wet zone, and based on the soil test values. And this is the same for phosphorus fertilizer for rice based on the soil test values. This soil test-based fertilizer application is very important because we have conducted and we have mapped phosphorus levels, soil available phosphorus levels in Putlam, Kurunagala, Polonnaruwa and Anuradhapura area in paddy soil. We found that there are, in Polonnaruwa district, there are 58% of the paddy extent have more than 10 available P content, ppm. That means it is excess. So when Anuradhapura, it is 80% have more than 10 ppm. That means we do not have to apply that P fertilizer into those areas. So if we use the blanket recommendation, we do not care that. So this is very important. And based on this, GN division-based fertilizer recommendation was introduced. And the soil test-based fertilizer application, soil test kit-based PK fertilizer application and correction of pH was introduced. It is an easy and quick method, reduce P and K fertilizer, increase fertilizer use efficiency and save time. And as a site-specific fertilizer application, map-based fertilizer recommendation was introduced. Let us see how to get the fertilizer recommendation on map-based. We have to open the Google Earth Pro app. And then we have to select the file. It should be in KML format. And then we can go and we can select the place we want, the paddy field we want, or the liyadda. So we can click here and we can get the fertilizer recommendation for the particular place. And the map-based soil reclamation, this was based on soil pH, soil salinity and organic matter. And then the seedling treated with phosphorus fertilizer in parachute nursery in rice cultivation was introduced. It saves 25 kilograms of TSP per hectare and reduce soil and water pollution. And the phosphorus fertilizer application in alternate season rice cultivation, here we can apply only in Yala season and it saves 50% of TSP. And nitrogen enrichment of partially burnt rice husk is one of the alternative to reduce the nitrogen use efficiency. From this we can cut down nitrogen by 50%. And the rice varietal response to low level of nitrogen also a good method. So we have identified some varieties given at the highest yield at 50 nitrogen kilogram per hectare instead of the recommended 100%. In future, also site-specific nitrogen, phosphorus, potassium application and the organic matter and the use of biofertilizer will further the chemical fertilizer use in future. And based on the soil salinity, soil pH and organic matter, we can produce the new fertilizer recommendation in future. And already we have mapped and we have developed soil EC and soil pH maps in paddy fields in Sri Lanka. And I would like to acknowledge our former soil scientist at RRDI, Dr. WMADB Vikramasinghe, Dr. WMJ Bandara and Mr. DN Sirisena for their valuable contribution to increase that fertilizer use efficiency in rice cultivation. Thank you very much.
Thank you very much, Dr. Ms. Ratnayaka. Very interesting. And you have almost covered a comprehensive review of the fertilizer sector, especially in terms of nutrient in rice. And yes, and also you finish your presentation within the allocated time. Of course, we will have about 5-6 minutes for discussion. Perhaps I think we have a lot of questions regarding fertilizers because we have passed a turbulent time. And yes, may I have burning questions? Please raise your hand. And no, it's clear. Okay, Upul, I think I have just noted down and few points in this presentation, but the most important thing is the way forward, the future. Now, in the absence of fertilizers, so the way forward, this is very important. What are we going to do? The recommendation is there. Of course, I always mention in all the forums that there are two things. The recommendation is different from the way they distribute the fertilizers. But recommendation is a scientific document. Now, for example, some people are talking about the new fertilizers. Of course, they are not really new, but say about compound fertilizers, perhaps to increase the efficiency. And then in the recent development, again, the other sources of fertilizer like phosphorus. Now, I know that personally, it's very difficult to get TSP sometimes because of the prices and other standard we have levied on that one, the required specification. In the absence of TSP, in case, what would be the other options you are having in the Department of Agriculture? For example, diammonium or monoammonium phosphate fertilizers. Do we have a recommendation? And then single superphosphate. What is your opinion? Perhaps I think the scientists in the audience also can comment on this one. So what would be the option of the Department of Agriculture in these scenarios?
Yes, sir. Thank you, sir. In this review paper, I did not consider that part. I considered only the proven technologies in this paper. But nowadays, that problem arises. So we have to find many alternatives for the chemical fertilizer. So there are so many organic fertilizers reached to our station also to test, and for the nitrogen, phosphorus and potassium fertilizers, and the single superphosphate and the diammonium phosphate also. There are so many requests, and so that we are going to test first. Before doing the field test, we are not going to recommend those things. At least our research station and the farmer fields. And for that, we are also developing some nutrient, nitrogen enrichment. I mentioned here, partially burnt rice paddy husk also. We have introduced that one also to replace urea by using the nitrogen sources to enrich that paddy husk. And the liquid fertilizer also we are developing to get as the supplementary, and then that single superphosphate and other fertilizers coming from the outside, we are testing.
Yeah, thank you. But always better to have a plan B or C. And yes, I'm not going to, say much about that one. Better to have the department. But for me, I think it's better to, because, if you want to do field research, that take another 1 year, 2 years, and 3 years. By the time things have changed again, perhaps I think my opinion is use your technical knowledge. Some of these are, you don't need to do field research. Perhaps I think you can use the technical knowledge. And I think this is the best forum to discuss these things. All the, scientists in the department, within the department, you can discuss these things and come out with a kind of a, practical solution in this type of, calamities. And okay, and better to have, I think it's always I believe that your technical knowledge and skills and field experience, use it, yes, to
Yes, sir, for some.
find a quick solution and answers. This is just a hint. Okay, any other questions? No, I'm the only person as usual. Right, anyway, thank you very much for very good comprehensive presentation. And I think CD deserve a round of applause. Thank you very much, Upul.
Okay, thank you very much.
Yeah. Right. Okay, now we will go for the other 3 presentations. Of course, the presentations, these 3 presentations will be evaluated by the judging panel seated behind you. And I think as usual in Asda to find best presentation and best paper. So we'll go for, okay. The next paper is by Dr. Dr. Kodikara, KMS Kodikara, and PS Sangwan. And Dr. Kodikara will talk on effect of different levels of phosphorus on availability of micronutrients. So effect of phosphorus on availability of micronutrients in soils and their uptake by rice under submerged and aerobic conditions, under 2 different conditions, submerged and aerobic conditions. Dr. Kodikara, as far as I know, after long time I met him and still attached to RARDC Bandaravila. So over to you, Dr. Kodikara.
Good morning, Chairman Sir, as well as Judge Panel and all audience. I warmly welcome all of you to my presentation on effect of different levels of phosphorus on availability of micronutrient in soils and their uptake by rice under submerged and aerobic conditions. Actually, this paper I presented from my research conducted at my PhD study, one part of that. Therefore, my co-author is my supervisor, Associate Professor P.S. Sangwan from CCS HAU, Haryana Agriculture University. First, we will get some introduction to my presentation. Normally, we know rice we are cultivated under mainly 2 water regimes. First one is aerobic. Actually, aerobic conditions we are maintained for the mostly upland condition with rain-fed. If we need some supplementary irrigation, we can practice for the aerobic rice. But in case of submergence, we are totally kept our soils with the stand-in water layer, basically at least up to the panicle initiation of the rice. With these 2 water regimes, normally we are growing, but majority of farmer fields in India they are doing submerged agriculture with the irrigation facilities. However, with any water regimes, the fertilizer management is most important. When concerning about the macronutrients, phosphorus are getting more important because it is growth limiting nutrient as well as essential plant nutrient. Therefore, we have to think about phosphorus management in the rice field under any one water regimes, that means aerobic or submerged. However, we have to manage the phosphorus better way. In other side, when we concerning about the phosphorus deficiencies, usually phosphorus deficiencies we can see more in the uplands than the submerged conditions because of the different soil chemistry in the soil. Therefore, different water regimes express different nutrient bioavailabilities, especially on phosphorus and some of other nutrients in even identical soil. Then we will move to the properties changes in the soil with the submergence. Actually, physical, biological, chemical as well as electrochemical properties will be changed with the submerge of the soils. As example, in the physical, there are changes of oxygen depletion, carbon dioxide accumulation, the compaction, bulk density, structure, then gas diffusion. All of that factors will be affected with the submergence of the soils. Then biologically, the reduced aerobic microbial activities, the mineralization, immobilization, such as factors will be affected with the submergence. The chemically soil reduction and micronutrient toxicities will be appear with the waterlogged or submerged conditions. As well as most important part concerning in my study, the electrochemical changes in submerged soil. Under these parameters, the first thing is decrease in redox potential. With the submergence, as usual, you know, the redox potential decrease drastically, and it will be caused to the different nutrient behaviors in soil. As well as soil pH will be neutralized with submergence. That means increase in pH in acid soils and decrease in pH in alkali soils. As well as specific conductance will be changed or increased with submergence. As well as cation exchange reactions, especially involving in ferrous, will be changed with submergence conditions. As well as soil sorption and desorption mechanism also change under the submerged conditions compared to the upland or aerobic condition. Here I am showing some example to change of these micronutrients, especially manganese and ferrous, how their reactions are changed under the different redox potential and how they are converted into available forms or changes of their chemistry under submerged conditions. Here also showing a similar thing with different nutrient or compounds, how to change their reactions according to the redox potential in soil and how we can manipulate or how we can utilize it for increase the soil nutrient availabilities in the soil. We can get some idea about redox potentials and their compounds in soils. With this concerning these factors, the flooding or submergence has major effect on the availability of macro and micronutrients. Some nutrients are increased in availability to the crops with the changes of the water condition, as well as some are subjected to greater fixation or loss from the soils with respect to water regimes and pea application. Appropriate management of pea fertilizer is a major concern for rice growing soils, and adequate amount of phosphorus are essential to increase the growth and yield. The surplus of pea application over crop demand causes to pea accumulation in soil with creating nutrient imbalances in agricultural lands and loss of runoff and erosion towards to the eutrophication of water bodies can be happen if we P fertilizer used without proper management. Actually, in Sri Lanka also we are facing this this situation, especially in the upcountry soils with vegetable and potato growing soils. There are too much high phosphorus are recorded. With this brief introduction and those facts, our objective was this study to find out the soil availability and plant uptake of zinc, ferrous, manganese and copper under different P levels in submerged and aerobic rice. Brief methodology for my research. The location was for the field activities. We have selected Rice Research Station Call. It is actually Haryana province. It is belong to Haryana province. And my laboratory works conducted at the Soil Science Laboratory, CCS, HAU, Haryana Agriculture University, Hisar, Haryana. The treatments, we followed two water regimes as a main factors, submerged water regime as well as aerobic condition. The 5 levels of phosphorus we tested, 0, 20, 40, 60 and 80 kilograms P2O5 per hectare, and design was 2-factor factorial RCBD. The 3 replication was practiced, and test crop was rice, and the variety was HKR 127. Then phosphorus and zinc sulfate, they are recommending as a basal, as well as some part of nitrogen also applied as a basal, and nitrogen added 2 splits after basal application, and other practices were conducted as the package of practices recommended for rice for Haryana. And we are not added K fertilizer due to the initial potassium level was high in that area. Therefore, based on soil test based fertilizers, we are not applying potassium fertilizer to my experimental plots. The samples was collected from 0 to 15 topsoil layer and collected at initially before planting, and after that 40, 60, 80, 100 and 100 days after the phosphorus application, and after the harvest we collected soil samples for the analysis, and we analyzed initial properties of the soils including pH, EC, organic carbon, available NPK, zinc, manganese, copper and ferrous. Then we continuously take the samples at above mentioned dates, and we analyzed availability of zinc, manganese, copper and zinc, ferrous. Finally, with harvest, we analyzed for micronutrients, zinc, manganese, copper and ferrous in grain and straw for our calculation, as well as grain and straw dry yield. Finally, nutrient uptake was calculated using below mentioned equation. These are the procedures for sample analysis. The pH was analyzed in the pH glass electrode method using pH meters, electrical conductivity meters for EC, organic carbon used Walkley and Black method. Available nitrogen was alkaline permanganate method, also P for phosphorus, available phosphorus. Available potassium was extracted from ammonium acetate and read it in flame photometer method. And the available micronutrients was extracted using DTP extractant, and reading was get using atomic absorption spectroscopy, as well as plant samples digested at nitric and perchloric 9:1 diacid solution, and read using atomic absorption spectroscopy. These are the experimental plots, some pictures of experimental plots, and you can see here under aerobic condition there was dry, we maintain it under dry condition, just only applied supplementary irrigation when rainfall is not enough, but under submerged condition we are kept nearly 5 centimeter water layer throughout the up to panicle initiation of the crop. These are results and discussion. These are the physicochemicals of soil properties of the initial samples, experimental site. pH was 7.6 like that. You can see here, these are the basic soil properties which are related to our experimental site soil. These are DTP extractable ferrous changes. Actually, phosphorus levels are not much effect on that. The significant effect was not showing. Therefore, I am here mention only water conditions affected for the ferrous availability. You can see here the huge differences between aerobic and submerged condition reported related to ferrous, and with the time period it was decreased. Basically, for the reason for that, insoluble ferrous forms will be converted into soluble ferrous forms with the submergence. Therefore, more ferrous Fe2 will come to the soil solution, and it will be cause to the increase of available ferrous. This is related to the manganese. The similar pattern is similar pattern was observed in manganese as well as ferrous. You can see here with the submergence, it is significantly increased manganese content in the soil compared to aerobic condition, as well as with the time it was decreased. Here also main reason for with the reduction of soil, the insoluble manganese forms will be converted into water soluble manganese form, and it will be available to the plants. These are copper, changes of extractable copper. You can see here there are no much significant impact. Just only water regimes will be effect only initial stages. That means 40 days after P application was observed changes of copper. It is also a similar scenario with the submergence. Most of copper insoluble forms will be converted into available forms. In zinc, it is reverse of that observation. That means aerobic conditions will be increase the zinc availability compared to the submerged condition. It is also observed only the initial stage of the experiment. There are also some reasons. The available zinc will be converted to the unavailable forms under submerged conditions. That are the reason for reduction of the zinc in submerged condition. These are uptake of concerned micronutrients. According to these figures, you can see all factors are affected significantly to increase the micronutrients in both season 2018 and 2019. The results experiment revealed that submerged water regimes increase significantly grain and straw yield by 27.9 and 34.5% respectively over aerobic conditions. Straw recorded higher concentration in micronutrient except copper in aerobic condition. P application rate up to 40 increased grain yield significantly, and further increase did not affect significantly. However, the sequence of micronutrient uptake in grain and straw were zinc, ferrous, manganese and copper, and ferrous, manganese, zinc and copper respectively in both water regimes related to grain and straw. Finally, my conclusion: growing rice under submerged condition with the application of 40 kg P2O5 can be recommended to obtain sufficient micronutrient availabilities and crop yield while optimizing the micronutrient uptake in the studied soil. My acknowledgements going to CARP for the financial support to my study and staff of the CCS Rice Research Station and the Faculty of Agriculture. Thank you very much.
Thank you very much, Dr. Kodikara, but you have taken a little more time than allocated time. But we'll have about 3 minutes to answer any questions regarding his study. If you have any questions, clarifications, comments. No, it's clear.
Thank you very much, Dr. Kodikara, for your presentation. It's a really interesting work that you have done. And I'm just curious, have you measured EH, redox potential, in these soils? What
No.
were the EH values? No?
Yeah, actually we are not measure the redox potential actually during our experiment, but pH and other soil properties we are maintained. Usually in our experimental soil, pH was around 7.5 nearly. Little alkaline soils are there. With the submergence, pH also little bit reduced actually compared to upland soils. Under submergence, it is near to the 7.
And secondly, if you have time, I would like to get some clarifications on your hypothesis. Like you had different phosphorus levels applied, and then you wanted to find out how the micronutrient availability varies with different levels of phosphorus.
Yeah.
Could you explain the chemistry behind that, like how phosphorus is affecting the micronutrient availability in submerged soils at least?
Yes, ma'am. Actually, I am going to that hypothesis due to my experience in upcountry related to Sri Lanka also. We have observed some specific places when we increase the phosphorus in soil, it will be badly affected to availability of some micronutrients as well as including calcium also. Therefore, I try to confirm it with my, this study, and actually here I am not presented it because there was not significantly differences there. Only water regimes affected to significantly to the concerned micronutrients, but in other nutrients and other properties significantly affected with the high phosphorus. But here micronutrients was not affected significantly. However, majority of micronutrients, they are showing decreasing trend with increase in the phosphorus. We observed that, but it was not significant.
It was not significant.
Yeah.
Thank you very much.
Thank you.
Okay, thank you very much, Dr. Kodikara. I think we need to stop there because to manage the time. And
Thank
yes, thank you very much for your presentation. Give round of applause. Okay, thank you. And we will move to the second presentation, in fact, rather third presentation of the session. So that is on plant growth promoting Rhizobacteria consortia for growth and yield improvement of rice. So it will be by Ms. WRKD WKV Vikramasinghe and D. Giriraj and K.S. Gopal. And perhaps this may be also part of the postgraduate study. And over to you, Ms. Vikramasinghe.
Very good morning to all of you. All of you are welcome to my presentation. Plant growth promoting Rhizobacteria are beneficial bacteria that colonize the plant rhizosphere and exert a lot of benefit effects to the plant growth through biological nitrogen fixation, solubilization in nutrient in soil, producing plant growth regulators, and protecting the plant by phytopathogens and improving the soil structures. The usage of plant growth promoting Rhizobacteria for increasing the plant productivity is one of the possible way to reducing chemical fertilizer usage. Recently, many scientists have apply multi-strains of PGPR because of then plant can take a lot of benefits from their beneficial effects. With that, I would like to introduce my topic, plant growth promoting rhizobacterial consortia for growth and yield improvement in rice. Myself and Professor D. Giricha and Professor Surendra Gopal from Kerala Agricultural University. Our objective was to evaluate the effect of novel PGPR-based consortia in plants' growth and yield of rice. So, before going to this experiment methodology, I would like to explain how we developed these novel consortia. All of these strains were isolated rice rhizosphere soils in Wayanad district by using different array of medium. For using Jensen Sega, we isolated nitrogen fixes, Picovaskia for P solubilizers, Alexandro for K solubilizers, and mineral salt medium amended with zinc oxide for zinc solubilizers. After isolation, the predominant isolates were purified and maintained. Then they were screened for their PGP activities such as production of ammonia, IAA, Sider4, HCN, and also they checked for their antagonistic activity against Rhizoctonia solani and Pseudomonas oryzae. And after that, these are all isolates under each categories were subject to their efficient character. For example, nitrogen fixers were subject to their nitrogen fixation efficiency by using microgeldal method, and for phosphorus solubilization efficiency by using more blue method like this, potassium and zinc efficiency by standard procedure measured. Then we rank the under each category our isolates by their functional ability and considering other PGP activities also, and most predominant 10 isolates were used for the consortial development. So before consortial development, we checked their compatibility by using cross-streaking method and confirmed by the dual culture method, and 3 PGPR consortia were developed. You can see in this table what are the components in the 3 different consortia. Each consortium consisted 5 strains, 2 nitrogen fixers, 1 P solubilizers, 1 potassium solubilizers, and 1 zinc solubilizers. After that, these 3 PGPR-based consortia were evaluated on rice growth and yield under pot culture and selected 2 superior consortia for further evaluation on the field evaluation. Today we are going to present the field evaluation of these 2 selected consortia. So next move today experiment methodology. It's having 5 treatments with 4 replicates. We conducted the research at Regional Agriculture Research Station Ambalavayal in Wayanad district. The crop used traditional variety Valichuri. It is 4 to 4.5 months rice crop. These are the treatment. The consortium 2 with 75% recommended dosage of NPK, consortium 3 with 75% recommended dosage of NPK. These 2 are we selected from the pot culture previously, and PGPR mix 1 with 75% NPK. The PGPR mix 1 is a commercial available biofertilizer in India, Kerala state in India. It was developed by Kerala Agricultural University. And T4 is a 100% recommended dosage of NPK. And farmer practice we use organic package of practice in the KAU, 5 tons for farmyard manure per hectare. These are the details. When we are using Biofertilizer, these rates are used at the seed treatment, at transplanting 10% solution dip 30 minutes, and field application, we use 2.5 kilogram of biofertilizer mixed with 100 kilogram organic, and field application were done. These same rates were applied for our novel PGPR consortia. These are the details. I already mentioned the consortium 2 and 3, what are the microorganisms included. And the PGR mix 1, this is a commercial fertilizer. It has 2 nitrogen fixer, Azospirillum lipopherem and Acidobacter crochrome, P solubilizer, Bacillus megatherium, and the K solubilizer, Pseudomonas. There is no any zinc solubilizer. Then we maintain 4 different nurseries with seed treatment, 3 biofertilizer treated and 1 uninculated, 4 nurseries. And at the plot size of 5 by 4, we transplanted after 21 days of seed. 21 days seedlings were transplanted. At that time, we done seedling dipping with 10% solution of biofertilizer for 30 minutes and transplanted. And also, they were feed application were done every month, 3 times with biofertilizer, the rate of 2.5 kilogram with 100 kilogram organic matter. Every 1 month after, we isolate the rhizosphere bacteria by using arrow of medium. For total bacteria, we use nutrient agar. For nitrogen fixers, Jensen agar. P solubilizers, Picovaskia, potassium alexandra, and zinc solubilizer using mineral salt medium amended with 1% zinc oxide. Then nutrient analysis of rice plants were done at flowering stage, nitrogen, P, potassium, and zinc by using these standard methods. And soil physiochemical and microbial biomass carbon analysis, the methods shown in that table. For organic carbon, Walkie and Black method, microbial biomass, fumigation extraction method, and available nitrogen alkaline permanganate method, etc. These standard methods were used for these analyses. Next, move the results and discussion. So before planting, initial microbial population and physiochemical properties were analyzed. This table shows the initial microbial populations, the total bacteria for 4.5, 10 to the power 7, and nitrogen fixers 8, 10 to the power 8, and phosphate 1.5, like that. And when we consider in the soil, it is low of organic matter, P, and K, sufficient amount in zinc. So this is the effect of PGPR application on the population of total bacteria in rhizosphere soil. You can see with all the treatment, every month we can see higher population in PGPR, that means biofertilizer treated treatment rather than T4. 100% recommended dosage of fertilizer. In farmer practice, organic matter, applied plots also showed higher total bacteria population in 2 months, after 2 months up to this you can see here the graph. Here also we can see all the 4 months. After 4 months, they maintain a higher bacterial population, T1, T2, and T3 treatment than the T4. Then these are the nitrogen fixers. Nitrogen fixers are significantly different in the after 2 months after planting and the 4 months after planting with biofertilizer treated plots than the T4 and T5. So you can hear also they maintain the all the 4 months higher population than T4 and T5. This is the population of phosphate solubilizers. Phosphate solubilizers also high in our biofertilizer treated plots, but we can see The phosphate solubilizer drastically reduced the after 4 months after planting. However, it was maintained higher population than initial phosphate solubilizer in the soil with that biofertilizer treated soil. This is the K solubilizers. K solubilizers also higher in the biofertilizer treated soil than the other two treatments, and they maintain significantly higher population throughout the growth period. And zinc solubilizer, interestingly, their population was high in our novel PGPR based fertilizer. The PGPR mix one also show lower population than these two. You can see here the population will maintain a higher level with this, but however, the comparatively the T3, our native PGPR consortia will maintain higher population. So next move to these are the field evaluation of our consortia and different treatments. And these are the effect of PGPR application on growth and yield of rice. There is no significant differences in the plant height. The number of tillers were higher in T4, that means 100% recommended dosage of fertilizer, and T1, and on par with the T2. And number of panicle per square meter also higher in that T4, and I can't see, sir. Number of panicles per square meter also high in T1 and T2. It is on par with the 100% recommended dosage of fertilizer, and grain yield also high in T1 and T4, and 1000 grain yield were higher all the T1, T2, T3 and T4 treatments than the T5. This is the harvesting stage of rice plants with different treatments. Then we analyze the nutrient content of plant tissues. The nitrogen content were higher in PGPR based 2 consortia and T4, it's on par, but PGPR mix 1 and farmers practice low nitrogen content, and phosphorus also we can see the same, only T1 and T4 on statistically on par. The potassium were higher in all the PGPR biofertilizer treated treatments, and 100% they are on par with than the farmer practice. Things was not significantly different. Then this is the effect of PGPR applications on nutrient content in soil at harvesting. There is no significant differences in soil organic matter, but when we consider microbial biomass carbon, we got higher microbial biomass carbon in biofertilizer treated treatment and the farmer practice, and nitrogen were high in that biofertilizer treated plots statistically on par with T4 treatment, that means 100% recommended dosage of fertilizer. In here we can see T3, that means PGPR mix 1 and T1 is significantly higher phosphorus levels given than also the 100% RDF, and potassium high in T3 and T4, that is the commercial PGPR mix 1 and the T4. Zinc content was not statistically different. So we can summarize our field experiments result. Combined application of biofertilizer with 75% recommended dosage of fertilizer maintain population of total bacteria, nitrogen fixers, phosphate and potassium solubilizers at higher level than uninoculated treatments throughout the growth period. Population of zinc solubilizer, that is with native PGPR biofertilizer had higher zinc solubilizer population. Then combined application of native PGPR based consortium with 75% chemical fertilizer improved plant growth and yield and help reduce 25% chemical fertilizer usage. Then we can conclude our result. New PGPR-based consortia increase nutrient uptake, growth, and yield of rice under field experiments. The native PGPR-based consortium 2 helps to reduce 25% NP and K fertilizer of recommendation without affecting the rice yield and nutrient content of plants and soil. The consortium 2 consisted Bacillus species AKNF3, Pseudomonas potida KGNF1, Bacillus megaterium, Acinetobacter chalcosticus, and Cytobacillus kochi. This application of PGPR in consortium with synergistic bacteria having multiple plant growth promoting traits such as nitrogen fixation, PK and zinc solubilization, IA production could be used for the production in a sustainable and environmentally safe manner. I have to say, we also analyzed their efficiency. So Bacillus species gave 9.33 milligram nitrogen per 1 gram sucrose in the in vitro, and Pseudomonas putida gave 8.33 milligram nitrogen fixed by per 1 gram carbon source. Bacillus megaterium was our highest P solubilizer among our isolate. It was about 134 milligram per the milliliter, and in addition to that, this Bacillus and Pseudomonas has IA production ability, and Pseudomonas putida has the antagonistic activity against the Xanthomonas oryzae. These are the, in addition to their efficiency of the other than N fixation, PK and zinc solubilization. Therefore, the findings of this research could be used for development of an eco-friendly biofertilizer for rice cultivation. Further studies in various soil types, plant genotype, climatic conditions are necessary to get conclusive results. I acknowledge my major advisor, eminent Professor Dr. D. Girija, Department of Agric Biology, and the Professor Surendra Gopal, Head of the Department, and all the advisory committee members, and Dean College of Agriculture, Ambalavala, for providing all the facilities to conducting field trial, and Sri Lankan CARP for providing financial support. Thank you very much.
Okay, thank you very much, Ms. Vikramasinghe, and you have taken 4 minutes extra. I think we'll have only 1 minute for the discussion, but anyway, let's have a couple of questions. There may be some. It's as usual, a sad situation.
I have one question from you. In Sri Lanka, we are in a division in, of course, information in the field of biology and microbiology. So based on your research, is there a potential for introducing this technology for local conditions?
Yes, sir. Surely. This is a very simple technology. Microbiology, that is the oldest technology in the science. So these are simply isolated from, we need only special media and quantify their efficiency. Because our soil has a lot of biodiversity. There is so many species. What we have to do, we have to isolate and quantify their abilities to do this nitrogen fixation, PK and zinc, and also other PGP activities. Because now the trend is use of multifaceted characters microorganism for development of biofertilizer. The one organism can fix the nitrogen, and also it can solubilize the P, and also that strain can produce the IA production and maybe siderophore production. There is a lot of PGP activities with some isolates. For identify some like these isolates, helpful to develop biofertilizer.
Okay, thank you. Any other? Just one question? Yes. Can you give the microphone, please? No, we can't hear you.
Optimum conditions.
No, can you repeat the question again, please?
The it's very simply, I would like to ask from the researcher this, have you any experience or develop the, understand the situation, the good optimum conditions in the, and the adipic factors for the optimal growth of these microorganisms?
Yes, it has depend on the strain, no? When we are isolated from the rice rhizosphere, there are optimum conditions for the submerged conditions. When we are isolated from the vegetable crops or upland, their optimum conditions may be that aerobic conditions. We cannot say Bacillus megatherium we can isolate from only from the rice rhizosphere or only from the some vegetable, tomato rhizosphere. Everywhere we can see Bacillus megatherium, Pseudomonas. What we are going to do, we are isolate native strains. That's the thing, because when we are isolated from rice rhizosphere, they are colonized better in the rice rhizosphere than other crops, because these microorganisms are attracted to the rhizosphere because of their phenolic compounds that exudates on the roots like that. So if we want to develop biofertilizer for rice, one factor we have to consider whether it is isolated from the rice rhizosphere. They can colonize better than the other strains. I can say like that. We can't say this bacteria has like the optimum condition is that like, so, okay. I think you get clear.
Okay, thank you very much. I think we have limited time. We need to stop the session before the tea and also next technical session and to be started in scheduled time. Okay, anyway, thank you very much, Ms. Vikramasinghe, and a round of applause to her as well. Thank you. Okay, this will be the last presentation of the session. That is done by Dr. Abethilakaratne, my old friend, and K. Dayani, SMA Samanmali, Kahavandala, and Ratna Kumara. So, a lot of co-authors. And Dr. Abethilakaratne will speak on effect of genotype, growing medium, and pot size on potato mini-tuber production using vermicompost, leachate in soilless culture. So, over to you, Dr. Abethilakaratne.
Good morning, everyone. In this morning, I am going to present the effect of genotype, growing medium, and pot size on potato mini-tuber production using vermicompost leachate in soilless culture. Potato is considered as the fourth major food crop in the world, and Sri Lanka also it is very important food crop and living many people in the upcountry area. Actually, we use the mini-tuber produce in the seed production to fast multiplication. Our recent studies reveal that the cost of cultivation is increased 2.5-fold, and 25% cost for the seeds and the other 25% cost for the seeds. There's a more tragic condition in the upcountry area. This will severely affect the resource poor farmers. Therefore, sustainable agriculture development is the very important, and we use vermicompost leachates to produce seeds at very low cost method. I'll move to the methodology. 2 experiments were conducted at the Agriculture Research Station Seethalia during the last year and this year, and 3 potato lines were tested, and SCP-2101 and SCP-2102 are recently developed in the Agriculture Research Station Seethalia because many potatoes imported from the other countries. These granola variety used as a standard variety for testing the other 2 variety. In the experiment 1, these three potato lines were grown with the wormy leachate, and tuberization, stolon formation, and bulking were studied after rogging out at 30 days after planting to 80 days after planting. In the second experiment, these three varieties were grown using the two pot sizes, 16 centimeter height pot and 22 centimeter height pot. And we use the 5 growing media, and normally use the coir sand mixture 2:1 is normally used in soilless culture in our research station, and other for coir sand vermicompost 2:1 ratio, partially burnt paddias refused 3:1 ratio, partially burnt paddias vermicompost 1:1 ratio, and vermicompost we use, and irrigated with the diluted vermicompost leachate. We use the plastic barrel to prepare the vermicompost, and usually we place crops residues and cattle manure 1:1 ratio, and we added the 250 Asian earthworms. Parameters are chemical parameters of vermicompost and vermicompost leachate is actually phosphate 1.87 in the vermicompost, but in the vermicompost leachate not detected the phosphate. And potash and nitrogen also very low in the vermicompost leachate. I'll move to the research finding. Actually, other studies reveal that the vermicompost leachate is a good one, and it has many nutrients. But our vermicompost has the more than enough phosphate and potash for the potato cultivation. I'll move to the research finding of the experiment 1. And the days to plant emergence is not statistically significant in the genotypes. In the plant heights, higher plant heights were observed in the second genotype, 2101 type. And after the 14 days after planting, heights is not changed. Stem number also higher in third genotype, 2102, and with the age, not change stem number. Compound limbs also higher in the third genotype, 2102 line, and this also not change with the harvesting age. Home fresh weight, very low in the granola check variety than the other two varieties. And this was increased up to the 60 days after planting, and decline when 18 days after planting. Home dry weight also get the same pattern and very low in the granola, and this also increase up to 70 days after planting and reduce the after that. Number of stolons not significantly different with the genotype and the age of harvesting, and the length of stolon also same. Number of tuber is higher in new 2 lines than the granola. And after the 14 days after planting, all the tubers are initiated and not change, the significantly not change. And tuber yield also lower in the granola, and others 2 are very high. And tuber yield gradually increase up to the 18 days after planting. And there is an interaction also, and the SCP-2101 rapidly increase the tuber weight than the 2102. And granola slowly increase the tuber weight. I move to the research finding of the second experiment. Plant heights also in second genotype 2101 line is high than the other tubes. And the growing media in the graph middle in the growing media and the right sides in the pot size, and not significant difference with the pot size and the different growing medias. Number of compound leaves also high in the G2 second line and 2101 line. And with the growing media, it's not change the compound leaves, but large pot, higher compound leaves than the small pots. Number of stem also higher in the 2101 genotype than other tools, but these are not changed with the growing medias, but higher number of stem found in the large pot than the small pot. Number of tube also higher in the second genotype, 2101 line than the other two lines, and this number of tube also not change with the soilless medias, but pot type, higher large pot is a higher number of tubers. Tuber yield also not difference with the genotype granola 2101 and 2102. But growing media also not difference is not significantly different. And the large pot size have the higher yield than the small pot. When we consider the average tuber weight, higher yield found in the granola, average weight of the tuber, higher weight found in the granola and the 2102 lines. And the 2101 line has the lower tuber weight. When we consider growing media, coir sand media and coir sand vermicompost media produce the very heavy tubers than other media, vermicompost, and the paddy has vermicompost, and paddy has refuse tea media. And average tuber weight do not increase with the or change with the pot size. In conclusion, with the vermicompost leachate, SCP-2101 produced the more tubers than the other two variety 2102 granola. And the large pot is very suitable to produce the seeds because more mini tubers and high yield observed in the large pot than the small pot. Therefore, our commercial cultivation also, next year onward, we use the large pots for the growing potatoes. When we consider 5 growing media mixtures, these 5 growing media mixtures are suitable for the mini tuber production. And the coir sand and coir sand vermicompost growing medias are suitable for producing the heavy mini tubers producing wood. And the tuber yield was great in 80 days after planting. And we would like to acknowledge the Horticulture Research and Development Institute Gannoria for providing the analysis of the vermicompost and vermicompost leachate. And with that, I would like to conclude my presentation. Thank you for your attention.
Thank you very much, Dr. Abe Tilagaratne. Sometimes I used to forget his name. And yes, you made your presentation and save a lot of time. So really nice to see that one. And yes, now you may have questions. So he allow us to ask more questions, I think, giving more time for questions.
May I ask a question? Thank you very much for your interesting presentation. And I was wondering whether you compared this with soil as a medium, and also with your, if you did not, with your experience, how is the yield parameters vary with having soil compared to what you have here as the soilless cultures?
In early, where there is a system, geophonic system produced in the soil, but there is a lot of diseases in the soil, and these soil system now not used, and in the soil-less culture, not use the soil media. And if we use the sand or any inorganic compound, actually we call as a hydroponic. If we use the organic material, coir or other thing, it's called as a soilless culture. In this soilless culture, we didn't use the soil to check.
Yeah, but with your experience, do you think if you use soil, would the yield change and how it would
No,
change?
no, but we use, now there's a research, ongoing research in agriculture research in Italy. They use the low-cost method as a soil-based system, directly apical cuttings, planting in the soil system, but they are fail to initiate. And other coir and sand media, very healthy growth. I think these are very balanced in the aeration and other water holding, or these things. And
So
very poor growth in the soil media.
Okay, thank you. And I assume you added fertilizer equally for all these except the vermicompost, did you?
Yes.
Okay.
Yes.
Thank you. Any more questions, please? No. Look like. Yes, I just want to know, because I am out of the date. About 6 years ago, I think I came to know all these things. And so granola still a popular variety, or how about the other recommended varieties by the Department of Agriculture responding to this mini tuber technology now? You have new varieties as well, because sometimes at that time, I think there's a claim by the farmers and the extension say that the reason for not popularing some of these recommended varieties that you don't get seeds or some other issues related to this one. But timely needed experiment. Just educate me on this, please.
Granola variety, last year farmers grown about 65%. But this year the scenario is changed, because the imported variety seed price is increased up to the 700 rupees per kilogram, from 400 to 700 rupees. Now, it's still the department produce granola variety, it's still the same last year price selling. Therefore, farmers purchasing, most of the farmers purchasing the granola in this year. And this year, I think the imported variety may not grown by the farmers because of the expensiveness of the seeds. And our new varieties, therefore, our new varieties also produce, and these tested system perform very well in our new varieties. And we hope to release in near future this variety after the testing in the multi-locational trials.
Okay, thank you very much. Thank you very much, Dr. B. Tilakaratne. I think if there are no any other questions or comments, so I think we'll go for the final remark. Before that, I think Dr. Abe Tilakaratne need a round of applause, please, for his excellent work. So, right, now we had 4 presentation, 1 review work, and 3 research work. And anyway, congratulations to all the presenters for their scientific work, especially Dr. Ratnayak gave a very good comprehensive review on the recommendation and fertilizers management, nutrient management in rice sector. But we need to think and beyond that ones to address the emerging issues of the country. I think how the Department of Agriculture is going to, address those issues as timely manner. Then the other two presentations are on their postgraduate studies, and I feel that they have learned a lot of technical matters and also how to conduct scientific study in a proper manner. Those are basically, I feel that the postgraduate studies are to make them more what is called to have a scientific approach. So I think they have gained it. I think those information, I think they will make use in our country, especially in relation to the biofertilizers. The studies by Ms. Vikramasinghe, the biofertilizers, I think, need to identify the local strains, and it's not appropriate, I think, not permitted to get the foreign or alien microorganisms to the country. But need to identify on the local conditions and see whether they are effective, especially the microorganism in relation to phosphorus solubilizing. And nitrogen, of course, we can manage in a different way sometimes, but nitrogen as well, but not much on potassium for me, I think, but basically phosphorus. It's the burning issue. It's going to be, I think our soils have sufficient amount of phosphorus in most of the soils. If you can find a suitable strain to solubilize the fixed phosphorus, so that would be really helpful in terms of country-wise and in terms of the agriculture sector-wise as well. But Dr. Kodikara's one is known, most of the informations are known, but it is very difficult one. The interaction of micronutrient with phosphorus, especially the availability of phosphorus. And he also used two conditions, the aerobic and the flooded conditions. And these are two extreme conditions we are affecting on the phosphorus availability. Because when you flood it, you change a lot of factors like pH, electrical conductive, EH and so on. So that would really affecting on the phosphorus solubilizing, very complicated study, but he has done a good job. And finally, Dr. Abhay Tilakaratne's presentation is on mini-tuber production, especially to see the numbers and the weight of mini-tubers in different medias, and try to address the limit of mini-tuber production. So to give a kind of a solution to the mini-tuber production as well. So anyway, excellent work, excellent presentations. I think they all need, again, a round of applause, because this is the only forum I feel that, we need, we appreciate all the scientists within the Department of Agriculture. I think all of them deserve the round of applause again. Don't be hesitate to appreciate your colleagues. And yes, then finally, just a word. In fact, I take this opportunity to thank the organizing committee of Asda. So I came to Asda after maybe 5 years. So there's a policy on mine regarding certain things by me. And if you don't have any work, don't peep in. And anyway, thank you very much, the Director General of Agriculture and Asda Organizing Committee, for inviting me to chair this session. I really enjoy, after many years with the colleagues of the Department of Agriculture, some familiar faces and some unfamiliar faces. Maybe the mind is not now not like those days now. Maybe dementia sometimes. Anyway, really enjoy the session, and thank you very much. And I wish all of you good deliberations today, fruitful for the country, and yes, be a member of the Department of Agriculture, be firm on your technical matters. Anyway, thank you very much. And so passing the microphone to the organizing committee. Thank you.
Yes, thank you very much, sir. And there's another important event, as our chairperson explained at the beginning. All the presentations except invited presentation done by Dr. Upul Kumar Ratnayaka were evaluated and the best presenter will be selected. So our eminent judging panel have done that job, and I would like to thank our judging panel, Mr. K.M. Kandaragama, Mr. Prasad Idame Korala, and Dr. Chammi Attanayaka for your duty. And yes, we got the result. So it's time to hand over the certificate of the best presenter in the session. I would like to invite Dr. W.M.A.D.B. Vikramasinghe to hand over the certificate to the best presenter of the session. The best presenter of the session is Dr. K.M.S. Kodikara. Thank you, sir. Thank you very much. And there's a small token of appreciation to the panel of judges and the chairperson. First, I would like to invite Dr. Vikramasinghe to hand over the token of appreciation to the panel of judges. Mr. K.M.A. Kandaragama, Mr. Prasad Idame Korala, and Dr. Chammi Attanayaka, please come and take your token of appreciation from the chairperson. Sir, please. Please hand over the token of appreciation to the... Yes. So wait for a moment. Finally, I would like to invite Mrs. Priyanjani Madana, Provincial Director, North Central Province, to hand over the token of appreciation to the chairperson of the session. So please. Now you have. Thank you, madam. Thank you, sir. And with this, I will wind up the technical session of the Asda 2022, Soil, a Challenge and an Opportunity for Sustainable Development. And I would like to take this opportunity to express my sincere gratitude on behalf of the Executive Committee of Asda to the Chairperson, Dr. Vikramasinghe, invited speaker, Dr. Upul Ratnayaka, and panel of judges to share their valuable time with us with your busy schedule. And express my sincere gratitude to all the presenters and the audience for being with us. Thank you very much. And also there's one small housekeeping notice. After this session, you can have the refreshment. Refreshment will be served in the lobby. And the next session will be started sharp at 11 o'clock. So please be in your seats on time. Thank you very much.
Ladies and gentlemen, please come and take your seat for the start next session. a keynote speaker. Ladies and gentlemen, please come and take your seat for the start next session. We have to start it sharp at 11 because invited presenters is looking for the session. So welcome to the next session. I am very pleased to welcome you all to the technical session, Smart Plant Protection, Reflect Your Heart. First of all, I would like to welcome our panel of judges, as traditionally 3 members are joined with us to evaluate presentations. Then, I pleased to welcome invited speaker, Dr. Wade Jenner, Center Director, Cabby, Switzerland, and Deputy Program Executive, Plant-Wise Plus, to the technical session. We are indeed honored to have you here with Sir. Now, I would like to introduce session coordinator to the audience. She has graduated from University of Peradeniya in Agriculture Honored Degree and obtained her PhD from Imperial College, University of London. She has more than 30 years of experience in Department of Agriculture. Over that time, she served to the department as Deputy Director, Additional Director, and Director HODI, and finally retired as a Director General of Agriculture. She is a member of many professional organizations and reviewer to the many scientific publications. She also provides consultants to the different institutes and has more than 30 publications locally and internationally. Please welcome our chairperson, Dr. Mrs. Dinadiri De Soisa, to the session Smart Plant Protection, Asda 2022. Dear Madam, I am cordially invite you to chair the session and wish successful sessions. Thank you.
Thank you, Rukmali. This session starts now, and we have a invited presentation by Dr. Wade Jenner, Deputy Program Executive, Plant-Wise Plus Center, Director, CABI, Switzerland. Dr. Jenner has professional experiences, and from 2009 to 2012, Integrated Crop Management Advisor, CABI, Switzerland. 2012 to 2016, Plant-Wise Program Support Manager, CABI, Switzerland. 2016 to 2021, Global Director for Health Systems Development, CABI, Switzerland. 2021 up to present, Center Director, CABI, Switzerland, and Plant-Wise Program Deputy Executive. He has obtained a degree in Biological Sciences in Augustana University College in Camrose, Alberta, Canada, and MSc in Behavioral Ecology and Biological Control at Simon Fraser University in Vancouver, British Columbia, Canada, and PhD in Parasitoid Ecology and Biological Control at Carleton University in Ottawa, Ontario, Canada. So he has many contributions to the scientific literature on a lot of subjects with one of our researchers as well, food information, plant health, and other subjects. So over to you, Dr. Wade Jenner, and his presentation will be on putting IPM principles into practice, lessons from a global program on plant health. Over to you, Dr. Wade Jenner.
Right, yes, thank you for the introduction. It is a real pleasure to be a part of your Asda Symposium this year. I've heard about this symposium for years. So I'll need to ask that everyone else on the Zoom call would mute their microphones and turn off their speakers. I'm getting some feedback here. I think it's better now. As I was saying, I've heard for years about the Asda, and it's a real pleasure to be a part of this this year. Yes, I will talk about putting IPM principles into practice, lessons learned from a global program on plant health. This is maybe a bit different from the typical presentations in this session, whereas I'm coming now from more of a social perspective, looking at how we change behavior. I'll be talking about the Plant-Wise program, which has been a more than 10-year global program run by CABI. First, I'd like to just say a word about CABI itself. This may be a new name for some of you in the room. So CABI means the Center for Agriculture and Bioscience International. We're a not-for-profit intergovernmental organization established by a United Nations level agreement. And what this means is that we're actually owned by our member countries. And Sri Lanka is one of our member countries, which has an equal role in the organization's governance, policies, and strategic direction. As a matter of fact, we have our major conference, our review conference with member country representation happening next week in the UK. CABI addresses issues of concern such as food security, food safety, and environmental health through both research and international development cooperation. And in addition to that work on the ground, we're also a major publisher of scientific information in different forms. Okay, now coming to the PlantWise program, I'll give a little introduction to this. I know many of you in the room are already familiar with this, but I'll assume not everyone. So PlantWise is a program that CABI has led for over a decade on a global scale, looking to increase food security as well as food safety and improving rural livelihoods in reducing crop losses. So here's just a little diagram of where we've been with the PlantWise program. Of course, Sri Lanka was a part of the family of PlantWise. The PlantWise approach, as we call it, refers to the concept of the program implementation. And one thing I'd like to point out here is that the permanent crop clinic program in Sri Lanka is the manifestation of the PlantWise approach in Sri Lanka. So very, very early on in the implementation of PlantWise, this was really adopted by the Department of Agriculture, and it was given its own name. And I should mention that I'll be talking a lot about plant clinics. Of course, the term in Sri Lanka is crop clinic, which is the same thing, but just out of habit, I'll be calling these plant clinics, and that's the term that you'll see in the slides. So in this schematic on the slide and on the screen here, it shows sort of a cyclic nature to the processes under the PlantWise approach. So most importantly, and what we see most visibly on the ground, is this process between farmers and plant doctors, where there's a service provided at what we call the plant clinic, where farmers bring their problems to the plant doctors, who then give advice on the spot to the farmers. So this involves making a diagnosis of the problem and making a recommendation of some sort. Now, what's special about the PlantWise approach is that it doesn't stop there. So there's a process of collecting information through that interaction between the farmer and the plant doctor that feeds into some sort of a central database that can then be used for other purposes. And in PlantWise, we call this the knowledge bank, the PlantWise knowledge bank. So clinic data would be submitted to the knowledge bank, which is then compiled and cleaned as necessary, analyzed by different experts in the country. And that can then feed back to plant doctors to help with learning, developing new strategies to implementing the program or the approach. And in addition, there can be other uses of this data, such as surveillance for new pests in the country, monitoring the activities of plant doctors both on the level of have activities taken place, or going deeper into the data to look at what kind of problems and recommendations are being given at clinics. This again then can feed back into the process of communicating to farmers by posting pest alerts or by informing what kinds of priorities should be taken up by extension services in a given period of time. So this is the PlantWise approach, which will be referred to throughout my presentation. Now, the plant clinic or crop clinic concept is common throughout all the PlantWise countries. So I'll just show a few slides here to make that point. So here's a plant clinic in China. Here's one in Western Africa in Sierra Leone. Here's one in Bolivia in South America. And here's one from many years ago in Sri Lanka. So it always involves a process of consultation between farmers who come with their problems, and it could be actual physical samples of a plant that has a disease or maybe an insect sample. And the plant doctor makes a diagnosis and provides a recommendation. And so CABI has worked with our partners, such as the Department of Agriculture and the provincial departments in Sri Lanka, to build capacity of extension workers, possibly other advisors in the country, to provide this basic frontline service. PlantWise is based on a few core principles. So one of these is strong stakeholder linkages. So in everything that we did through PlantWise, we tried as much as possible to link different actors in the plant health system. Again, the plant health system is referring to the collection of stakeholders involved in supporting farmers. And this could be directly through advisory services or the provision of inputs, or maybe more indirectly through research and regulatory services. Another key principle of PlantWise was ensuring quality advice. The PlantWise program was very much about information exchange, making sure that information is available to those who need it, so with the ultimate target being farmers, and looking at how we can not only deliver advice, but ensure that this is based on science-based information. And as mentioned, we promoted very much a two-way information flow. It wasn't only about pushing information to the farmers, but using that interaction as an opportunity to provide intelligence to other stakeholders in the system to inform their decisions and planning. And lastly, and related to this, is the equitable accessibility. This is referring to information. How can we make information more easily available? And this could be through digital tools, which of course is becoming more and more relevant every year, but also through the services provided physically by extension services, how to make that outreach as inclusive as possible. Now, as mentioned already, the PlantWise approach involved a lot of capacity building that CABI initiated, but of course, as much as possible, we tried to hand that role over to our partners. And through PlantWise, we worked with a number of different stakeholders in countries. And the collection of our partners in a country varied from country to country. So always at the base of it was extension and plant protection. Although to different extents, we may have linked with different diagnostic or research services, or possibly with agro-input suppliers, both the intermediaries as well as the retailers at village level. In some countries, we worked very closely with farmer or community-based organizations, both for running the actual plant clinics themselves or for helping to organize and plan how such services should be provided. We worked a lot with post-secondary educational institutions. One of the things that we've done was to integrate the plant doctor training that we provide through PlantWise into standard post-secondary education. And finally, in a number of countries, we worked with different NGOs. And again, not only CABI working independently with these organizations, but trying to facilitate the collaboration amongst them within a country. And that then creates a stronger plant health system. So, as mentioned, we did a lot of training of plant doctors through PlantWise. Over the roughly 10 years of the program, there were more than 15,000 trained across the countries in Asia, Africa, and America. And Sri Lanka was no exception to this. We had, I believe, at least 1,000 extension staff trained in the modules for plant doctor training. And supporting that, of course, was the development of reference materials, as we call it. So these could be fact sheets or in this case here, we call this a pest management decision guide. And these are documents that help an advisor to be aware of the options available for dealing with particular problems. And as mentioned on the slide here, always with an IPM focus. So PlantWise was very much, not entirely, but very, very much focused on plant protection. So looking at pathogens, insect pests, or weeds, and how to manage those sustainably. When we talk about the outcomes and the impact of PlantWise, we always envisaged two primary pathways, and I'm showing just one at the moment here on the bottom of the slide. We call this the plant clinic advice adoption pathway. So once we have plant clinics running by plant doctors, as farmers visit these clinics, they receive advice on the spot in real time. This is advice that the farmer can use on her or his farm to deal with the problem concerned. And of course, the impact comes through the application of the advice, leading to, ideally, an increase in yield, so higher productivity, and therefore higher income. And this is the most visible part of PlantWise. This is what we often see in the reporting and the documentation of the program. The less visible pathway but equally important one is what we call the plant health system change pathway. And this comes back to that point of linking stakeholders. How can we increase the efficiency in the way that the different organizations linked to plant health interact and deliver better services for farmers? And the catalyst, the lead-in to this pathway for us through the PlantWise approach was primarily the clinic data. So the information that's gathered by plant doctors at field level can feed into decision-making processes. And this can be done really analytically through a database, or it could also be done more informally through meetings between stakeholders. But an example of this could be the detection of new pests. And we have evidence through PlantWise over the last decade in various countries where through the role and the responsibility of advisors, of extension workers in doing this general surveillance for pests, there have been cases of new pests detected and reported, and then leading to action from the respective authorities in the country. And this could be in the form of eradication, an immediate eradication of a brand new pest in a country, or it could be through awareness raising to ensure that both the farmers and the broader public are aware. So this might mean a quarantine pest must be controlled through the restricted movement of goods in the country or further into the country from outside. And that then has also an indirect link to farmers changing their behavior. So both pathways ultimately lead to reduced pest damage and therefore increased productivity at field level. So with these two pathways, there's then the challenge of addressing how do we measure our success. So PlantWise, again, is very much about disseminating information, trying to find the most effective ways to do this. And so in the PlantWise approach, we employ a variety of outreach methods, ranging from one-to-one interactions between farmers and advisors at a plant clinic, for instance, up to mass communication through popular media like radio and television. And throughout this, the campaigning that we've done has, as I said, almost always been about plant protection, and therefore we always promote very strongly an IPM approach. But how exactly do we measure that success in promoting IPM? We know that we've reached a lot of farmers through PlantWise globally for this roughly 10-year period. We could say that we've somehow reached more than 55 million farmers globally. And based on the data that we've received from partners in Sri Lanka, for Sri Lanka alone, this is probably around 3 million, in particular through the operation of the crop clinics and the PCCP, as well as some plant health rallies and other larger dissemination of information through radio and so on. I'd like to break down this number, though, because there's sometimes a misunderstanding that this reach would all be through plant clinics. As a matter of fact, plant clinics don't reach that many people compared to things like mass extension campaigns. So of the 55 million globally, approximately 2 million were reached directly at plant clinics, farmer physically attending a plant clinic. And then through our programming, roughly 500,000 were reached through what we call plant health rallies. These are brief interactions in the public to talk about a particular problem. And then through the mass extension campaigns, here we've reached nearly 10 million based on our estimates, because this is not face-to-face, this is through radio and other means. Now, what's really interesting is that through different studies, we've assessed the level of farmer-to-farmer sharing, and it's very clear this is a very important part of the dissemination of information. Now, in PlantWise, this wasn't a method of communication that we explicitly addressed or promoted. So we worked mostly with intermediaries, such as many of you in the room, to look at how can we get information out to farmers in the first place. But it's always worthwhile to note the level of sharing between farmers. And in general, on average, we could say that farmers tend to share information that they've received, if they deem it valuable, with another 4 farmers. This is a number that we find keep coming up in our studies. On average, about 4 farmers receiving information from their fellow farmers. Now, of course, these are very different communication channels, and they have different degrees of success in promoting adoption of new practices. And one thing I'll point out here is that when it comes to farmer sharing, while it happens, we need to be aware that through the what we call it the telephone game, every time you pass information through another intermediary, the message gets a bit distorted. And this definitely does happen. We did a social network analysis in China and Zambia about 7 years ago, which showed that while they do share information, it's usually only parts of it and sometimes quite distorted. This is a graph that we like to show quite often when talking about our plant-wise approach. Shown here on the y-axis, y-axis is reach. Quite often, the more you reach, so when you reach out through radio and television, you can reach thousands or millions of people, but there tends to be a lower impact. To achieve higher impact, you tend to need a more intensive interaction with the farmers. And so what this graph shows is that along this continuum, you have different approaches to outreach with things like radio at one extreme end, and then regular on-farm consultation or farmer field schools, which are very time intensive and very, very personal with farmers at the other end of the spectrum. And here you achieve typically higher impact. And so throughout the plant-wise program, we were always very much aware of this trend and trying to think, okay, we could say that a particular service like SMS tends to fall somewhere around here, but are there ways that we could push that further to the right on the impact pathway in using it as a delivery mechanism? So of course, the key step between reach and impact is adoption. How do you convince farmers to pay attention to the message you're providing and take it seriously and actually apply the advice given? So how do you convince farmers to adopt IPM to achieve the impact that you desire? Well, one of the key points I'd like to make here is that it's very important to have a trusted source as the deliverer of the information. So in particular, independent advisors who are looking out for the interests of the farmers above all else tend to be among the most trusted by farmers. And this includes government extension agents typically. And so having them serve as plant doctors at plant clinics was a very obvious starting point for us in PlantWise when we introduced the program in a country. And I would like to maybe just say that this tends to be in contrast to maybe somebody who's giving advice, but also at the same time expecting to make a profit, a financial gain. And this is where we always felt that in the PlantWise program, there's a potential conflict of interest when agro-input dealers, for instance, are the source of information. A farmer needs to be very aware that this often comes with a bit of an expectation of a sale being made at the same time. And again, this came through in our studies where we interviewed farmers that among their most trusted advisors were government workers or other independent advisors. A challenge, though, is that there's simply a limit in how many people can be reached through such a service. Now, linked to that aspect of trust, what we did find in studies, and here we had a set of studies in 7 countries around 2015 to 2017, we saw really high rates of adoption by farmers on the advice given by plant doctors at clinics. So again, this is simply through interviewing farmers, asking them what advice they were given, and what was the result on their farm? Did they apply the information or the advice given? And typically, around 90% of farmers said that they adopt the advice given. And this is excellent news. Here is a clear way for us to influence the behavior of farmers. And I'll give one example. So in the PlantWise program, while we addressed food security in particular, we were always very much interested in promoting safe use of pesticides. And this includes being very selective on which pesticides to use. So we created what we call the red list pesticide, the PlantWise pesticide red list. And the red list pesticides are active ingredients that are considered to be acutely toxic to humans or subject to international conventions. And so on this list, we had all of the WHO Class 1A and Class 1B active ingredients, as well as those listed under the Stockholm Convention on Persistent Organic Pollutants, Montreal Protocol on Substances that Deplete the Ozone Layer, and the Rotterdam Convention on Prior Informed Consent. So our take on this was, let's try to avoid these when giving advice. Let's try to encourage farmers to look more to greener alternatives, or at least less hazardous ones, for their own health as well as for the health of the general public. Now, in this slide, what we're showing here is the proportion of recommendations given by plant doctors. This is observed through the clinic data collected. The proportion of recommendations that contain a red list pesticide. So in other words, higher is bad, a lower number is good. Now, I need to mention this is global data. This is not just Sri Lanka. So Sri Lanka is a part of this, but a relatively small part because there are more than 30 countries that make up this data set. But what we observed in early years of the program was that always around 3% of the recommendations given included at least one so-called red list pesticide. Now, 3% is a nice low number. That's good. Of course, ideally, we'd have this at 0. And I'll just go into a bit of explanation here. So in 2017, for instance, 3% of farmers was equivalent to about 300,000 queries or recommendations given at plant clinics. Because again, we're talking about an annual reach of about 250,000 farmers in general. So in 2017, there were roughly 300,000 farmers who would have received advice that included a red list pesticide. At the end of 2017 and going to 2018, we made a concerted effort with our partners across all plant-based countries to really be aware of red list pesticides and to try to phase these out from the recommendations being given. And what we saw was an excellent result. There was an immediate decrease beginning in 2018, which continued throughout to 2020. And this was effectively a 50% reduction. And again, we're talking about quite small proportions. It's a change from roughly 3% down to about 1.5%. But that is actually quite significant when you look at the absolute figure. This is a change of around 150,000 farmers no longer receiving advice to use red list pesticides. And we can trust that this had some impact on what farmers were actually using because we see from our other studies that they do adopt the advice given by plant doctors as a trusted source. And of course, the adoption of plant clinic advice does bring impact. That's of course very important. So we also did many studies to assess what is the impact in terms of yield or income for farmers. So I've just given a few examples here from Rwanda, Malawi, and Kenya, where we see 20% increases in yield for maize producers, or 20% for tomato in Malawi. In Kenya, there was a massive increase. This is because it coincided with the introduction of the fall armyworm in Africa. So it was a bit of an exceptional year, but the good advice led to those farmers attending clinics having far higher yields than those who didn't. And as mentioned in the point here, this means a higher income for those farmers going to plant clinics and getting advice. Another interesting observation was that farmers who applied more methods to address pests, and in particular, this was a study on fall armyworm management from Zambia. So those farmers who applied more methods tended to do better in their production and income. So the more they applied an IPM approach, the more successful they were. Now we come a bit more into the social aspect. Who exactly is benefiting from plant clinics? Who's benefiting most? And again, I'm speaking globally, not only in terms of Sri Lanka here, but we do observe from the clinic data and through interviews with farmers that in most countries, there's a strong dominance of clinic users being men. And this is perhaps not surprising, although given the role of women in agriculture, where they make up around 50% of the agricultural workforce, it would be nice to see a stronger female participation in advisory services like plant clinics. And I will make a point that in Sri Lanka, certainly based on my own observations there, as well as looking at the data, the ratio of clinic users between men and women is more balanced than the average across plant-wise in general, but still a tendency for men to be more common users of plant clinics as a service. And the point that we always like to make in plant-wise is that this can be addressed, for instance, by considering the location of your clinic, the time of day, or the frequency of the clinic. Or very clearly, having female plant doctors also seems to encourage female farmers to come and seek the service as well. And one thing I do want to make is, one point I want to make is that this is not actually only relevant to plant clinics or crop clinics. This is to any service provided all of these considerations are important when planning an outreach activity. Now, one other point that we did observe through our studies is that not only do farmers benefit from using clinics, so those who use clinics have an increased improvement in their production, but at the beginning, plant clinic users tend to be slightly better off. That means they tend to have a slightly higher education. They might have a little bit more land. They might have better resources already at their disposal than those who don't use clinics. And what this tells us is that however it is that plant clinics are being run, they seem to either appeal or be more accessible to farmers with slightly more resources. And this is the kind of thing, again, that needs to be taken into consideration when thinking about where do we run clinics, who is involved in planning them and informing the public about it. And this is why we then come to the other pathway and how else can we reach farmers? And so in our other pathway, we had things like mass extension campaigns. This is using outreach methods like radio, television, SMS. So identifying a particular topic through the information gathered through plant clinics and other on-the-ground services to plan a particular campaign, let's say. And this, of course, is nothing new, nothing introduced by PlantWise, but we worked with our partners to try to plan these in very careful ways that would address the concerns, the constraints of farmers accessing this additional service. So what we saw from some studies on campaigns in Africa related to fall armyworm management is that the exposure to a campaign, and again, this could be radio, it could be video, this did stimulate the adoption of agricultural technologies that were introduced by, for instance, the government. Radio reached the most people. This is not surprising, I suppose. And also not surprising that another approach like using video screenings at village level had a greater impact on farmer knowledge. I think there's something much more engaging about a visual mechanism of communicating. But what was very clear from our studies is that exposure to multiple approaches led to the greatest possible positive change in knowledge and adoption. This is why it's important to think about not only a one-off campaign or approach, but looking at a combination of outreach methods on a particular topic. So we see very clearly that while mass communication has its limitations because there's no opportunity for back and forth exchange as you have in a personal interaction, it can still be very effective if designed and executed well. And one very clear thing that we learned is that a mass campaign must be very simple and clear in its messaging, preferably focusing on just a single problem, and ideally one that people are searching for a solution already. So it's something that they need rather than trying to push something entirely new on them that they weren't necessarily asking for. And as mentioned before, it's important to consider multiple ways of getting information to those who need it. So my last slide, and this is a parting thought. While it's very important to get the technical message right and to ensure that the advice given is scientifically sound, it's extremely important to also consider the human aspect to the dissemination of information. This is a key factor in ensuring that we can make an influence on the final outcome of the adoption of the advice. And one thing that we've learned as an organization over the past 10 to 15 years is that yes, it's important to have technical experts involved, but involving experts in things like gender and behavioral change can revolutionize the way that an organization delivers its services and the impact that it can have on a country. So with that, I'll say thank you. And I hope that was clear throughout and that there were no problems with the video sharing at all.
Thank you, Dr. Jenner, for your nice Thank you, Dr. Jenner, for your nice presentation on PlantWise program, putting IPM principles into practice, lessons from a global program on plant health. So we actually invite our participants to get any clarification on this. We have 5 minutes for you. One question from me, Dr. Jenner. Now, have you done any impact
I'm sorry, I
study?
can't.
Have you?
I'm sorry, I can't hear your audio.
Can you hear now?
No.
No, it's so.
Your voice was. There we are. That's here. That's here. That's here. That's here. That's here. That's here. That's here.
Can you hear now? Your audio. There may be a signal problem. Can you hear now? Anyway, we will wind up here with this presentation. Thank you very much, Dr. Jenner. We will contact you with this messaging.
Thank you to all. Yes, I'll be happy to take any questions afterwards by email or whatever. That is fine.
We will go on. We will go on to the next presentation. Of antifungal mechanism in lacto-fermented palm kernel cake against Coledotrichum gliosporidae by HRP Fernando, ASM Hussain, BJ Mujahideen, NL Yusuf, NK Mohammed, SH Afrin. The paper is presented by HRP Fernando.
Thank you very much, dear Chairperson Madam, my dear sirs and madams in judge panel, and dear ladies and gentlemen. I am going to present my research work about the optimization of antifungal activity and determination of antifungal mechanism of lacto-fermented palm kernel cake against Coledotrichum gliosporidae. This is the outline of my presentation. Introduction part. Anthracnose fungi, name is Coledotrichum gliosporidae, destroy the post-harvest quality and reduce the shelf life of many fruits and vegetables. Actually, chemical treatments used to control anthracnose in pre-harvest and post-harvest stages nowadays since several decades. However, has been developed social protest for the chemical treatments for the fruits and vegetables, especially fresh producers, due to its some hazardous effect and residual toxicity, carcinogenicity, genetic mutations, environmental pollution, and many reasons. Then our scientists paid their deep attention for the discover any sustainable solution for the preserved fruits and vegetables. They found it for the solution as biopreservation. Biopreservation we can define as the use of antagonistic microorganisms or their metabolic compounds to preserve food and feed. One of the famous antagonistic microorganism group is lactic acid bacteria. Lactic acid bacteria fermentation process is the very good procedure to develop antifungal compounds for the many pathogenic groups, especially bacteria, fungus, also protozoa. However, agricultural byproducts have a number of benefits to use fermentation substrates. In our previous research studies, we found, we studied about with some agricultural byproducts, namely rice bran, PKC, watermelon rinds, pineapple peel, and orange peel. However, finally we got the good result with palm kernel cake. PKC, short form of the palm kernel cake, fermented with Lactobacillus plantarum. It is a very famous lactic acid bacteria group. Exhibited the best antifungal activity against Colitrichum glysporiates. However, production of antifungal compounds through lactic acid bacteria fermentation depends on several factors such as pH, substrate type, substrate ratio, lactic acid bacteria strain, and many factors. Actually, industrial lactic acid bacteria fermentation is cost-effective process because the cost is very high in these days, the fermentation substrate and starter cultures and any other materials. In this study, Box-Bernken design under the response surface methodology was used to optimization procedure because the very important to reduce in cost and increase the antifungal activity of our final product. Furthermore, there are several different antifungal compounds can be contained in our optimized antifungal mixture, final product. Therefore, different antifungal compounds have different antifungal action mechanisms on fungi and also bacteria. Therefore, identification of mechanism is very important for the industrial level application. These are objectives my research work. First one, to optimize the fermentation conditions, obtaining maximum antifungal activity for lacto-fermented palm kernel cake with Lactobacillus plantarum. Secondly, to identify the related antifungal mechanism of optimized fungal growth inhibitors against Colitrichum glysporiates. This is methodology. First part, optimization of the antifungal compounds, actual cultures and substrate. Lactobacillus plantarum cultivated in high quality expeller pressed palm kernel cake PKC. Lactic acid bacteria culture was grown in MRS broth and Colitrichum glysporiates growth on PDA. This is the procedure to producing culture and substrate. This is the optimization procedure Box-Bernken design under the response surface methodology we use as the response optimizer. Independent variables and levels fermentation conditions are here in the table. We got 3 independent variables: substrate ratio, fermentation time, and temperature. As well, coded and actual values we can see here, response parameters. These lower and upper levels selected by the series of preliminary optimization studies and literature. This is the lowest level, medium level, and higher level, upper levels of our parameters. The optimizing model was developed using Minitab 17 statistical software, and the second degree polynomial of the design was expressed using the following formula. This is the formula we used for the develop our model. Determining the response parameters, we got 4 response parameters. First one, antifungal activity, total peptide concentration, pH value, and total phenolic content. These are the very important criteria, otherwise parameters for the against Colletotrichum gracisporiate fungi. Therefore, we studied with these four parameters. However, after that second part, determination of antifungal mechanism, for explain the antifungal mechanism, we find out the inhibition of conidiac germination. Secondly, preparation, this procedure to preparation of fungal mycelia for the further assays. Third one, intercellular leakage, especially total soluble protein, total sugars, and DNA leakage we measured with this procedure. Total glycerol concentration, because fungi is very important glycerol for their cells and other cell membranes process. Finally, morphological changes in treated and untreated fungal mycelia. Statistical analysis, we use statistical analysis, optimization. For the optimization, statistical significance of the regression equation of tested using F-test. The ANOVA was performed to determine whether the quadratic models are significant. P-values were used for the check the significance of coefficient. Antifungal mechanism, RCBD, was used for the experimental design. The data was subjected to analyze to ANOVA with using the SAS 9.4 package, and means compared with using the Turkey multiple comparison test with confidence levels 0.05. Results and discussion. This is first part, optimization study. Final models explaining the relationship between the response parameters and the independent variables are shown in figure equations 1 to 4. These are the equations for the response parameters with our model to develop our model. The regression equations predicted values are presented in below. These are the coded value. After that process, these one the independent variables, different independent variables for 15 runs. After that, these are the response variables, Y1 pH value, Y2 peptide concentration, Y3 phenolic content, and Y4 Colletotrichum gracisporiate inhibition. The significance of the fit of second order polynomial of pH value, total phenolic content, peptide content, and percentage of Colletotrichum gracisporiates was evaluated implementing the analysis of ANOVA. We can see clearly in this table the main effect X1, X2, and X3, and quadratic levels others 3, and interactions of them. However, we can see here the p-values of all main effect in significant level of all response parameters. In here we can see lack of fit values all insignificant, and R-square values nearly 1. Then we can suggest that this model, we developed model is adequate for the our objective, and this model is the accuracy for to optimizing our antifungal activity. As evidence, the surface plots provide the interactions among the independent variables. This is the first target, our lowering the pH, reduce the pH, because some organic acid highly depend on the fungi and bacteria. Therefore, we try to reduce the pH in our product. This is the how to affect the substrate ratio and temperature for the pH. Then the second picture, fermentation time and temperature, how affect the pH. Third graph, substrate ratio and fermentation time, how affect to the pH value. However, finally, we got the lower pH value in these conditions. This second target, peptide concentration. Third target, Colletotrichum gracisporiate inhibition. These three targets, mainly these three targets, we achieved with our final product using this model. These are the response optimizer for the optimum conditions of PKCl fermentation with Lactobacillus plantarum. We can see here substrate ratio is the 24.74. Fermentation time is 96 hours and temperature is 37 centigrade. Under these conditions, we got the good pH value, maximum antifungal activity, peptide maximum concentration, and TPC maximum concentration. This is the validation of the model. Finally, we use the 4-sample t-test for the validation. We can see here all parameters experimental value and fitted values. EV means experimental value, FE means fitted value. These are, after compare with t-test, p-value is insignificant, all p-values. It means this model is correct and accuracy for our work. Antifungal mechanism result, inhibition of conidia germination. We use the control and 50% MIC, minimum inhibition concentration and minimum inhibition concentration. We can see here the significant levels of compare with control. 50% MIC and MIC reduced almost conidia germination of the Clostridium glysporides fungi. Inhibition of agastrol synthesis, second graph. This is the agastrol synthesis. Negative control is nearly 65 micrograms per milliliter, but positive control and PKCl1, this is our product, reduce the production of agastrol. However, agastrol is the key component of the fungi. They cannot survive without agastrol. This is the effect of fungal treatments on intercellular leakages. First column, soluble protein concentration, second column, total sugar concentration, and third column, DNA concentration. We can see here positive control and negative control in higher, our product and positive control, higher leakages of sugar, total sugars, protein and DNA, but negative control, it means without treatments, very low intercellular leakages of the fungi. This is the morphological changes of treated fungi. We can see here, this is the healthy fungal filament without any treatments. This second picture is the treated mycelia with our product. This third picture is the one conidia and collapsed mycelia filaments after treated fungi. Cells were collapsed, cracking and shrinkage and swelling. In addition, some of the mycelia were enlarged. The cell wall has been undergone the partially swelling. This is the conclusion of my study finally. Box-Burnken design under response surface methodology was effectively applied to optimize the fermentation conditions for enhance the antifungal activity of PKCl1. The substrate ratio, fermentation time and temperature are influenced to the produce antifungal compounds. The combinations of optimum fermentation conditions were the substrate ratio of 24/7/75, fermentation time 96 hours, and fermentation temperature is 37 degrees centigrade. The inhibition of Clostridium glysporides by PKCl1 was observed 80%, 8%, 0.8. However, finally, we can conclude the PKCl was affected to the cell membrane, cell structure, cell wall, and biosynthesis pathways of Clostridium glysporides. My acknowledgement, I would like to my sincere thankful to Sri Lanka Council for Agriculture Research Quality for funding my studies, and Ministry of Agriculture, University Putra Malaysia, and supported everyone for my Food Research Unit Institute, Department of Agriculture, my directors and everybody. This is reference. Thank you very much. Thank you, dear sir and madam.
Thank you, Dr. Fernando. Now, paper is open for discussion. 5 minutes discussion.
Dr. Fernando, you used high quality expeller cakes, your studies.
Yes, sir.
What do you mean by high quality, and what are the parameters you
yes, sir.
have seen? And also, why not the normal expeller cakes?
Yes, sir. There was using two methods for extracting palm kernel oil. First one, chemical extraction. This chemical extracted palm kernel cakes is not suitable for the like these studies because they are mixed with some chemical. Chemicals also have some antifungal and antibacterial effect. Therefore, I selected to expeller pressed palm kernel cake. It is the physical methods for the pressing the extraction oil. This high quality means mean value, very high impurity, high purity without any impurities, and 100% farm kernel cake.
Thank you.
One clarification, Dr. Fernando. Is it oil palm or coconut palm?
Oil palm, madam.
Okay, right.
Yes.
Any other question?
Dr. Pranandu, congratulations.
Thank you, madam.
It's a very important study, I think. And is it in the practical stage now, or do you have to
Yes,
develop
madam.
it more for practice use?
Yes, madam, this is one of the middle part of my study. Finally, we developed one antifungal coating product. We can use this product with many biopolymers. Example, chitosan, maltodextrin, potato dextrose, potato starch, inulin. We can make coating, coating with using these antifungal compounds. However, we used to, with chitosan, they give good result, and we have one Malaysian patent also for this product.
Okay. I just want to know how is it been going to use practically? Is it sprayed onto the fruits?
Yes,
or?
madam, both can. First method is very clear method, dip in. We suggest to with conveyor belt after washing and drying mango fruits. Then we can dip in with antifungal coating bath, going through the antifungal coating bath. Secondly, we can spray.
Okay, thank you.
Thank you.
I have another question also.
Yes, sir.
How you compare with the physical treatment, physical method like hot water dipping and this method? Which one is more effective to control?
Yes, sir. These are actually very old method, hot water treatments and some thing. Actually, those are effective. It is I cannot say it is not effective, but many hot water treatments combine with chemicals, not only hot water treatment. I have used a long time with some mango companies, growers in Sri Lanka, like Nelna. They use some hot water treatments with together chemical treatments, example Benomil, like that. But actually very low successful with only hot water treatment. Second one, the mango, especially TJC, developed lenticell damage with hot water treatment. Then I try to develop another biopreservation method for the this solution.
Okay, Dr. Fernando, now the time is up. So thank you very much for your excellent presentation.
Thank you very much.
The next paper is on bioefficacy of selected novel insecticides for the management of whiteflies and aphids in bitter gourd and brinjal in Sri Lanka by J.P. Marasinghe, R.J.K.N.P. Samarasinghe, S.R.K. Madugalla, L. Nugaliyadda, K.S. Hemachandra, and S.H.P.P. Karunaratna. Presented by J.P. Marasinghe.
Thank you,
Over
Madam.
to you, Ms. Maras.
Thank you, Madam. Good afternoon, Madam Chair, distinguished guests and colleagues. I am going to present my study on bioefficacy of selected novel insecticides for the management of whiteflies and aphids in bitter gourd and brinjal in Sri Lanka. And this is one part of a series of studies, and the background is that there were plenty of farmer complaints about the poor performance of pesticides against whiteflies and aphids, and also there were related issues encountered by the extension staff of the DOA revealed at PTWG meetings. And in addition, the widespread of vector-borne plant viruses indicate the controlled failure of whiteflies and aphid vectors. The whiteflies and aphids, the prominence as major pests, the two major whitefly species are Bemisia tabaci and Trialurodes vaporiorum. And Bemisia tabaci is included in the 100 worst invasive pest list by IUCN. And Mycus persicae is the most resistant aphid species worldwide, and all three of them inhabit brinjal and bitter gourd. And they transmit different types of plant viruses and suck sap from leaves and reduce the vigor of plants in severe outbreaks, and also secrete honeydew that supports sutimol growth and resulting reducing the photosynthesis. Whiteflies and aphids are difficult to control due to many reasons. They lay a large number of eggs and have a large progeny and have several generations per year, and they are polyphagous, can fly and also disperse through the wind, and can evolve as different genetic forms or virulent types. And most importantly, they have mechanisms to develop resistance against insecticides. And our recent studies reveal that the development of resistance against the recommended insecticides is one of the underlying causes for control failure of these pests. And whiteflies and aphids have developed resistance up to different levels against several recommended pesticides. However, insecticides are undoubtedly the major pest control method at present, and DOA has continuously recommended insecticides belonging to different groups for whitefly and aphid control. Several recommendations against whiteflies have been withdrawn with the evident resistance information, and there was a need to introduce novel insecticides, preferably with new modes of action. And what is the mode of action? The process of how an insecticide works on an insect at a molecular level is the mode of action. And why is it good to know the mode of action? Because knowing the mode of action of an insecticide is key to the resistance management. And these are the different mode of actions. Insecticides can be categorized into different major classes based on their mode of action. And just to give you a brief idea on how the insecticides function in an insect, they act on target proteins and alter the target proteins, and in return the biological responses are altered. And the key functional proteins that are targeted by insecticides are ion channels, enzymes and receptors in insect bodies. And there were several compounds with novel mode of action that have been newly introduced to Sri Lanka during the study period. There were sulfoxafluopyridifuron, pymetrozine, pyrifluquinazole and spiromesiphene. And talking about sulfoxafluo and fluopyridifuron, they belong to 4C sulfoxamines and 4D butyronolates. They are neuromuscular poisons acting on nicotinic acetylcholine receptors. And in normal condition, when an impulse is transmitted, neurotransmitters, that is like acetylcholine, bridge the synapses, and acetylcholine is released in response to the nerve impulse and activates the nicotinic acetylcholine receptors. These sulfoxafluo and fluopyridifuron are competitive modulators of nicotinyl acetylcholine receptors, and they bind to the sites of acetylcholine and then desensitize the receptor. Therefore, hypersensitization occurs and then paralyze occurs and the insect dies. And so these chemicals, neonicotinoids and all these are neuromuscular poisons. However, depending on the site they bind and how strong they bind, their action could vary. And pymetrozine and pyrifluconazole are 9B pyridine azomethine derivatives. They are also neuromuscular poisons, but they act on TRPV channels that exist in specialized sensory neurons that detect stretch. That is in antenna and then in joints in arthropods. So the locomotion of the insect can decoordinate, and also rapid feeding cessation occurs due to TRPV modulators. So these two are TRPV modulators. And the last one is pyromazepine, that is 23 tetronic and tetramic acid derivative. This is different from the all other previous told insecticides. This is a growth and development disruptor. And insects have exoskeleton that contain chitin, and exoskeleton does not expand, and it needs to be replaced by a larger one at molting, and need chitin synthesis, which is controlled by the growth hormones. And this insecticide act on acetyl-CoA carboxylase enzyme, which is required for lipid biosynthesis, and because of the prevention of lipid biosynthesis, incomplete molting occurs and the insects die. So the objectives of this study was to test these new insecticides with relatively new modes of action against whiteflies and aphids in agricultural crops, and to make recommendations for their suitability for sustainable management of these pests. This was a bioefficacy evaluation study with a series of field trials against whiteflies and aphids, and conducted from 2015 to 18. And each insecticide was tested in 3 consecutive seasons with the randomized complete block design with 3 or 4 replicates. And sulfoxaflor and pymetrozine were the first to be tested from 2015 Yala, 15-16 Maha, and 16 Yala at farmer field at Mathale. With bitter gourd variety was used, and the DOA recommendations were practiced. And the second set was flupyridifuron, pyrifluconazole, and spiromazepine from 2017 Yala, 17-18 Maha, and 18 Yala in the same place with brinjal varieties. And this is the treatment schedule using sulfoxaflor and pymetrozine, and the rate of applications and dosages. The dilution rates are given here, and the tested seasons are given. And for the other 3 insecticides, these rates were used, and during these seasons they were conducted. And the 2 spray applications were conducted, commenced after 4 weeks of planting, and the second spray after 7 to 10 day intervals. And the whitefly and aphid counts were taken just before spraying and 7 to 10 days after pesticide application. And the number of insects in 5 leaves per plant in 10 randomly selected plants were counted. The data were analyzed. Pre and post counts were separately analyzed using analysis of variance, and means were separated, and the percentage efficacies were calculated using this simple formula. And also the average % efficacy of insecticides were calculated. Accordingly, this table shows the bioefficacy test results for sulfoxaflor and pymetrozine. And these graphs show the same, and can be explained better than by the table. And you can see in the first, in each treatment, the first column shows the pre-count and the second one, the post-count, and the third column, the third bar, the percentage efficacy. And all the pre-counts were non-significant, therefore I didn't give the values. The post-counts on the bar, you can see the letters that differs from the control, the letter in the control, so that gives a significant value. And the percentage control is given on the third bar. And you can see in 2015 and 16 Maha and 16 Yala, the untreated control gave a negative effect with very high population of insects. This is for whiteflies. And this table gives the counts, aphid counts in bitter gourd field trials. And these graphs show how the percentage efficacies vary in between the treatments and the untreated controls. And this one gives the bioefficacy evaluation of the other 3 insecticides conducted from 2017 Yala to 2018 Yala. And due to the lack of extent of lands to accommodate all the insecticides, we had to split the trials and do it in separate trials. Therefore, in 2017 and 18 Maha, we had 2 trials, and also in 18 Yala, we had 2 other trials. And similar to the previous one, the pre-counts, post-counts, and the efficacy, percentage efficacy are given in these graphs. And this one gives the results of the efficacy of on aphids. And these graphs show the effectiveness similar to the previous one. And the average percentage efficacies were calculated using the effectiveness of each season. So against whiteflies and aphids, you can see how much effective each insecticides were, and they were compared with the global data reported in literature. And accordingly, pyrifluquinazone and flupyridifuron have exhibited a similar effectiveness against local whiteflies and aphids compared to the global literature at comparable rates of application. And 2 out of 3 pymetrozine products that gave slightly lower efficacies than the third against aphids is expected to be due to the differences in solubilities or manual mixing errors. The efficacy against aphids of the best performed pymetrozine product was comparable with global data. Then efficacy of spiromesifen was moderate. However, the effect on immature stages was not counted because it acts on immature stages of whiteflies as well. And the average efficacy of sulfoxaflor against the aphids were comparatively low during the tested seasons in 2015 and 16. However, during 2017 to 18, as a treated control, it gave a higher efficacy comparable to the global data. And these are special characteristics of these products. Sulfoxaflor is has high aphicidal activity according to the literature, and has systemic and contact action, complex and unique interaction with the nicotinyl acetylcholine receptors, and works through contact and through ingestion as well, translaminar and xylem mobile. And likewise, these insecticides have different specific characteristics. And the conclusions are all 5 insecticides gave significant results compared to the untreated controls or treated controls. The overall efficacy against two insect groups was at different levels in each of the insecticide. Considering the overall effectiveness and specific product characteristics including the mode of action, all 5 insecticides can be used at the tested rates for successful control of whiteflies and aphids in brinjal and bitter gourd. However, use in alternative terms, considering the mode of action, is important to delay the resistance development in targeted pests. And suggestions for insecticide resistance management. Well-planned insecticide resistance management program is needed. A combination of insecticides with different modes of action per season is suggested with the only maximum of 2 applications per 1 mode of action. An integration of other management practices is vital as appropriate. For example, physical barriers, as we have tested and presented last year. These net barriers can be used to control whiteflies and aphids in bitter gourd. And some other different management practices can be integrated with this one. And I acknowledge the studies team and Mr. Ranjit Padmasiri and Ms. Sandamali Dayavans of ODI and the farmer, Mr. Andra, and the relevant pesticide registrants. Thank you.
Thank you very much, Ms. Marasinghe. Now, the paper is open for your discussion. If you have any question or clarification, please ask. Ms. Marasinghe, now are these insecticides recommended?
Yes, madam. 3 of them are recommended and 2 others are under evaluation. The technical dossiers are under evaluation.
Okay. If there are no other questions, we will wind up for her excellent presentation.
Thank you.
Now, the next presentation is on effectiveness of selected post-harvest treatments for population suppression of grain moth and rice weevil on stored paddy by S.R. Sarachandra, and there are about 12 other officers, a very, very big team, and with scientists working here. So I am not going to tell all these names to reduce the time. Anyway, I will congratulate that kind of a large team work together for a scientific work. So over to you, Mr. Sarachandra.
Good afternoon, Chairman, Madam, Sir and Madam, panel of judges, ladies and gentlemen. All are welcome to presentations of our team on effectiveness of selected post-harvest treatment for population suppression of grain moth, Cetotroga cereella, and rice weevil, Cytophilus oryzae, on stored paddy. This is the outline of our presentation. First, we move into the introduction parts. Rice is stable food more than 3.5 billion people around the world. Rice very susceptible to post-harvest losses. Insect causing significant post-harvest losses of grains, 10% in the temperate regions, while 50% in the tropical regions. Cause direct and indirect losses by stored pest, major stored pest in paddy in our country, rice weevil, grain moth, almond moth, rice moth, lesser grain borer. Among them, rice weevil and grain moth are the most common and destructive pest in the storage facility in our country. Rice weevil, Coleoptera, a weevil attacks seed of several crops, rice, wheat and maize. grain moth, Lepidoptera, a moth, feed on seeds of rice, wheat, barley, maize, and sorghum. Insect infestations cause both qualitative and quantitative loss of storage paddy, loss of seed viability, loss of nutrients, loss of weight, contaminations with live and dead insects or their body parts and excreta, webbing. Those are the damage by the major post-harvest pest in paddy storage. Therefore, post-harvest treatment are essential to reduce losses associated with insect pests in storage facility. This is the physical, chemical and non-chemical methods can be used for that. Actually, this is a practical issues, very big issues in the farmer level and large scale storage facilities. There are many complaints. At the moment, no any recommended insecticide for stored pest in paddy in our country. Therefore, we have to develop sustainable integrated approach to solve the farmer problems. Therefore, we plan to this study to find out the integrated approach to minimize the avoidance of the post-harvest losses, as well as in farmer level and storage facility, they use many chemical, illegal, unrecommended chemical for these problems to direct contaminate with the grains. This is the illegal applications. Therefore, we try to minimize and avoidance that problems. The objective of study to evaluate the susceptible level of selected rice varieties for major rice pests in our storage facility, to evaluate the efficacy of selected post-harvest treatment, manage storage pest in rice. Then we move into the materials and methods. Evaluations of most vulnerable paddy variety for Cerealla and Oryzae infestations in our storage facility. Experimental site, rice warehouse located at RRDI Batalagoda. Experimental design was complete randomized designs with 3 replicates. Treatment 1, 8311, treatment 2, 8303, treatment 3, 8362, and DG300. Those are the tested varieties for this study. 20 kilograms of paddy for each replicate we use this study. The infestations level of Cerealla and Oryzae were monitored 2 weeks interval within the 6-month period. The natural infestations of Cerealla and Oryzae were recorded separately from 500 grams of representative sample for replicates twice a month along with the 6-month storage period. Then evaluations of different treatment for control of Cerealla and Oryzae in 8311 under storage conditions. Experimental site, same location, and experiment design was RCBD with 3 replicates. 8311 was stored under 29 temperature, ambient temperature, and 67 relative humidity. Dried to reduce moisture content up to 13 to 14%, and amount of paddy for 10 kilograms per use for this experiment. This is the second part of the study. The treatment, 7 treatment with 3 replicates were used for this experiment. 2 weeks interval sun drying, first one. Second one, 4 weeks interval sun drying. Third one, air dried citrus and neem leaves treatments. Fourth one, tricyclam hydrogen oxalates. Fifth one, acetamiprid. Sixth one, deltamethrin and untreated control. Those are the treatment for this study. Treatment 1 and 2 subjected to continuous sun drying at 2 weeks and 4 weeks interval. Treatment 3, 15 grams per kilogram of air dried citrus leaves and 25 grams per kilogram of air dried neem leaves were added into the each replicate of treatment 3. Treatment 4 and 5, 6 treatments were treated with tricyclam hydrogen oxalate, acetamiprid, deltamethrin respectively. Cotton cloth bag were used for all treatment to store paddy. Empty cotton bag treated with insecticide, tricyclam hydrogen oxalate, acetamiprid and deltamethrin. Each bag was immersed in the selected chemical solutions for 10 minutes to facilitate absorption. Then treated bag with air dried for 24 hours. After the drying process, measured paddy were filled separately, those bags. The natural infestations of Seriella and Oryzae were recorded separately from 500 gram of representative sample for replicates twice a month along with the 6-month storage period. Then this is the destructive sampling methods were used for this experiment, and number of dead and live Seriella and Oryzae were recorded as a data. Collected data were subjected to normality test, and data were analyzed according to the ANOVA procedure using a statistical package, and mean were compared to LSD test. Now we move into the results and discussion parts. This is the important part of this study. To evaluate most vulnerable paddy variety for Seriella and Oryzae infestations, 8311 showed significant higher infestations with Seriella and Oryzae. Then BG300 variety had significant lowest infestations of Seriella and Oryzae compared with the other tested varieties. Then second highest pest infestations was showed in 8303, which had no significant difference among 8362. So same results both variety. This is the average number of live grains moth count in different rice varieties. This very clearly show that the highest and lowest variety. Second one, average number of live rice weevil count in different rice varieties in the 6-month time period. Then evaluations of different treatment for control of Seriella and Oryzae in 8311 under storage conditions. This is the 6-month time period data, tested treatments, and this is the clearly show that how to variate the populations build up in the storage bag in different treatments. So live weevil count under different treatments over time 6-month period. This is the 7 treatments vary with the time period. Their populations build up inside the storage bag. Next one, dead grain moth count under different treatments in the 6-month time period. Then dead live counts in different treatments in the 6-month period. How to vary with the 7 treatments for a 6-month period. According to the results, chemical treatments show a great suppressance of populations build up both species compared with other 2 treatments. For chemical treatments, deltamethrin showed higher level of suppression of populations establishment of both species, Seriella and Oryzae, compared with the other 2 chemical treatments. Thiazide hydrogen oxalate and acetamiprid equally effective to population suppressions of both Seriella and Oryzae species in the 6-month period. Thiazide hydrogen oxalate in 69% suppressions, in Seriella, Oryzae 82% suppressions by the thiacycline hydrogen oxalate, acetamiprid 66.86% suppressions of the Seriella species in the tested period, and also 82% suppressions of the Oryzae in the storage period in the acetamiprid. 2 weeks interval sun-drying methods was effective than 4 weeks sun-drying methods. The air-dried citrus and neem leaves treatments show the least effect of the population's establishment of both species. Tested treatments, botanical products are the least effectiveness of the tested period for both species, Oryzae and Seriella. Then conclusion, 8311 was the most susceptible paddy variety in stored pest tested varieties. BG300 was the highest tolerant variety to growth and development of Seriella and Oryzae in both species in the paddy storage. Sun-drying of harvested paddy at 2 weeks interval showed higher efficacy against pest infestations than 4 weeks interval sun-drying methods. Then the botanical treatments as applications of air-dried citrus and neem leaves are effective than the control treatments. compare with the control treatments, botanical treatments also effective than the control treatments, but tested other treatments compare with the botanicals, lower efficacy recorded the tested period. This is the take-home message for farmer level and storage facility. 2 weeks interval sun-drying methods can be suggested in small-scale paddy storage, mainly for seed purpose and consumption. During commercial production, the chemical treatment thiazide hydroxylate acetamiprid can be practiced for bagging material to obtain proper protection from the stored pest. Because already no any chemical recommendations, but we comparatively safe chemical, two safe chemical selected for this study, and deltamethrin class 2 chemical, comparatively toxic chemical, but only we suggested bagging material, dip with the bagging material for before storage the paddy. Therefore, no direct contaminate with the chemical for our paddy, therefore no harmful effect for this procedure. But practical situations, farmers and large storage facility owners apply chemical direct for the grains. It is very harmful effect. Therefore, these findings in future confirm another test. We can suggest that for the storage facility do these findings solve their problems. Small scale and consumption purpose, sun-drying methods, because it is a labor consuming methods, this is not practical for the large scale facilities. Therefore, in large scale facilities, we have to apply integrated approach with those chemicals we suggested. Thank you very much.
Thank you very much, Mr. Sarachandra. Now the paper is open for discussion. 5 minutes.
Mr. Sarachandra, can't we use polysac or gunny bags instead of cotton bags for the storage?
Yes, sir. First time we want to confirm what are the products or treatments effective. Therefore, we use cotton cloth bag for first time to find out the effective products. Then we have to apply polysac bag and outer applications. We have to confirm the effectiveness of at that time, because the gunny bag is okay, because gunny bag can absorb the chemical. It is okay, no any problems. But in polysac bag, there is a problem because no absorption. Therefore, we have to apply outer surface of the polysac. There, at this purpose or no any problems, because not direct contaminations with the grains. Therefore, no harmful effects. That trial will be tested in future applications method. These steps we try to find out the effective safer chemical or any other treatments for solving that problems. Therefore, in future we have to do another experiment, applications methods in outer layer of the polysac bag and how to efficacy of that time. Deltamethrin, madam, available, madam, under restricted applications. This is a class 2. Actually, deltamethrin is a class 2 comparatively toxic chemical, but broad spectrum chemical effective than the other two chemicals. Therefore, strictly apply only bagging material, no problems. But some farmers apply more toxic chemical direct to grain. It's a very harmful thing. Therefore, I think if there is a recommendations, we can minimize the misuse of the chemical and misuse of the illegal procedures to control the post-harvest pest. Therefore, recommendation should be needed for the minimize that problem.
Any other question? Mr. Sarachandra, why did you choose class 2 chemicals?
Madam, 2 chemicals, class 3.
Class 3, okay.
one chemical class 2 because that chemical broad spectrum and highly effective, madam, according to our experience, other crop as well. Therefore, we try to most effective one and other safer 2 chemicals, and most effective one is strictly used in the bagging material. No, madam, problem.
Broad spectrum means you are testing only for rice weevil, no?
Yes, madam. Rice weevil and grain moth. 2
grain
species,
moth,
madam.
okay.
Coleoptera and Lepidoptera.
Okay.
Thank you, madam.
Any other clarification or question? If not, we will wind up with saying showing your appreciation to him for his excellent presentation.
Thank you, madam. Thank you, judges, sir and madam, and ladies and gentlemen.
Next presentation is on pesticide use by rice farmers and presence of residues in rice grain, an evidence-based study in selected rice growing districts in Sri Lanka by P.W.I. Lakshani, C. Mahagamage, K.S. Hemachandra, M.K. L.K. Rajapaksha, L.A. Hemamali, W.P.A.P. Dhanapala, L.R.H.B. Kumari, and D.G.G.S. Dharmaratna. Paper is presented by P.W.I. Lakshani. Over to you, Lakshani.
Thank you. Good afternoon from Madam Chair and the judge panel and my dear colleagues. Today I am going to present about pesticide used by rice farmers and the presence of residues in the rice grain, an evidence-based study in selected rice growing districts in Sri Lanka. Here's the content. As introduction, rice is a staple food in Sri Lanka. Nearly 2.85 millions of metric tons consumed by domestic use, and it's most popular cereal crop and very important source of carbohydrate and protein. Per capita consumption is 107 kilograms. And now I move to what is pesticide residues. It refers to the pesticide that may remain in food after they are applied to food crops. So residue means not only a pesticide, it can be derivatives, metabolites and related oil compounds. And but the harmful thing is bioaccumulation of residue can build up harmful levels in the body as well as in the environment. So maximum residue level is a maximum concentration of the pesticide residues to be legally permitted on food, commodities and animal feeds recommended by the National Regulatory Authority or Codex. So objectives, first one is to quality assurance and method validation of pesticide according to the Sante guideline and use in catches, and then second one is to evaluate pesticide usage in selected district in Sri Lanka by farmer survey, and then third one to determine and compare the prominent pesticide residue in meal raw rice and selected location in Sri Lanka, and then compare and evaluated detected levels in pesticide residue in meal rice in selected district with FAO and Sri Lankan MRLs. So methodology, first selection area and pre-tested questionnaire, then selection of pesticide for the study, then third one is the analytical method validation and performance, then sample collection and pesticide residue analysis. So selection of area, in my research I selected 5 districts. In Anuradhapura, Rajangane, Nachaduwa and Galenbindunuwa, from Kurunagala, Ibagamu, Mahava and Variapola. In Ampara district I selected Lindawur, Akkarepattu and Mayaduna. In Polonnaruwa district, Giritare, Hinguragoda and Madirigiriya, and in Hambantota, Veeravilla, Yodakandi and Beralihala. So these are the selection areas. 60 randomly selected rice farmers were selected, and we collected information on educational level, usage of insecticide, fungicide and herbicide, and how they handle the pesticide. Likewise, pesticide mix in practice and identification of the pest and stage application of the pesticide and usage of the nozzles and as well as time of application. Nearly 342 pesticides now currently registered in Sri Lanka, but they categorize into 123 active ingredients. So these are the insecticide, fungicides and herbicide recommended. as DO recommendation. So this is the most important thing, analytical method validation and performance. It will take more than 6 months because there's very huge procedure for validation. We want a highly traceability certified reference material, and then chemical, only LC-MS, MS categories of acetonitrile, acetone and other chemicals, and apparatus. They are very cost. So this method validation have to be carried according to the SANTE guideline. It's SANTE 12682 2019 by European Commission for Guidance Document as Analytical Quality Control. So this is the instrument we use. We are very lucky to have an instrument in our central lab. So this is a Thermo TSQ Endura LC-MS MS system. In my previous research, I only validated 6 or 7 pesticides, but in that study, I validated 57 pesticides. But now it up to 127 pesticides. So here that machine is can divided into 2 parts. It's a HPLC system, here HPLC system, and this is a mass spectroscopy system. So it has mass analyzer with 3 quadrupole system. So we can analyze up to 1 ppb level. So it is legally permitted according to the EU requirement. So here's the UHP details. Like trial and errors, we have to build the methodology. So here we use Thermo Scientific Hyperseal Gold AQ column, and there are 2 mobile phase A, B. One is with water and formic acid, then other one is methanol with formic acid. And flow rate is 300 microliter per minute, and column compartment is go to 40 Celsius. Here there's a ramping and gradient program. So in MS part, there are several categories we have to feed. So likewise, this is the MS details of the instrument. So this is the most important one. This is the quantitation ion, confirmation ion and calibration range of selected pesticide. For 57 pesticide, we have to select what is the most suitable quantitation ion, confirmation ion, and what are the calibration range. Because we have to acquire R-squared as more than 0.999. So summary validation. So we have to acquire linearity with R-squared value. So the RST should be lower than 20%, and it had to be lower than MRL levels, and response and region blank and other control summers have to be lower than 30% by difference rate. And most important thing is average recovery for each spike level have to be 70% to 130%. So here we get OLA comply, so our method is validated. Here's the spectrum we got from the LC-MS. We can see 57 pesticides. Some pesticides have very good response and others have some lower response, but we have mass analyzer. From that we can confirm what is the pesticide presence in the sample. So here's the validity of results. We got accreditation till May 30th of May 2024. So we participate international FAPAS accreditation system. They send some samples for us. So we have to analyze and send the results within 1 month period. So in that FAPAS proficiency testing schemes, we got set score as 0.1. So it's a good score, so our results are valid. So sample collection, milling and storing. So all the sampling according to ISO 874 and the recommended methods of sampling by the FAO with compliance with MRL. So we have to composite the sample. So 6 to 2 paddy samples were prepared as raw rice using small scale milling machine. So there are a variety of rice was selected, but they all are selected randomly. So here are the sample details. Pesticide residue analysis consists with homogenization, extraction, cleanup, and identification. So there are several techniques, but nowadays clutches is the most famous thing. So it's a quick, easy, cheap, efficient, rush, and safe methods. Here we use AOC 2007-01 according to AOC 2012. So extraction procedure is homogenization of the sample and then weigh the 5 gram sample, then we have to add with 10 milliliter of acetonitrile and then vortex with 1 minute and centrifuge under 3000 RPM for 3 minutes and then transfer the supernatant layer and then it's have to be mixed with the PSA other material and centrifuge in under 3000 RPM and then transfer and filter to PTFE. So here's the catches extraction and cleanup some images. Research and discussion, plus I will move to survey analysis. So education level, 60% of primary education, while 35% of guiding education and only 5% secondary education. So usage of weedicide, 60 farmers use 30 numbers of generic pesticide formulation to control weeds, but prednisolone is the most famous one. So insecticide, the 6 numbers of farmers use 11 kinds of generic insecticide formulation, but carbosulfan and thiamethoxam are the most popular ones. And usage of fungicide, it's a marvelous 90% of farmers not use fungicide, and only the remaining 10% use tebuconazole. So here's the usage of tanks and nozzles. 75 farmers will not use separate tank, while 25% of farmers will use separate tanks and nozzles. And identification pests by farmers, farmers get instruction and agriculture officers and label instruction by 56%, while label instruction by 10% and instruction of salesmen by 25%, while other 9%. Pesticide mixing practice by farmers. Some farmers use premixtures while other tank mixtures, and here 73.3% of farmers use no mixtures. So pest identification methods, apply pesticide after pest incident was observed 53%, while farmers apply pesticide before the pest incident was observed 46.6%. Time of application pesticide, in the morning level 83 farmers applied pesticide, and in the evening 70% of farmers applied pesticide. So then we moved into research and discussion about sample analysis. I think this is the most important part, and you will wait for the results. So study samples collected from January to March in 2020. So only 6 pesticides were detected from that 62 samples. Only 6 pesticides, they are thiamethoxam, imidacloprid, carbofuran trihydroxy, and profenopos and buprofosine and tebuconazole. So, but none of the pesticides going more than Sri Lankan MRLO codex. So here's the pesticides. Only 3 samples get contaminated with thiamethoxam in Ampara Karipattu, and 2 samples contaminated with imidacloprid, and it's from Mantota Viravila and Ampara in the wood, and 2 samples carbofuran trihydroxy. It's some metabolite products of carbofuran. Really, it is not carbofuran. Sometimes it may be because carbofuran banned in 2015, and some of the metabolite can be remain in the soil. So that found in Anuradhapura, Rajangana and Galembiduna area, propanopos, but that's the thing, the propanopos is not recommended. So, but we found 3 samples contaminated with propanopos in Anuradhapura, Nachaduva and Ampara. Buprofosine in Ampara Karipattu, only 1 sample, tebuconazole, only 1 sample, in Ampara and in the wood. So multipesticide residue analysis, out of 62 samples, only 9 samples showed 12 detection, only 12 detection. It means 0.33% detection. It's a very low detection. So no pesticide detect samples available Polonnaru and Kurunagala district. So mean detected percentage of thiamethoxam, imidacloprid, propanopos, buprofosine, and the tebuconazole in all districts are 4.8, 3.2, 3.2, 4.8, 1.6, and 1.6 respectively. The highest contamination detected dry samples available at Ampara district, but it's only 108 ppb. They are ppb levels. Neither any samples exceeded the FAO nor Sri Lankan MRLs. So conclusion, 56.7 farmers used prednisolone weedicide, while 26.3 used carbosulfan methoxam as insecticides, and about 90 farmers were not using fungicide, and 93% of farmers use information from agriculture instructors. 73.3 farmers do not practice either tank or pre-mixtures, and pesticide application before and after the pesticide attack was 46.6 and 53.4, and 75% of farmers do not separate tanks and nozzles are different pesticide application. Only 0.33% of total detection were ranged between 10 ppb to 100 ppb, and highest contamination was reported as 180 ppb for imidacloprid dry samples available in Ampara district, and it's not exceeding the it's not exceeding the MUMRL of 1,500 ppb. None of the detection were reported for the samples available Polonnaru and Kurunegala district, and this detection percentage are very low. But carbofuran banned in 2005, yet to be residue found. Propionopause is not recommended for rice. So suggestion, we have to cover all the district as a surveillance assessment, and clearly indicates the necessity of a comprehensive monitoring program for safeguard the quality of the rice in Sri Lanka, need for extensive farmer awareness programs on safe margins. Here I acknowledge Dr. G.A.W. Vijayasekara and Dr. J. Sumit, the former Registrar of Pesticide. They are the people who established the residue lab, and also Ms. C. Magamage, she is the person who established the central lab, and I acknowledge them. So they are the reference, and then thank you.
Thank you very much, Ms. Lakshani. Now the paper is open for discussion.
Ms. Lakshani, thank you very much for your nice presentation. When you did the analysis, did you take grain samples or the after the removal of husk or the whole the paddy?
Yeah, these only meal samples.
Yeah, meal samples. That means
we
only
are not removing the husk. It's not polished sample. Only we remove the. We not polish the sample. Only milled.
Okay. Because, you know, very recently,
yeah.
I came to know that
yes.
With the removal of husk, with the removal of other parts,
yeah.
so even even the toxicity is there, it
yeah.
will be removed because it has been proved. I think you also know that.
Yes,
yes,
I know that, sir. Yeah. But in the study, it's revealed that not polishing, the residues are very low. Yeah. So if you polished it and then analyze, it will come to very low levels.
Oh, even then it is at maximum within MRL level. So yes. It is happy to know that.
Yes.
Yeah, thank you.
Any other question?
Ms. Lakshani, you have done a good study and your results are also good. But I think you have done the study in a wrong period. I'm not sure because during 2020 farmers have not used much pesticides, I suppose. So do you plan to do the same experiment or repeat the same experiment during when the farmers are using more pesticides, when the pesticides are in the market freely available in the future?
Yeah, but in that era the pesticides are not banned because in 2020 January from March, so also that pesticides available at market. So that time we can test the samples again, but the problem is that the era that we purchase LCMS MS equipment, so we have to validate the instrument and we have to apply for the accreditation because we have accreditation till 2024. So that's why then we analyze because we have to validate the matrix. So rice is one of the matrix. So we can do in another time analysis, but the problem is chemicals are very, very expensive.
Okay, thank you.
Do you plan, Ms. Lakshani?
Yes.
Do you plan to analyze it from fresh vegetables and fruits?
Yes, I have done so many experiment for residue analysis and nearly published 3 articles on vegetables and fruits and also some imported samples, imported fruit sample, orange sample, apple samples. But this matrix is new for us. So according to Sante guideline, we have to validate 11 matrix. So vegetable fruits, we can found some pesticide, but when we compare it with rice, so detection level is very much low because from 57 pesticide, 62 samples, only 9 samples are contaminated and they are below to MRL levels. It means FMRL and Sri Lankan MRL, because we have some MRL levels we gazetted by Register of Pesticide Office. So according to that, levels are very low.
Okay. Thank you. Any other question? If not, we will thank Ms. Lakshani in the usual way. Thank you very much. This is the final presentation, so I will give a brief encounter on the papers presented in this session. First paper was on, actually it's an invited presentation by Dr. Wade Jenner, Deputy Program Executive, Plantwise Center, Center Director, Cabby, Switzerland. He has given a very good account on Plantwise program globally and especially in Sri Lanka, capacity building, training, development and distribution of reference materials, impact of plant clinic, advice adoption by the farmers in Sri Lanka and in other countries. So we will highly appreciate his work and the presentation. The next presentation is was by Dr. Fernando, Dr. H.R.P. Fernando. That is optimization of antifungal activity and determination of antifungal mechanism in lacto-fermented palm kernel cake against Coledotricum gliosporiates. Now, actually, he has done validated model, was tested and found very good results on antifungal activity on Coledotricum on mango fruits. And the timing was very good, and I congratulate him for his presentation. Next one is by Ms. Mara Singha, bioefficacy of selected novel insecticides for management of whiteflies and aphids in bitter gourd and brinjal in Sri Lanka. She has tested novel insecticides against whiteflies and aphids in brinjal and bitter gourd and found some effective insecticides, and some were recommended and the others are on pipeline. So I also congratulate her for her excellent presentation, and the timing is also good. Next presentation is on effectiveness of selected post-harvest treatments for population suppression of grain moth and rice weevil on stored paddy by Mr. Sarachandra. He has tested post-harvest treatments to manage grain moth and rice weevil in stored grains of different varieties, and found some treatments were quite good and the other treatments were not good. So his presentation is also very good and gives some treatments can be recommended. And next presentation was on pesticide use by rice farmers and the presence of residues in rice grain, an evidence-based study in selected rice growing districts in Sri Lanka. So she has done a survey on the use of pesticide and residue analysis in rice grain, and this is a very good thing for this kind of scenario in Sri Lanka, because everybody is concerned about the pesticide residues and health issues. So this is also a very good presentation, and I congratulate all these 4 scientists for the very good presentations and very good scientific evidences giving us, and as well as the general public of this country. And I congratulate all those officers and we will appreciate their presentations in the formal way. Ms. Rukmali will give you some kind of announcement.
Thank you, Madam. And now it's time to hand over the certificate for Best Presenter in the session. So I cordially invite our chairperson, Madam, to hand over the certificate. And the Best Presenter of the session goes to Dr. H.R.P. Fernando. So I cordially invite sir to come forward and receive your certificate. And then I would like to hand over token of appreciation to our panel of judges. Again, Madam, I cordially invite you to the hand over the token of appreciation. And Dr. Rajapaksha, Ms. Damayanti Galania, and Mr. Ekanayaka, please come forward and take your token of appreciations. And thank you, Madam. Then I would like to give token of appreciation to our chairperson. So I cordially invite Dr. Shiromani Rajapaksha, Director General, Ministry of Plantation Industries, to hand over the token of appreciation. So, Madam, please. Thank you, Madam. Finally, I would like to take this opportunity to express my gratitude on behalf of Asda Committee. Asda Committee, on behalf of Asda Committee, Chairperson Dr. Mrs. Jinadiri De Soisa, and invited speaker Dr. Wade Jenner, and the panel of judges to share their valuable time with us, with their busy schedules, and express my gratitude to you, audience, being with us with this event successfully and the active participation. Thank you very much. And there is another announcement. There is another session at NAICC at 1 p.m. and the lunch will be at ISTI. Thank you very much.
ඒගොල්ලෝ අරගයිනේ කාලෑවිල්ල ඒගොල්ලෝ එතනින් අස් කරගන්න.
Check one.
Test one, check. Test one. ආය අනිත් එක යකෝ. අනිත් එක. එකයි on කරේ. Test.
see all of you smiling, probably good news that you can have your tea and get back. Thank you. Ladies and gentlemen, can we all please rise to receive our chief guest and the other dignitaries? A very good afternoon, distinguished ladies and gentlemen. Warm welcome to the ceremonial closing session of the annual Symposium of the Department of Agriculture. For me, it's an honor, privilege, and a pleasure to be associated with this prestigious event from 2013 continuously. As you all are aware, the annual Symposium of the Department of Agriculture, the Asda, is the event eagerly looked forward each year by all of us in the Department of Agriculture, Ministry of Agriculture, university, academia, other scientists, and the private sector being engaged in agriculture. It creates an ideal scientific platform for scientists and extensionists to gather, discuss, debate, and exchange views on most important and timely issues towards the national agriculture development. Asda is a showcase of technologies generated by the scientists and extensionists of the Department of Agriculture, and it is a forum to disseminate those agriculture technologies among agriculture community. Ladies and gentlemen, Seed the Land, Feed the Nation is the theme of Asda 2022 in accordance with Sri Lanka's recent endeavor to revitalize the entire nation with regard to the food security after long-lasting socio-economic issues in the recent past. Having gone through the fruitful deliberations of Asda 2022, we have approached to the most exciting moment of the day, and that, of course, is the ceremonial closing sessions of the annual Symposium of the Department of Agriculture. Well, I'll be failing in my duties if I do not extend a warm welcome to our chief guest, Honorable Mahinda Amaravira, Minister of Agriculture. Warm welcome to you, Honorable Sir, and Honorable Mohan De Silva, State Minister of Agriculture, and our other dignitaries. In keeping with tradition, we now move on to the lighting of the oil lamp. Can I please invite Honorable Mahinda Amaravira, Minister of Agriculture, Honorable Mohan De Silva, State Minister of Agriculture, Mr. Rohana Pushpakumara, Secretary to the Minister of Agriculture, Professor Ogata Kazuo, Emeritus Professor, Kyushu University, Japan, Dr. Gamini Samarasinghe, Additional Secretary, Agriculture Technology, Ministry of Agriculture, representing Additional Secretaries of the Ministry of Agriculture, Dr. Shalomi Krishna Raja, Director General, National Botanic Gardens, representing the other government institutions, Dr. Ajanta De Silva, Director General of Agriculture, Senior Professor Udit K. Jayasinghe, Vice Chancellor, Wayamba University of Sri Lanka, Mrs. H.M.J. Ilankun Manike, Additional Director General of Agriculture, Development, representing the Additional Director Generals of the Department of Agriculture, Mr. K.D. Pushpa Ananda, Director, Seed and Planting Material, Development, representing the Directorate of Department of Agriculture, Professor Sarat Kodituaku, Dean, Faculty of Agriculture, University of Peradeniya, representing the academia, Professor Chalinda Benaragama, Editor-in-Chief, Annals of Asda 2022, Mrs. Priyanjali Madana, Provincial Director, North Central Province, representing the Provincial Directors, Mr. S.M. Nagawata, District Director, Colombo, representing the District Directors, and Mr. Janaka Gunasekara, Director Agriculture, A-Bar and Company Limited, representing the private sector. a representative from the paper presenters of Asda 22, and also we invite Mrs. Disna Ratna Singha, chairperson of Asda 22, to light the oil lamp in keeping with tradition. Ladies and gentlemen, please be standing for the national anthem. Also remain standing to observe a minute's silence in memory of those who've rendered yeoman service to uplift the agricultural sector who are no more with us. Thank you very much. You may be seated. Ladies and gentlemen, on this momentous occasion, now we have the pleasure of inviting the person who is leading the Department of Agriculture and determined to make changes by strengthening the networking among all relevant stakeholders in Sri Lankan agriculture to meet the growing demand from the agriculture sector, even under utmost difficult he's in the recent past. Ladies and gentlemen, he's none other than Dr. Ajanta De Silva, Director General of Agriculture, and I cordially invite him to deliver the welcome address. I've had the wonderful opportunity of working with him from 2013. Everybody, put your hands together and give him a round of applause.
Very good afternoon to you all. Honorable Mahinda Amaravira, Minister of Agriculture, Wildlife and Forest Resource Conservation. Honorable Mohan De Silva, State Minister of Agriculture. Mr. Rohana Pushpakumara, Secretary to the Minister of Agriculture. Professor Ogata Kasau, Chief Advisor, JICA and the Emeritus Professor, Keichu University, Japan. Professor Chalinda Benaragama, Chief Editor of the Asda 2022. Mr. Tasuya Yamadu, Chief Representative, JICA, Sri Lanka. Mrs. Disna Ranasinghe, Chairperson Asda 2022. Director Generals of other departments, former secretaries, additional secretaries, vice chancellors, Director General of Agriculture, former Director General of Agriculture in the Department of Agriculture, additional Director Generals of the Department of Agriculture, principal scientists, principal agriculturists, university academia, private sector, other distinguished guests, ladies and gentlemen. Once again, good afternoon to you all and distinguished guests, ladies and gentlemen. It's a great honor and privilege to welcome you all to the annual symposium of the Department of Agriculture, Asda 2022, as we celebrate 24th year from the inception of Asda, which was in 1999. First and foremost, I respectfully welcome our chief guest, Honorable Mahinda Amaravira, Minister of Agriculture, Wildlife and Forest Resources Conservation, who is here today despite your busy schedule and has found time to grace this occasion. Thank you very much, sir. I would also like to welcome our guest of honor, Honorable Mohan De Silva, the Minister of Agriculture, the State Minister of Agriculture, who is with us to grace this occasion. Thank you very much, sir. I take this opportunity to welcome the Secretary Minister of Agriculture, Mr. Rohana Pushpakumara, who is always behind with us from the right beginning to the last moment in supporting all initiatives of the Department of Agriculture. Thank you very much, sir. I am pleased for the presence of keynote speaker, Professor Ogata Kasuo, the Chief Advisor, JICA, and the Emeritus Professor from Keichu University, Japan, and also supporting the JICA initiatives in Sri Lanka. I am sure his keynote address will give some insights for the reworking Sri Lanka agriculture based on Japanese experience. I have the pleasure of welcome all the Director Generals, additional Director Generals of other departments, former secretaries, Vice Chancellor, YM University, additional secretaries, Director General of Agriculture, from other institutes, and additional Directors Generals, directors from, and the Dean's Faculty of Agriculture, Director PGIA, University of Peradeniya, and all the university academia, private sector officials, other distinguished guest members of the Directorate and officers from the DOA, Department of Agriculture. Ladies and gentlemen, agriculture is once again making headlines due to the price for food pricing and also for the food insecurity predicted by various parties. This makes a challenge for the Department of Agriculture to increase food production and also the productivity in the country. Yet, I feel Department of Agriculture is capable in all aspects to meet this challenge. To bring this revolution, country requires finding viable solution to a number of complex technical, institutional and policy issues, including market, connecting to markets, rural non-farm employment, trade policy, and food price stabilization. The Department of Agriculture's directions for the past several decades have brought this country to a hunger-free nation by introducing high-yielding varieties with associated technologies for all agroecological regions in Sri Lanka. A multidisciplinary approach to varietal improvement and crop production related problems, collectively planning and testing, including unique model of multidisciplinary approach, facilitate the rapid generation and identification of appropriate high-yielding varieties, and the development of improved package of practices for different agroecological settings. Department of Agriculture was able to develop high-yielding varieties, hybrids, combining desired level of resistance to biotic and abiotic stressors, adaptation to diverse agronomic variables and cropping systems, and meeting prescribed quality standards. This led to achieve food security even during hard period owing to natural and man-made disasters. The Department of Agriculture has experience in overcoming agriculture-related problems even during extreme situations and has been able to manage and guide the country to produce enough food to feed the nation. Our technology generation and dissemination targets are ambitious, but achievable. This has been shown in recent recommendations. The studies have also identified some of the challenges that we will face right now. The challenges underline the importance of planning our strategies carefully and executing it strongly. Honorable Minister, ladies and gentlemen, the aim of the Asda is to bring together scientists who will present and discuss their research results and latest advances in the field of their respective disciplines. Scientists of the Department of Agriculture presented new scientific innovations in the field of agriculture, which will be in great value and reshaping the agriculture production system in Sri Lanka. Agriculture begins with seed and continues to feed people. The Department of Agriculture generates technology for each of these scenarios, and the theme of the Asda 2022 this year, the seed, the land, feed the nation, matches very well with the Department of Agriculture mandate and to overcome present challenges of food insecurity. The Asda 2022 had technical sessions and we successfully completed today, had 20 scientific presentations, 15 short communications, 5 posters, 2 research notes, and 2 review papers. Out of these, 12 peer-reviewed papers are published in the Journal of Tropical Agriculturist, which is one of the oldest journals in the tropics. Selected papers are also being available in Govikam Sangarava in both Sinhala and Tamil language. I expect all these new innovations to be delivered to the end users who are the farmers through our extension arm of the Department of Agriculture. I take this opportunity to thank Asda 2022 Chairperson, Mrs. Dishna Ranasinghe, also with along with the executive committee, who were engaged during the whole year to bring this Asda 2020 to a great reality. I gratefully acknowledge the role played by Professor Chalinda Bendragama, Head, Department of Crop Science, Faculty of Agriculture, University of Peradine, as a Chief Editor of Asda 2022. Thank you, Professor. I will not forget the generous support from all private sector agencies, Food and Agriculture Organization, JICA, who always with the Department of Agriculture endeavors. Thank you for everyone. Ladies and gentlemen, let's all get together and produce more food while conserving the natural resources in this country to feed our nation. Once again, welcome you all. Thank you very much.
Thank you very much, Dr. Ajanta. Ladies and gentlemen, we now move on to another very special moment. This is where I would like to invite a very special personality to deliver the keynote address. Ladies and gentlemen, he's a personality who is the Chief Advisor of JICA project on research and education capacity improvement in dry zone agriculture at the University of Jaffna in the northern province of Sri Lanka. He's also an emeritus professor of the Kyushu University, Japan. He retired from Kyushu University in March 2021, and during his tenure, he was the senior vice president and director of the Institute of Tropical Agriculture and director of the University Museum. He has been in Sri Lanka since July 2022, and he will be here until 2024 as a JICA long-term expert. He has a number of intellectual publications and international publications to his credit on insect taxonomy and physiology, his specialty in entomology in particular, taxonomy and ecology of ants, and however, his interest is not just in the field of biology because he has experience in various international development projects in the field of agriculture in countries such as Bangladesh, Malaysia, Thailand, Vietnam, Myanmar, and other Asian countries. His career has expanded from natural history to international development of agriculture and higher education as well. So without further ado, it's my pleasure in inviting our keynote speaker, Professor Ogata Kazuo. Everybody, a round of applause.
Thank you for the introduction. Honorable Mahinda Amarawira, Ministry of Agriculture, Honorable Mahan Priyada Sharna De Silva, State Minister of Agriculture, Dr. Ajanta De Silva, the Director of the Agriculture, Mr. M.B. Al Kushimakumala, Secretary Ministry of Agriculture, Ms. Dishina S. Ratrasinghe, Chairperson of Asda, distinguished guests, ladies and gentlemen, good afternoon. It is my pleasure to have an opportunity to talk in the 24th Asda. I'm Kazuo Ogata, Chief Advisor of JICA project in University of Jaffna, and Professor Emeritus of Kyushu University, Japan. This year marks the 70th anniversary of the establishment of diplomatic relation between Sri Lanka and Japan. I'm honored to be able to speak on agriculture during the important year. My talk today includes the experience of Japan, which is not just a description of history, but intended to show lessons learned. Now, what do you know about Japanese agriculture? This is a description that are in the textbook of geography of Japan in junior high school. The textbook says we have some characters such as small scale with an average land of 1.8 hectare per household. And mostly farmers are part-time and intensive agriculture with high input. And the Japanese agriculture intending to conversion from rice to diversified crops and organic farming. And we have difficulties facing major 3 points. The first is low self-sufficiency, where about 60% or more agricultural products are imported. The competition with international products, where the price of Japanese agricultural product is high, and aging farmers, where nearly 70% of the farmers are aged of 65 or more. So I think some of the points are shared. Now, my talk today is divided into 4 parts. The first is modern history of agriculture in Japan. The second is Sri Lanka-Japan Agricultural Cooperation. And third is the smart agriculture by Ministry of Agriculture and Forestry Fishery, and the trend in the world. So the modern history of agriculture in Japan. Modern means, in our case, began with the Meiji Restoration in 1868, about 150 years ago. The country of samurai, until then, became a constitutional monarchy, introducing Western institutions, systems, technologies, and promoting rapid modernizations. Now, this is one of the examples of local agricultural production in Japan. The paddy rice yield in Yamagata Prefecture, Yamagata located in northern part of Japan. And the data is from 1885 to 2001. Oh, it's a very long range data. So this graph shows the how the rice yield has been changed. And the tendency shows rising to right, but you may notice some fluctuations in the ancient time, you know. So because of the location, the area tends to have affected by cool temperature. The farmers have had tackling this natural risk historically. The occasional drop, like here, 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 and here, showing the serious damage caused by the cold temperature or the disease. In 1890, the 2 new technologies are coming from southern part, Kyushu. One is hollow dry field with hose, and second is seed selection with salt solutions. It's very simple technology, but these new technology contribute to some of early development this time. The blue letters here show the variety name, rice variety name. The first popular variety was Kameno, which has potential high yield, but unstable. Until 1950, the yield shows high fluctuation. And after, the farmers attribute stable contribution to the shifting new varieties, like those varieties. In 1950s, the special cultivation method called protected semi-irrigated rice nursery was introduced and spread. So this was effective for preventing the cold temperature damages. And the end of 1916 to 1970s, because of overproduction of rice, so the government changed the policy to suppress the production of rice. This is so-called production adjustment policy, or say conversion from quantity to quality. As a result, in 1980s, you can see that the yield of the rice is rather flat. So this is an example that the improvement of farming, development of new variety, and the policies affect the change of rice production. Presently, the Yamagata prefecture has the position of top 4 in the rice production of Japan, and is famous for the good taste rice. Now, this was a case of Yamagata, but look at Japan as a whole. Here is the history of the yield of the rice in 1880 to 2000. And the line shows production, input, and productivity. So you can see the modern history of Japanese agriculture can broadly divided into 4 phases. The phase 1, it was initial growth. It's considered to the early growth period when the farmers developed superior variety of rice and introduced organic fertilizer, resulting in a significant increase in the production. And this phase, phase 2, is the time of until 1945, showing growth, then slowed. This is due to the depletion of farmer-based technical resources and labor shortage caused by 2 world wars. Now, phase 3, this period, the period of rapid growth after the World War II, the agricultural output increased due to the development of input and mechanization. And from the 1960s onward is the phase 4. This is a period of rice overproduction and trade liberalizations. The country once again entered a period of stagnations. However, you can see this line, that even during this period, productivity is still high, surpassing that of the phase 1. So this is believed to be due to the fact that despite the decrease in labor force, the private sector concerning agriculture was able to maintain the production by developing high technological capabilities to meet the needs of the market. Well, and this figure shows the change of the policy concerning about the distribution of rice. So the early stage, 1942 to 1995, the rice distribution was strongly controlled by the government. Designated collectors, designated corporations, licensed wholesalers, licensed retailers. So we have a security at that time, but very strongly controlled. And nowadays, you see that the marketing is very free. So there's no such government control, but they still have stockpile because this is very important. Now, the consumer can buy the farmers directly or a very simple way. As a result of this policy change, now we have so many rice varieties, hundreds of rice varieties. And these are quite different. You see, say, for example, this is the sweet to simple lines of taste and texture, rose to choy lines, horizontal. So each variety has different taste. And because the producer wants to make a new variety of profitability. So that's the present situation of Japan. Now finally, I just summarize the agricultural policy trends for these 70 years. Prior to 1960s, eradication of rural poverty and securing for goods supply is the major agenda. But in 1960 to 80, reduction of income disparity between agriculture and industries. So policy change like that. And then 1980 to 2010, this is the age of internationalization, adjusting towards internationalizations, bringing market principle to the agricultural sectors and integrating the concept of rural area food into agricultural policies. And now, 2010 to present, new agricultural reform to enhance competitiveness. So that's the very short flow of Japanese agriculture. Now, I just want to say about Sri Lanka-Japan agricultural cooperations. So we have modernization 150 years, but this has 70 years. So this might be something. This person was my boss. He is a professor of Institute of Tropical Agriculture. He always said to me, "Oh, Mr. Ogata, the curry is, best curry is Sri Lanka." He always told me. This was because he was in Sri Lanka. Well, in his history, he was here, Central Agricultural Research Station, Peradeniya, Ceylon at that time. And that time, the topic was study on diagnosis, identification, and control of chili virus disease in Ceylon. Wow, it was 1969 to 1971, and he published his result in 1975 with the co-authors of Bandaranaike and Hemachandra. But at that time, this publication is Sri Lanka. So he's something, you know. The time was changed during that short time. Well, that's just a story, but the whole flow of international development is also changing under the paradigms of economics and keyword. Agenda is also changed now, that age of SDGs, and Japan's style in agricultural cooperation also changed. So 1960s, the age of technology and skill transfer. That's also in the same as in Sri Lanka. But this story will be given tomorrow. Sorry, I have just touched this part. But we started the international cooperation in 1954. Japan joined the Colombo Plan, and at the same year, we had a participation from the technical training program in Japan from Sri Lanka. So here's a number of agricultural documents preserved in JICA library, the JICA reports. And this is something like, you know, the 1918 to 1919, it shows very high reports. You know, this building also built in 1980, 87, 86, something like that. And at that time, very big project was here by JICA, and also quarantine project in 1990s. So that was the age. And now, although there's no large-scale assistance by JICA, but fine-tuned cooperation that support the development of Sri Lankan agriculture continue even today. So anyway, the detail will be given tomorrow. Please expect that. Well, the next topic is the smart agriculture in Japan. This is the present agricultural trends. Probably this is the same of your countries. And in our case, smart agriculture means agricultural technology cross advanced technology. That means existing agricultural technology means expert skills, delicious breed and brands, safe and secure agricultural products. Multiply advanced technology such as robot tractor, assistant suits, or drones. Here's some example of advanced technology. All these something robot, like cabbage harvesting robots, tomato harvesting robots, strawberry harvesting robots, and autopilot rice transplanter, and of course drones, and we also are developing the so-called power assist suits that can be helpful for all the farmers. Now, but that sort of smart farmer is really useful. Now, the Ministry of Agriculture started so-called project for on-farm demonstration trials of smart agriculture. Well, this project is demonstrate the smart agriculture utilizing advanced technology and to accelerate the social implementation of smart agriculture. The objective of this project is to actually introduce the smart agriculture technology to production sites, conduct technological demonstrations, and clarify the effect of the introduction of technology on business management. So you see that 220 sites and various type of farming such as paddy, upland, open field, greenhouse, blah, blah, blah. Now, here's one of the results. The case of paddy. You see the working hours in paddy field farming. The light green is conventional area, and dark green introduced area. And yes, in some extent, the working hours are reduced, but just not so much, just 3%. I don't know the evaluation, but you can see the hilly and the mountain areas. Certainly, this paddy working hours are drastically reduced using by smart agriculture. This may be something. So, but still, there's some warning that because smart agriculture must not be over conflict. And the smart agriculture does not guarantee economic variability. This was the issue, still issue, and has issue to be improved both in farmer side and government side. So those are the smart agriculture in Japan. Now, and in smart agriculture, university also play a very important role. I would like to introduce one of my colleagues, a professor of the Kyushu University, Takashi Okayasu. He is a professor of agricultural machinery, and he has a very nice greenhouse. Look at that. His greenhouse is equipped motor unit, RGB camera, radar unit, sensors, so on. So he is observing the crops very carefully, closely. And this is one of the examples of his study on strawberry. Well, images. He took many photos, images, and classified the growth stage, flower, green, red, is okay. But learning. So growth feature extraction is learning by obtaining by deep convolutional neural network algorithm. So he used AI this point. And look at this. Individual recognition, location of estimation fruit, and also height and width will be measured. And AI learn, oh, this fruit is okay, this fruit is not still ripe, so on and so on. This is one. And other aspect of his study is visualizations. So since there are so many images and counting the fruits and measuring the green areas, so the data can show flower, unripened food, and ripened fruit, and leaf area ratio also measured from the data. And third point is that based on those data, so the machine can make a decision making. When should we harvest it like that? So these 3 points are the major role of his study. And what we say this type of study is called phenotyping, not genotyping, phenotyping. This is one of the examples, trend in plant phenotyping publications. Look at this. From 1997 to 2017, this author, Costa et al., counted the number of publications. They counted about 1,800, something like that. But look at this. Until 2007, the publication is not so high, occupied just 6 points or less. But after 2010, the publication of phenotyping increased drastically. Comparing with other genomics, precision agriculture, proteomics, say for example. Now, this type of study might be the mainstream of smart agriculture, might be. Well, that's the smart agriculture and the example of the study. Now, finally, I'd like to show the trend in world, not just Japan, but well, this part, again, I should say, will be described tomorrow. The smart food chain was a keyword in JICA. It's just food chain, but comparing with the smart agriculture, the digital ICT technology, AI technology, smartphones, and other devices are used to smart production, processing, and transactions in each process across the process. Say, for an example, in the breeding, genome editing, real-time measuring of biological information, and say for the production sections, crop protection technology, autopilot robot tractor, that sort of components are, say, smart food chain, SFC. The JICA will introduce those components in developing world. But you see, the needs and introducing technology mapping is a little bit difficult. Say, for example, in the vertical line shows the scale of farmers, large, small, and horizontal axis shows the profitability. And this area, that means small farmers countries, is more focusing on the higher needs of effective policy making. But in this area, that means high profitability crop and or large scale farmers, then the import support might be more useful. The area easy to introduce smart food chain related technology and packages. I think Sri Lanka is this sort of categories. And of course, many international organizations also use so-called digital transformations. World Bank, FAO, IFAD, WEB, CGIL, and your country also now are changing the e-agriculture, I know. So, but finally, I would like to introduce the case of Netherlands. Yeah, the Netherlands is the world's second largest exporter of agricultural products. 65% of Netherlands net export come from agriculture. And 10% of its workers are employed in agriculture. And the university, again, play a very important roles. In Wageningen University, one of the top universities in the field of agriculture, is the very nice facilities called Unifarm. The university and National Agricultural Research Institute have been merged into this one organization, completely merged. And in a large research facility called Uniform, the growth of plants in various environments is automatically measured and converted into the data in a huge greenhouse. And you see this wall, you see so many plates. The plates coming from the name of private company. That means this farm is not just controlled by the government or university, but so many monies are coming from the private company. So that is why they can push the actual agricultural research. This is one of the examples. Sorry, this is not Japan. But anyway, well, to summarize, Japan as a whole, we had so many difficulties and issues revealed by COVID-19 pandemic. So actually, we have slow digitization, and the economy is fragmentation of traditional connections, and societies are vulnerable to uncertainty, and the government is insufficient in administrative management, and infrastructure is securing social infrastructure in digital age. So the COVID-19 pandemic clarified this situation, but we must go. We must tackling using the digital technology as well. And lastly, I just say something about international development. Well, agriculture plays an essential role for the country's economy, but as country develops, the contribution to the growth become decline. Look at the above graph, which shows the share of labor and GDP in agriculture. Upper red line is share of labor in agriculture, and lower black line is the share of GDP from agriculture. And Sri Lanka is here. But you cannot expect that your country is going this way or this way. Look at this example. This is a case of China, Nigeria, and Brazil, just small, but each country has different history to the reduction of the agricultural growth, the contribution. But this is more common pattern here. Now, the World Bank reports show more generalization of development. The below graph shows the agricultural contribution to the country's growth. And this horizontal graph, line shows the poverty ratio of rural to total. The implication of this graph is that as countries develop, the contribution of agriculture is low, but the poverty moves from here to here. That means the poverty shifting to the urban area. Say, for example, in India, during the 10 years development, the dot coming from here to here, agriculture is less contributed. And this is a case of Indonesia. The plot is coming from here to here. That means shifting from urban, rural to urban. And this is the case of Brazil. The poverty is coming from rural to urban. And Sri Lanka is here. I don't know which shift, but probably the shifting to urban. The point is that desperations. So in this stage, we have to care about desperation between agriculture and other industry, rural area and urban areas. So that's something, example. That's all my story. Sorry, this is a case of Japan, but if you are interested, and you can learn something. Thank you very much.
Thank you very much, Professor, for your educative and excellent presentation, of course, amended with Japanese experiences. Well, in order to appreciate Professor Ogata's wonderful presentation, I cordially invite our chief guest, Honorable Mahinda Amaravira, Minister of Agriculture, to present a token of appreciation. I also would like to invite Dr. Ajanta De Silva, Director General of Agriculture, and Mrs. Disna Ratnasinghe, Chairperson Asda 22, to join in. Professor, we invite you to receive this token of appreciation. Everybody, put your hands together for a round of applause. Thank you very much. Moving on, in addition to the paper published in Asda Annals from Asda 2015, we have made a new initiative to publish selected articles submitted to the Asda in the Journal of Tropical Agriculturists, the oldest agriculture journal in the tropics. This time in Asda, there are 12 research papers which are published in issues of 170th volume of Tropical Agriculturalist. Research papers with practical importance on agriculture have been translated to both Sinhala and Tamil and now published in Govikam Sangarava and Kamat Tolil Vilakkam. Further, there are 8 research papers, 15 short communications, 6 research news, 2 review papers, and 7 posters have been published in Asda Proceedings 2022. Now let me cordially invite Mrs. Disna Ratnasinghe, Chairperson Asda 2022, to hand over the annals of the Asda 2022 Tropical Agriculturalists and other agricultural publications to the Director General of Agriculture, Dr. Ajanta De Silva, who in turn will present them to the distinguished guests. We now move on to the Asda highlights, a short video presentation on agriculture research and development, covering, of course, the different disciplines related to the present-day agriculture, along with how Sri Lankan agriculture is being transformed with innovative and sustainable technologies. Ladies and gentlemen, have a look at this video. This is time to view the highlights of Asda 2022.
Thanks to the Mother Nature's precious gifts of fertile lands, abundant supply of water, diverse and conducive climatic regimes across the island, coupled with the true commitment of our ancient kings for ensuring national food security and prosperity, made Sri Lanka was once known as the granary of the East, producing staple aplenty without harming the ambient environment. This well-known and documented agricultural prosperity of our mother nation was purely based on the traditional but scientifically proven technologies which were deployed in a manner of sustainable and eco-friendly context, with wisdom, knowledge and practices handed down from generation to generation. However, the absence of irrigated upland agriculture practices on a sustained basis in our ancient agricultural systems over this period is a noteworthy feature, to put it in records. After the collapse of hydraulic civilization-based agricultural prosperity of the country in the 12th century onward due to multifaceted reasons, until the independence from the colonial rule in mid-20th century, there was no remarkable or significant food crop-based agricultural development in the country, both in terms of technology and infrastructure. Instead, a very primitive subsistence-level farming practices were in place, which produced food commodities merely sufficient for household consumption, given that seasonal weather pattern remained similar in the general climatology of different geographies of the country, and otherwise communities to become highly vulnerable for food insecurity and starved or malnourished in the worst case scenarios. Nevertheless, successive governments of post-independence in Sri Lanka attempted to develop the domestic agricultural sector through the development of irrigation infrastructure by large-scale river-basin development programs, namely Galloya, Walava and Mahaweli. While provision of institutional support by timely delivery of credit, inputs and extension services where marketing was overlooked wittingly or unwittingly in the most instances. Meanwhile, the major focus of then agricultural policy of respective governments of the country was to achieve self-sufficiency in food commodities, mainly in rice, the staple, while additional investments were also made in the restoration and rehabilitation of existing irrigation schemes with the support from global development partners. These initiatives were of immense use in relieving the growing pressure of the population on the food and nutritional security of the country, while uplifting the means of livelihood in rural communities, which accounts over 80% of the total population of the country. The situation remains true even in the current national circumstances of Sri Lanka. During the last couple of decades, tremendous achievements have been made by the Department of Agriculture on productivity enhancement of food crops such as paddy, other field crops including condiments, fruits and vegetables in the country. For example, Sri Lanka achieved a near sufficiency in its staple rice in 2010 and the same level of production of maize in 2013. To achieve these milestones in line with the National Agricultural Development Agenda, a large number of high-yielding crop varieties coupled with their ability to sustain and produce significant yields under an array of biotic and abiotic stresses rising due to climate change have been developed and released to the farmers during recent times with required technological know-how to obtain the potential yield of each of those crop varieties. This entire exercise has been accomplished not as a sole duty of a single division of the department, but rather opted to adopt a holistic approach with an unmatchable team spirit among all divisions of the Department of Agriculture at the center, along with officials of the provincial departments of agriculture in the country. Meanwhile, we are compelled to accept the bitter truth and fact that our country came almost to a standstill position like other countries in the world with the sudden outbreak of COVID-19 pandemic in mid-2020, which was immediately followed by the current economic recession of the national economy and ongoing disturbed security situation in the Eastern Europe. Under these national and global circumstances, the whole agricultural sector of the country has faced an unprecedented challenge with the net food deficit, which is further exacerbated by the frequent and intense occurrence of extreme weather events under the changing and variable climate. Hence, it is beyond doubt that there should be a paradigm shift in National Agricultural Development Agenda to revive its gone glory to safeguard the national food and nutritional security at least by 2025. Such approaches will ensure that agriculture sector will not only be responsible for producing food for the nation, but also be an export-oriented and attractive employment-generating sector of the national economy to be in line with the current government's policy and global commitments such as reduction of greenhouse gas emissions from agricultural sector as per the nationally determined contributions NDCs to the UNFCCC. We in the Department of Agriculture firmly believe that it is not an unrealistic and unachievable task for us, although no officer in the department and its provincial arms can go back and make a fresh start to meet these new challenges ahead. We are committed to start from now and make a brand new ending. That is the broad meaning of the Asda 2022 theme. Seed the land, feed the nation. 4th Annual Symposium of the Department of Agriculture, Asda 2022. 6 technical sessions of the Asda 2022 were successfully conducted a while ago, where 19 research papers and 1 review paper were presented by the scientists and agriculturists of the department. The abstracts of the 12 peer-reviewed research papers full papers of the 8 peer-reviewed papers, 15 short communications, 5 posters, 2 research notes, and 2 review papers will be published in the annals of the Department of Agriculture. Those 12 peer-reviewed papers, which will be published as abstracts only in annals, will also be published as full papers in the forthcoming issue of Tropical Agriculturist, the oldest agriculture journal in the Tropics, volume 170, 2022. Technical sessions. Genes for food prosperity. DMJB Senanayaka. Differential reaction of Ratu-Heenity accessions available in Sri Lanka for brown planthopper resistance. The study reveals that the presence of a BPH10 gene alone or with BPH2, BPH3 and BPH13 genes play an important role in determining the resistance of Ratu-Heenity accessions to a mixed BPH population. D.S. Kekulandara. Morphological and molecular characterization of traditional rice cultivar Hondarawala. Analysis of morphological and molecular diversity of 21 Hondarawala. Accessions conserved in PGRC confirmed that there is considerable morphological and genetic diversity available among accessions of the same cultivar. W.A.R. Dhammika. Assessment of genetic purity of maize hybrids MI maize hybrid 03, MI maize hybrid 04 and MI maize hybrid 05 using SSR molecular markers. 3 SSR markers designated were found to be effective in determining the genetic purity of MI maize hybrid 03, MI maize hybrid 04 and MI maize hybrid 05. W.M. Ranjala Kumari. Genetic diversity of local and exotic finger millet Aleusine coracana germplasm in Sri Lanka. The molecular diversity analysis of 73 local and exotic finger millet germplasms showed the high genetic diversity with average of 3.6 alleles per locus and grouped the germplasm into 3 main groups. This information is vital to systematic conservation and utilization of trait-specific accessions in the breeding program of Sri Lanka. Soil, a challenge and an opportunity for sustainable development. W.M.U.K. Ratnayak. Strategies recommended to minimize fertilizer usage in rice cultivation of Sri Lanka. This paper reviews what the scientific literature has to say on fertilizer recommendation for paddy in Sri Lanka, highlighting the importance of soil test-based fertilizer application. KMS Kodikara. Effect of different levels of phosphorus on the availability and uptake of micronutrients in submerged and aerobic rice. It was found that growing rice under submerged conditions with the application of 40 kg P2O5 per hectare can be recommended to obtain sufficient micronutrient and crop yield while optimizing micronutrient uptake. WRKDWKV Vikramasinghe. Plant growth promoting Rhizobacteria consortia for growth and yield improvement in rice. Based on the high growth yield of the rice plant, the nutrient content of plant tissues and soil nutrient content at harvest confirmed that the application of Consortium 2, which consisted of 5 native PGPRs with a 75% recommended dosage of fertilizer, performed best in field conditions. PD Abhayatilakaratne. Effects of a genotype growing medium and a pot size on potato mini-tuber production using vermicompost leachate in soilless culture. This study confirmed that larger sized pots are suitable for the production of a higher number of mini-tubers with a higher yield, and the maximum tuber yield could be obtained by harvesting potatoes 80 days after planting in a soilless culture system treated with vermicompost leachate. Ancestors to nourish the nation. HMSN Herat, transferring of genetic male sterility trait into the female parent of local chili, Capsicum annum. The male sterile line of seed parent and maintainer line of the female parent, the Galkiriagama inbred line, can be successfully developed for commercial production of hybrid seeds to reduce the cost of production. EKEC Nayana, new cluster onion variety propagated through bulblets. A new high-yielding variety with large-sized bulbs and high punchant was developed using conventional plant breeding techniques, followed by selection of the best performers and release for general cultivation in Sri Lanka. HMPS Kumari, evaluation of selected local and exotic common bean germplasm for genetic resistance to rust pathogen Euromyces appendiculatus. Bean germplasm with UR3, UR5 and UR11 rust resistance genes were identified as the much needed resistant genes for bean rust disease, and pyramiding of those genes is important in bean crop improvement in Sri Lanka. Smart plant protection reflects your heart. RP Fernando, optimization of antifungal activity and determination of antifungal mechanism in lacto-fermented palm kernel cake. The results of this study reveal that the box-Benken design is an appropriate model to optimize the fermentation conditions and to maximize the antifungal activity of PKCl1. JP Marasinghe, bioefficacy of 5 insecticides, the management of whiteflies and aphids in bitter gourd and brinjal in Sri Lanka. As all 5 insecticides have different modes of action, a well-planned resistance management program with a structured and a restricted mode of operation of these chemicals, in alternative terms, could be recommended. SR Sarachandra, effectiveness of selected post-harvest treatments for population suppression of grain moth and rice weevil on stored paddy. 2-week interval sun drying method can be suggested in small-scale paddy storages, while in large-scale facilities chemical treatments can be used to spray on bagging material to obtain proper protection from grain moth and rice weevil. PWY Lakshani, pesticide used by rice farmers and the presence of residues in the rice grain, an evidence-based study in selected rice growing districts in Sri Lanka. This study attempted to investigate of the use of pesticides by farmers and a random multi-pesticide residue analysis in rice grain samples. The results revealed that the presence of pesticide residues in rice was low. Agronomy makes miracles for seed and soil. KGDS Bandara, enhancement of quality seed production of sesame through uniform desiccation and defoliation using chemical agents. The application of glufosinate, ammonium and pelargonic acid can be used effectively for the purpose of desiccation and defoliation in sesame cultivations for seed production. RGS erosion, determination of cardinal temperatures for pollen germination and pollen tube length of selected Capsicum annum accessions. It was revealed that pollen germination percentage and pollen tube length were useful indicators for screening of high temperature tolerant Capsicum accession. D. Karunanga, evaluation of high yielding varieties and fertilizer mixtures for sweet potato pot culture. This study confirmed that the Gunnerer White is the most suitable variety for sweet potato pot culture with upright growth. Negotiated vision, a novel agricultural extension approach. KAS Tilakaratne. Adoption and uptake pathways of recommended practices among the mushroom farmers in Kegalle district, Sri Lanka. This study revealed that the extension services have helped bring out the innovators and early adopters among the farmers. However, the technology adoption was mostly through a peer learning system and promotion of peer learning systems for farmers and strengthening of farm organizations. B. N. Abhay Singh. Do farmers acquire agricultural knowledge better from superior farmers? A quasi-experimental study among paddy farmers in Anuradhapura district. The results reveal that contract farmers are superior to followers but not effectively different. Thank you, Stuti Nandri.
Ladies and gentlemen, you just watched the highlights of Asda 2022 on video. And now it's my pleasure in inviting a distinguished gentleman who is responsible for making the overall policy directions for the agriculture sector in Sri Lanka. He's, of course, none other than the Secretary to the Ministry of Agriculture. I kindly invite Mr. Rohana Pushpakumara, Secretary, Ministry of Agriculture, to address this August gathering. Everybody, let's have a round of applause.
Good evening, everybody. Indeed, it is a great pleasure for me to address the gathering of Asda 2022 in this evening. As the Secretary to the Ministry of Agriculture, I highly admire the presence of Honorable Minister of Agriculture, Honorable State Minister of Agriculture, Professor Ogata and other JICA representatives, university academia, and eminent scientists gracing this great occasion. I came to know that this glamorous function unveils the findings of researchers and extensionists of the Central and Provincial Department of Agriculture, Sri Lanka. I am sure that these findings and facts bring a big momentum to the agriculture development in the country. Everybody knows the hardships which we have been facing due to sudden outbreak of COVID throughout the world during last 2 to 3 years. We all witnessed the impact of the massive economic crisis occurred throughout the world. Sri Lanka had no way to escape either from COVID or the economic crisis. You all envisage the mixed and different reactions appeared across the country with the government's decision to turn the whole country organic. The Ministry of Agriculture had to monitor the situations closely with the stakeholders to maintain food crop production to feed the nation. I am extremely happy with the role played by you all in the Department of Agriculture to regain agriculture prosperity under these crisis situations. Honorable Minister's guidance was invaluable to take the agriculture sector of the country to the correct path once again. As in any system, we also experience shortcomings, backlogs, and delays in the process of achieving the targets in crop production and productivity in the country. Most of them were due to the matters beyond our control. Higher prices for fuel in the world market have been the main reasons of high prices of inputs and collapsing of value chains in the food crops of the country. In spite of the escalated prices of inputs and items in the agricultural sector, the country has not been experienced big shortage of food items. I also wish to emphasize the importance of carrying out the role of the Ministry of Agriculture on a firm policy, and we, the Ministry of Agriculture, works towards that. I kindly request the Department of Agriculture to intensify its research program on local hybrid development, which is indeed essential to reduce seed cost and the dependency of the country on imported seeds. At the same time, I do not forget to appreciate your successful stories on local hybrid developments such as chili, maize, and pepper. I know the hardships that you experience due to the unavailability of human resources, especially scientists and extensionists. The Ministry of Agriculture is paying its utmost attention to solve the recruitment problem of the agriculture service officers for the Department of Agriculture. I request you all, scientists, academia, let us work here together till we see the prosperous in our sector. Wish Asda 2022 and all of you success. Thank you very much.
Thank you very much, Mr. Pushpakumara, for your sincere wishes. And on this momentous occasion, ladies and gentlemen, I have the pleasure of inviting our next speaker, who is a lawyer by profession. He later became an active politician and appointed as State Minister of Agriculture and Cabinet Minister of Water Supplies. With the recent ministerial portfolio changes, he took the oath to drive the agriculture sector towards prosperity as the State Minister of Agriculture. Without further ado, I have the pleasure of inviting Honorable Mohan De Silva to address this gathering.
සුබ සන්ධ්‍යාවක් තමුන්නාසල හැම දිනාටම මම ඊටාත්ම ස්තුතිවන්ත වෙනවා අද මේ ඊටාම වැදගත් අවස්ථාවට මට ආරාධනා කිරීම සම්බන්ධයෙන් මේ අවස්ථාවට ප්‍රධාන ආරාධිතය හැටියට සම්බන්ධ වන අපේ රජයේ කෘෂිකාර්මික වනජීවි හා වන සම්පත්ගරු අමාත්‍ය මහින්ද අමරවීර මැතිතුමනි ඒ වගේම අපේ අමාත්‍යාංශයේ ගරු ලේකම්වරයා හැටියට කටයුතු කරන රෝහණ පුෂ්පකුමාර මැතිතුමන් වගේම අපේ කෘෂිකර්ම දෙපාර්තමේන්තුවේ වර්තමාන අධ්‍යක්ෂ ජනරාල්වරයා හැටියට කටයුතු කරන ආචාර්ය අජන්ත සිල්වා මැතිතුමන්, ඒ වගේම ජෛක ආයතනය නියෝජනය කරමින් මේ අවස්ථාවට සම්බන්ධ වන මහාචාර්ය ඔගාටා මැතිතුමන් ප්‍රමුඛ ජෛක ආයතනයේ සියලු දෙනාන්, ඒ වගේ මේ අවස්ථාවට සම්බන්ධ වන අපේ අමාත්‍යාංශ හිටපු ලේකම් මහාචාර්ය උදිත් ජයසිංහ මැතිතුමන් ප්‍රමුඛ සියලුම මේ විද්වත් සභාවේ ඉන්න තමුන්නාන්සේලා හැමදෙනාගෙනුත් අපේ හිටපු අධ්‍යක්ෂ ජනරාල්තුමන් මතක වීරකෝන් මැතිතුමත් මෙතන ඉන්නවා. එතුමන් ඇතුළු තමුන්නාන්සේලා හැමදාම ගෞරවයෙන් අවසර ඉල්ලනවා. ඉතින් මට මතක විදියට 2020 අවුරුද්දෙත් මේ වගේම අවස්ථාවකට සම්බන්ධ වුණා. ඇත්තටම මම ඊටාත්ම පුදුමයටපත් වුණා තමුන්නාන්සේලාගේ දක්ෂතාවයන් දැකලා. මොකද අපේ රටේ සාමාන්‍ය ජනතාව අතර තමුන්නාන්සේලා මේ කරන වැඩ කොටස පිළිබඳව තියෙන අල්ප දැනුවත්භාවය. දැන් මේ ඇත්තටම මම ඉල්ලීමක් කරනවා මේක මීට වැඩිය තමුන්නාන්සේලා කරන කාර්යභාරය අපි ජනතාව අතරට ගෙනියන්න ඕනේ. ඉතින් මම හිතන්නේ ඒක මම හිතන්නේ ජනතා නියෝජිත හැටියට මම හිතන්නේ ගරු එමත්තුන් මේ අවස්ථාවේ ඉන්න නිසා ඒක අපේ පැත්තෙනුත් මම හිතන්නේ කළ යුතු කාරණයක්. ඒ නිසා ඉතින් ඇත්තටම අද ඒ කාරණේ තමුන්නාන්සේලා අවධානයට කරන්න ඕන. ඒ වගේම අද දැන් මේ තමුන්නාන්සේලා ගිය අවුරුද්දේ එහෙම නැත්නම් ඉස්සෙල්ලා අවුරුද්දේ තමුන්නාන්සේලා කරපු පරේෂණවල ඒ ප්‍රතිඵල අපිට පෙන්වන අවස්ථාව. ඒ ඇත්තටම අපිට මට ජනතා නියුතයට තියෙන එක ප්‍රශ්නයක්. දැන් අපි නිදහස ලබන අවධියේ වෙනකොට මම හිතන්නේ කෘෂිකර්මාන්තයෙන් අපේ ජාතික ආර්ථිකයට මීට වැඩිය දායකත්වයක් ලබා දුන්නා. පරණ මහ බැංකු වාර්තාවලට ගිහිල්ලා බැලුවොත් මම හිතන්නේ ඒක අපිට බලන්න පුළුවන්. හැබැයි එන්න දැන් ඒ කාලේ මෙච්චර තාක්ෂණය දිනුනේ නැහැ. නමුත් මේ එන්න දැන් අද වෙනකොට අපි නවීන තාක්ෂණ යොදාගත්තත් මම හිතන්නේ අද එන්න අපි අපේ ජාතික ආර්ථිකයට ලබා දෙන පංගු අඩු වෙලා තියෙනවා. ඉතින් මම හිතන්නේ ඒක මම හිතන්නේ අපි සොයා බැලිය යුතු කාරණයක් කියලා අපි දකින්න. ඒ වගේම මම හිතන්නේ සමහරක් වෙලාවට අපි ජනතා නියෝජිත හැටියට ඊටාම සත්භාවයෙන් ගන්න තීන්දුව තියෙන සමහරක් වෙලාවට තමුන්නාන්සේලා විද්වතුන් හැටියට ඒ කියන දේවල් අපි සමහරක් වෙලාවට ප්‍රතික්ෂේප කළා ගන්න තීන්දුව తీරන වලින් රටට යම් ප්‍රශ්නත් වෙලා තියෙනවා කියන කාරණේ කියන්න ඕනේ. හැබැයි මම දකිනවා එතනදිත් සමහරක් වෙලාවට එකම ශ්‍රේෂ්ඨයේ ඉන්න විද්වතුන් විවිධ අදහස් පළ කරනවා. එතකොට මම හිතන්නේ ජනතා නියෝජිත හැට මොකද අපිට මේ විශේෂ දැනුමක් අපිට මේ සම්බන්ධයෙන් නැහැ. අපි ඇත්තටම අපේ ලේකම්තුමා එහෙම නැත්නම් තමුන්නාන්සේලා කියන දේවල් තමයි අපි ක්‍රියාත්මක කරන්නේ. එතකොට අපිට තියෙන ප්‍රශ්නේ තමයි ඇයි එකම ශ්‍රේෂ්ඨයේ ඉන්න විද්වතුන් එකම කාරණයක් උදෙසා විවිධ මත පළ කරන්න කියන කාරණේ. ඉතින් මම හිතන්නේ මේකට හොඳම උදාහරණය තමයි අපි පහුගිය ආණ්ඩුව කාලේ හැටියට කාබනික පොර අපි හඳුන්වලා දුන්නා. අතිගරු ජනාධිපතිතුමායි වගේ හිටු ගෝඨාභය රාජපක්ෂ මහත්මයා බොහොම සත්භාවයෙන් ගත තීන්දුවක්. ඉතින් එතකොට එතෙන්දී එකම ශ්‍රේෂ්ඨයේ ඉන්න විද්වතුන් විවිධ මත පළ කරා. ඒ නිසා මම හිතන්නේ මම මෙන්න මේ ප්‍රශ්නේ තමුන්නාන්සේලාගේ අවධානයට යොමු කරා. ඇයි මෙහෙම වෙන්න කියන කාරණේ මම හිතන්නේ තමුන්නාන්සේලා අවධානයට යොමු කරන්න ඕනේ. ඉතින් කොහොම වුණත් මම ඊටාම ඉහළින් අගය කරන ඔබ තමුන්නාන්සේලා විද්වතුන් හැටියට මොකද අපි මම හිතන්නේ අපි මේ කෘෂිකර්ම ශේෂ්ත්‍රය තමයි අපේ රටේ ජීවනාලි. මම දැන් තමුන්නාන්සේලා ලබා දෙන ඊටාම ඉහළ දායකත්වය අපේ රට ඉස්සරහට ගෙනියන්න මම ජනතා නියෝජිත හැටියට ඊටාම ඉහළින් අගය කරනවා කියන කාරණේ කියන්නට. එතෙන්දී අපිට හොඳ වැඩ කරන්න පුළුවන්. කරුව අමාත්‍යවරයෙක් ඉන්නවා මහින්ද අමරවීර මැතිතුමන්. වගේ ලේකම්තුමා ඇතුළු සියලු දෙනා අපිට උදව් කරනවා. වර්තමාන ජනාධිපතිතුමා ඇතුළු රජයත් ලොකු උත්සාහයක් ගන්නවා කෘෂිකර්ම ශේෂ්ත්‍රය ඉදිරියට ගෙනියන්න. එතෙන්දී කෘෂිකර්ම රාජ්‍ය අමාත්‍යාංශය හැටියට අපිට ලබා දෙන්න පුළුවන් යම් සහයෝගයක් තියෙනවා නම් ඒ සහාය ලබා දෙනවා කියන කාරණේ ඊටාම ඉහළින් අගය කරන ගමන් ජපාන් රජය සහ ජයික ආයතනය අපේ රටේ කෘෂිකර්මාන්තය නගා සිටවන්න ලබා දෙන ඉහළ දායකත්වය පිළිබඳව අපේ රජය වෙනුවෙනුත් මගේ ගෞරවනීය ස්තුතිය පුදන තමුන්නාන්සේලා හැම දෙනාටම සුභානාගතය ප්‍රාර්ථනා කරමින් මගේ අදහස් ප්‍රකාශ කිරීම අවසන් කරන බොහොම ස්තුතියි.
Stuti, thank you very much, Honorable Sir. Ladies and gentlemen, we're now moving on to the most exciting moment of Asda 2022. This, of course, is the presentation of awards I kindly invite Dr. Achanta De Silva, Director General of Agriculture, and Mrs. Disna Ratnasinghe, Chairperson of Asda 2022 on stage to receive the medals, awards, and plaques from the sponsors of Asda 2022. May I request Dr. W.M.W. Veerakon, President, Sri Lankora, to hand over the award and the plaque of the eminent agriculturalist to Dr. Achanta De Silva, Director General of Agriculture. I'm sure we can put our hands together for a round of applause. Thank you very much. May I now cordially invite Mr. Chamila Senanayaka, General Manager, CIC Agribusinesses, to hand over the gold medal of the best agriculturalist to Dr. Achanta De Silva, Director General of Agriculture. Please, a round of applause. We do appreciate your valuable contribution. Thank you. Let me now invite Mr. Atauda Javardhana, Chief Executive Officer, Director of Innovative Pesticides Marketing Private Limited, to hand over the gold medal for the best scientist to Dr. Achanta De Silva, Director General of Agriculture. Why not? A round of applause for him as well. And may I also cordially invite Mr. Upali Yapa, Senior Research Officer, COPIA, on behalf of Director COPIA Sri Lanka, to hand over the cash award for the best agriculturalist, best scientist, and best paper of Asda 2022 to Dr. Achanta De Silva, Director General of Agriculture. Thank you very much, Dr. Achanta De Silva and Mrs. Disna Ratnasinghe. Thank you. You may go back and sit down. Thank you. Ladies and gentlemen, Sri Lanka Korea Alumni Association, Sri Lankora, in collaboration with Asda, initiated an annual award scheme in 2015 to appreciate eminent agriculturalists who had done an exemplary contribution, who made an exemplary contribution to the Sri Lankan agriculture. Executive Committee of Sri Lankora has carefully evaluated the contribution of many scientists, academics, and policymakers in order to select an eminent agriculturalist for Sri Lankora Lifetime Award 2022. Well, let's find out who has been selected by looking at this video presentation.
Three, two, one.
On the 2nd of June 1953, a blessed of rural mother of a farming family in the remote village of Panamure in the Ratnapura district gave birth to a beautiful boy who later completed his primary education at Kahavatta Mahavidyalaya, then secondary education from Gankanda Central College, Pelmadulla. He got through his GCE advanced level examination in Vidyaratna College, Horana. In 1974, this young boy entered the Faculty of Agriculture in the University of Peradeniya and graduated in 1978 with an honours degree in agriculture. He worked a short period for Mahaveli Development Project and then joined to the Sri Lanka Agricultural Service in January 1980. His first placement was to Ratnapura District as Segment Agriculture Officer and then to Ampara District as Assistant Director of Agriculture. He was instrumental in introducing and managing training and visit system of agricultural extension approach. He dedicated his career for agricultural extension, especially with rice farmers in high potential areas of the island. In 1987, he received his master's degree in agriculture extension education from University Illinois in USA. In the early 1990s, After spending his young and energetic days in the career mixed with farmers of the dry zone with utmost scarification, he became rice extension agronomist at headquarters of the Department of Agriculture in Peradeniya, coordinating all rice productivity improvement activities in the island. He pioneered the rice production Yaya approach to improve the productivity of rice to become as self-sufficient nation in staple food at that time. He also worked as Deputy Director Extension and Communication at the Rice Research and Development Institute, Batalagoda, coordinating all rice crop development activities. Later, he moved to the Ministry of Agriculture as Additional Secretary to coordinate agriculture technology and agricultural development of the country. He was one of the pioneering members responsible for the establishment and managing of Sri Lankora since 2005. During the last 4 decades in the field of agriculture, served as an extensionist, agronomist and agricultural development administrator. Sri Lankora is honored to confer his prestige Lifetime Award in 2022 to Mr. Shanta Siri Emitia Goda.
So now, ladies and gentlemen, you know who wins the Lifetime Achievement Award. And to make this very important presentation, I cordially invite our chief guest, Honorable Mahinda Amaravira, Minister of Agriculture, to present the plaque, and Honorable Mohan De Silva, State Minister of Agriculture, to present the cash award, and Dr. Ajanta De Silva, Director General Agriculture, and Mrs. Desna Ratnasinghe, Chairperson Asda 2022, also on stage. Everybody put your hands together, give them a round of applause. Ladies and gentlemen, it's my extreme privilege to invite Mr. Shanta Emitia Goda, who has made immense contributions to the Sri Lankan agriculture sector, including agriculture extension, rice production, and agriculture policy matters in various capacities as Agricultural Officer, Assistant Director of Agriculture Extension, Deputy Director Extension, and Additional Secretary, Agriculture Department in the government sector. And moreover, he has extended his services beyond the government sector, and he has marked his footprint in the private sector in various associations and international agencies working towards the achievement of excellence in agriculture in Sri Lanka. Ladies and gentlemen, put your hands together to appreciate his valuable contribution to the agricultural sector. Yes, Mr. Shanta Emitia Goda. Yes, please stay on. We'd like you to please take your seats. In fact, I would have loved if you all gave him a standing ovation, everybody. It's not too late. Please stand up, because I was reading his entire profile and didn't realize he was already on stage. Please put your hands together and give Mr. Emitia Goda, well, you guys sit down, Mr. Emitia Goda, you may sit. Everybody is giving you a round of applause for your valuable contribution. Thank you very much. Thank you. That's what happens when a compere has to read a long profile. Can I also invite on stage Mr. Rohana Pushpakumara, Secretary, Ministry of Agriculture, Professor Chalinda Benuragama, Editor-in-Chief, Asda 2022. And now, ladies and gentlemen, it's the awarding of the best research paper of the Asda 2022. I would like to invite Professor Chalinda Benuragama, Editor-in-Chief, to express his views and announce the best paper of Asda 2022. Professor Chalinda Benuragama is a senior lecturer attached to the Department of Crop Science, Faculty of Agriculture, University of Peradeniya, and currently serves as the head of the department. He gave his immense contribution to complete the editorial work in time to publish the Annals of the Department of Agriculture as the Editor-in-Chief of Asda 2022.
distinguished members in the head seats, ladies and gentlemen. If someone questions what has research done for you in agriculture, the answer is very straightforward. Research has altered our life and the world in general massively. The standard and the quality of living we enjoy today is much higher than it was for previous generations. New discoveries will continue to be made, and as a result, it is expected that future generations will have even better living standards. In agriculture, scientists actively seek for discover methodologies that will increase crop yields, improve crop productivity, reduce loss due to diseases and insects, develop more efficient equipment, and increase overall food quality. In addition, researchers look for ways to increase farmers' profits while protecting the environment, where Sri Lanka is no exception. As the custodian of agriculture in Sri Lanka, the Department of Agriculture plays a pivotal and vital role in the generation of new knowledge and technology through research and development, targeting a positive impact on the overall development of the agriculture sector in Sri Lanka. So, ladies and gentlemen, this annual Symposium of the Department of Agriculture, Asda, creates a platform to stage the findings of untiring research and development efforts of the Department of Agriculture as an annual scientific forum for its multiple stakeholders. This Asda 22 unveils such commendable studies conducted during 2017 to 2022. I must really appreciate all the efforts made by the scientists in the Department of Agriculture, particularly during the COVID-19 pandemic. The whole country was at a lockdown. Even until recently, we were on rosters, but all the researchers continuously worked. So this is what we have witnessed today. So, ladies and gentlemen, I'm greatly honored and privileged to be here today on this podium as the Editor-in-Chief for the Annals of the Sri Lanka Department of Agriculture 2022. Most importantly, my presence in leading the editorial board symbolizes the eternal collaborative friendship that the Department of Agriculture has with the universities, particularly with the University of Peradeniya. As we already witnessed, we have seen, 63 research efforts were submitted this time, out of which only 19 full papers, full-length articles, empirical studies were staged today. The research studies included in the annals of the Asda 22 cover evidence-based studies on almost all scientific aspects of agriculture, including agronomy, plant breeding, agricultural economics, food science and technology, biotechnology, plant protection, post-harvest physiology and technology, agricultural extension, and many more related areas. These studies have been conducted mainly by the scientists and agriculturists of the Department of Agriculture, and sometimes in collaboration with some scientists of other related institutes as well, including foreign collaborators. After all, I had a Herculean task in front of me. That was the selection of the best research paper submitted to the Asda 2022. In this endeavor, I have taken the following 10 criteria into consideration to select the best. Clarity of focus. We wanted to see whether the focus was clear, just to see why they have done this kind of a study. And then the national, regional significance of the results, how important the findings are, and then the contribution to the development of science, because the science should progress. So we wanted to see how the contribution towards the science is. And not only that, we wanted to see the practical applicability of the findings, because it should go to the farmer one fine day. And if it is a research, if it is a good research, it should be repeatable. So we wanted to see the methodology in particular. If it is repeatable, then that research can be considered as a good one. And then we looked into the clarity of presentation of the results, because after all, that's what we see as the general public or the scientific community, what they have done. In terms of the tables, figures, the data, and how they have done the statistical analysis. So we wanted to see that. And then, after getting the data, how they have discussed their results, because we wanted to see how they placed their results, their outcomes, compared to the findings of some other places in other countries, elsewhere. And particularly, we wanted to see scientific writing skills as well here, because it's a different skill. It's not just writing, it's scientific writing. And we want to see how the work has been conducted, the collaborations, because research is a huge teamwork. It's a huge teamwork. No one on this earth cannot perform individually to make a good research publication. So we wanted to see that teamwork within that article. And finally, the most importantly, the attitude of the researcher, the research team. We wanted to see how far they have gone in complying to all the guidelines given by the Asda organizing committee, because otherwise the committee is in trouble always if the authors do not comply to the guidelines. So, all these 19 empirical studies submitted this time were evaluated based on the aforesaid criteria and ranked. Ladies and gentlemen, all the distinguished guests in the head seats, of all, the Best Paper Award at the Asda 2022 goes to the paper titled Optimization of Antifungal Activity and Determination of Antifungal Mechanism in Lacto-Fermented Palm Kernel Cake against Colatrichum gliosporidis, authored by Ranjit Piaseri Fernando. The co-authors are A.S.M. Hussain, B.J. Muhaldin, N.L. Yusuf, N.K. Mohammed, and S.F. Arifin. Particularly, we wanted to see how applicable these findings are. So this is, ladies and gentlemen, to me, one of the very, very most important findings which definitely take the fruit industry to a totally different height. So, my congratulations to Mr. Ranjit Pesar Fernando. Ladies and gentlemen, please round of applause. And to his team. Of course, my heartiest congratulations goes to all the other authors as well. I wish to extend our sincere appreciation to all the authors, all the other paper writers, the paper submitters, and independent reviewers, because that also was a Herculean task, for their excellent and magnificent helping hand, and that was extended during the whole process, and their prompt response as well. And finally, I take this opportunity to thank the Asda 2022 team, headed by Madam Disna Ratnasinghe, for the very friendly atmosphere created by all of you to me to work with very much ease. And to all the scientists, I humbly request, don't stop in searching for science. Sky is the limit. Because science never solves a problem without creating 10 more, as George Bernard Shaw says. Thank you very much.
And to present the award for the best research paper, can I please invite our chief guest, Honorable, or rather, one of our guests of honor, Honorable Mohan De Silva, State Minister of Agriculture, also Mr. Rohana Pushpakumara, Dr. Ajanta De Silva, and Mrs. Disna Ratnasinghe to present the award for the best research paper of Asda 2022. Please read the name of the recipient again. You can read the recipient? Yeah, please.
Once again, the best research paper of the Asda 2022 goes to Mr. Ranjit Piaseri Fernando.
sure we can put our hands together for a better round of applause. Winner of the best research paper of Asda 2022. Thank you very much, Professor. Thank you. I now have the pleasure of inviting Dr. Lakshmi Priyanta, an Executive Committee member of Asda 2022, to hand over the all-important list of award winners to Mrs. Disna Ratnasinghe, Chairperson, Asda 2022. Why the silence? Everybody, a round of applause. And I will receive the all-important awards. Right, here we go. This is the time to award the best agriculturalists and the best scientists of Asda 2022. The event has been practiced since the inception of Asda, and it is a rewarding mechanism for the agriculturalists of the Department of Agriculture who have excelled in their streams and disciplines. Their service to uplift agriculture in the country is so huge, and hence their appreciation should not be overlooked. This time also a best scientist and a best agriculturalist has been identified, and this award for the best scientist is presented to the best researcher for his or her uninterrupted scientific endeavor, contribution to agriculture while promoting his or her career. The best agriculturalist, on the other hand, takes technology to farmers and to other stakeholders and does an unmatched service to the nation. That is why both of them are presented with gold medals in accordance with the customs of the annual symposium of the Department of Agriculture, Asda 2022. And they've selected the best scientist and best agriculturist of the year 2022 by an eminent panel of judges. Let me tell you who the judges were. Dr. Lal Kurpuarachi, former Dean of Sanasa Campus, Professor D.K.N.G. Pushpakumara of the Faculty of Agriculture, University of Peradeniya, Mr. Pasan Ratnayaka, Secretary, Provincial Ministry of Agriculture, Central Province, Mr. Atauda Jayawardena, Chief Executive Officer, Director, Innovative Pesticides Marketing Private Limited, and Mr. Varuna Madhavana Arachi, Managing Director of CIC Agribusiness. Everybody put your hands together and give our judges a round of applause. Time now to present the award for the Best Agriculturalist of the Year 2022. Born in March 1967 in Moratuwa, who got her primary education from a government school, Paluvelgala, also Holy Family Convent, Maravilla, and completed her secondary education at St. Paul's Girls' School, Milagiriya, Bamlapetia. Subsequently graduated from the Faculty of Agriculture, University of Peradeniya in 1995, and she obtained her MSc degree in Food Science and Technology from the University of Peradeniya in 2010. Due to her interest in law, she entered Sri Lanka Law College and completed her preliminary exam first year of the Tennis at Law. Well, she joined the Sri Lanka Agricultural Service in 2000 and started her career as a lecturer at the Sri Lanka School of Agriculture, Variapala. And during the latter stage, she worked as the principal of the school. Then she got a transfer to Sri Lanka School of Agriculture, Kundasale, in 2012 and served as a lecturer and finally as a vice principal. In 2018, she was appointed as the District Director of Agriculture, Badulla. She contributed to agriculture production, development, and marketing of Badulla district in coordination with the Ministry of Agriculture, technical departments, farming community, and administrative bodies. She has also contributed to the agriculture development by planning and implementing a number of projects. In 2019, ladies and gentlemen, she reached another turning point in her career. She was appointed as the Provincial Director of Agriculture, Uva Province, giving her the entire responsibility of the agricultural development in the province. She coordinated several nationally and internationally funded projects in order to uplift the livelihood of the farming community in the Uva province. She pioneered to commence the NVQ Level 4 course to Field Assistance Agriculture in District Agriculture Training Center in Bibile. She has immensely contributed to the Sri Lanka Organization for Agriculture Professionals, Sri Lanka Institute of Agriculture, and the Sri Lanka Agricultural Extension Association as an office bearer of the executive committees. She is a professional fellow in agriculture extension of the Sri Lanka Institute of Agriculture since 2020. Well, I'm not going to read out her name, but before that, we'd like to invite our presenters, our chief guest, Honorable Mahinda Amaravira. Sir, can I have you to step forward? He will present the gold medal. Honorable Mohan De Silva will present the certificate. Mr. Rohana Pushpakumara will present the cash award. And of course, we'd like to invite Dr. Ajanta De Silva and Mrs. Disna Ratnasinghe to also join in. Yes, ladies and gentlemen, you hear the fanfare. The best agriculturalist of the year 2022 is Mrs. Aisha Manel Devi Gunawardana. Everybody put your hands together for a better round of applause. Congratulations. And while our presenters are there, we are moving on to the award for the Best Scientist of the Year 2022. The recipient started her primary education at Kumbukwaba Mahavidyalaya. She qualified for university entrance from Ibba Gamur Central College. She graduated from the University of Peradeniya with a BSc Agriculture in 1993 and obtained her master's degree in Natural Resources Conservation from the Asian Institute of Technology, Thailand in 1999. She obtained her PhD in geoinformatics from the University of Peradeniya in 2019. She joined the Department of Agriculture in 1996 and was stationed at the Field Crops Research and Development Institute, Mahailuppalama, as a research officer in the field of soil and water management. In 2006, she assumed duties at the Rice Research and Development Institute, Vatalagoda. She then was promoted as Principal Agricultural Scientist, Soil Science in 2017 and served as the Additional Director at Rice Research and Development Institute from 2018 to 2020. During the last 10 years, she was able to release 6 technologies related to the field of water management, soil fertility, and GIS to the farming communities in Sri Lanka. These include alternate wetting and drying technique in paddy cultivation, map-based fertilizer recommendation for rice, reclamation of paddy cultivating soils based on soil maps, phosphorus fertilizer application in alternative seasons in rice cultivation, parachute cultivation technology with phosphorus-treated seedlings in rice, soil test kit-based P and K fertilizer application, and correction of soil pH in rice cultivation. She, ladies and gentlemen, was the focal point for specialized expert system for agro-meteorological early warning system, and she worked as the convener of the FAO project on irrigation and water management in paddy fields in Sri Lanka. She served as the working group coordinator for soil and water management, natural resources, and plant nutrient management. She worked as the Assistant Secretary 2018 to 2019 and editor from 2019 to date of the International Network of Water Ecosystems of Paddy Fields. She has more than 40 research publications on water management, soil science, and geoinformatics related to rice cultivation. During the last 10 years, she has received NRC Merit Awards for scientific research in 2017. She was a member of the Most Outstanding Rice Project in FRC member countries in 2015, and she received the Best Presenter Award of the Asda in 2014 in the Soil and Water Management category. She received the President's Award for Scientific Publication, and moreover, her work in the field of agriculture was commended by the Director General of Agriculture in 2021. Ladies and gentlemen, the best scientist of the year 2022 goes to Dr. Miss Upul Kumari Ratnayaka. Honorable Mahinda Amarivela will present the gold medal, Honorable Mohan De Silva, the certificate, Mr. Rohana Pushpakumara, the cash award. Everybody put your hands together for the best scientists. Thank you very much, our presenters. Thank you. And congratulations to the winners. Please, you can take your seats. Thank you, sir. Well, we also had a poster presentation session, and the title was a study on grasshoppers and their possible damage to rice crops. We had selected a winner. The award has already been given. For your information, the winner was Mr. S.R. Sarat Chandra, Assistant Director of Agriculture from RIDI, Batalagoda. Everybody put your hands together and give him a round of applause. And now, ladies and gentlemen, we move on to yet another very special moment, and I have the utmost pleasure in introducing this personality. He is none other than our chief guest of Asda 2022. After his yeoman service in the Angunup Alassa Pradesha Sabha and Southern Provincial Council, he has been serving as a Member of Parliament since 1994 to date. And during this period, he held several ministerial portfolios such as health, fisheries, power and energy, and many more. He also held a number of positions in various parliamentary committees, public, private, and social institutions. Currently, as the Minister of Agriculture, he's working hard to ensure the national food security, facing many hardships due to the current socio-economic crisis in the country. He, ladies and gentlemen, is a great personality in the Sri Lankan political arena. Without further ado, I have the pleasure of inviting our chief guest, Honorable Mahinda Amaravira, Minister of Agriculture, Wildlife and Forest Resource Conservation, to address you all.
Good evening. Honorable Mohan Priyadarshana Silla, State Minister of Agriculture, Mr. Rohana Pushpakumara, Secretary to the Ministry of Agriculture, Dr. Ajantha Silla, Director General, Department of Agriculture, Mr. Yamada Tetsuya, Chief Representative, JICA, Professor Ogata Ketsui, Chief Advisor, JICA, Professor Charlinda Benragama, Chief Editor, Asda, Mrs. Disna Ratnasinghe, Chairperson, Asda 2022, representatives of JICA, distinguished invitees, ladies and gentlemen. I sincerely express my gratitude to the Director General Agriculture and the organizing committee of the 24th Asda. Your invitation for this event is a great honor to me. I can see many eminent scientists, Department of Agriculture officers, university academia, and many other distinguished guests are participating in this event. I reflect that all of us are working together for the development of the agriculture sector in the country. All of us are aware that COVID outbreak and subsequent economic crisis created a very bad economic situation throughout the world. As a country, we also have experienced a very difficult period in all the aspects. It has become a difficult task for the government to provide essential inputs to the farmer community of the country. High cost of fertilizer and fuel in the international market made in the situation worst. The government is doing its maximum to recover from this bad situation. As the Minister of Agriculture, I really appreciate your findings and technologies unveiled in this Asda event. You also know that some of the technologies which you generate do not go to the farmer at the grassroots level. This is a matter we should consider seriously. We must think of passing messages, findings, technical know-how to the farmers and other interested people efficiently and effectively. This requirement has been more essential than ever due to the prevailing situation of the country. High-yielding varieties and associated technologies are very important to the country. You all should take more effort to develop varieties of crops which we cultivate here. We were unable to provide the whole crop seed requirement in the country. For paddy, we have been successful in providing seed paddy to the farmers. But our quality seed paddy provision is around 20%. We should take immediate steps to increase this share to a considerable level. I am happy to say that Department of Agriculture has already been able to start the production of planting materials for existing vegetables such as carrot and radish. We import seed materials of most of the vegetables and crops like maize. The Department of Agriculture should develop a sound research, extension, and seed development program to produce local crop varieties and their planting materials locally. I request you to study on such programs implemented in countries like Thailand, Malaysia, India, and Bangladesh. It is great to know that you have awarded your scientists under the categories of the best scientists and the best agriculturalists. Such efforts will definitely encourage the officers of the Department of Agriculture to work hard to achieve the largest of the Department of Agriculture. You have not forgotten the service of past eminent scientists who have rendered much valuable services to the agriculture sector of the country by offering them due to recognition under the category of eminent scientists. I feel it is better to promote good agriculture practices, which is a blend of chemical and organic inputs to achieve the production in harmony with the environment. The government has taken steps to provide the farmers their chemical fertilizer requirement at a subsidized price. The government is also considering offer some money to the farmers to purchase organic-based fertilizer. Agrochemicals and other inputs are available in the market. As a country, we hope for a better Maha season this time and also hope we can stay at the same level or better. Let me congratulate all who got rewarded today
and I expect more technical advancement to support the agriculture sector in Sri Lanka. I thank you all for giving me this opportunity to participate in this event. Let us work together and rise. Thank you.
Thank you very much, Honorable Sir, for your valuable words. And now, for the announcement of the Asda Chairman 2023, I would like to invite Dr. Ajanta de Silva, Director General of Agriculture.
Yeah, once again I thank Mrs. Dishna Ratnasinghe for job well done for 2022. Thank you, Mrs. Dishna. The next is to nominate the energetic leader for the next Asda 2023. He is none other than Dr. Harsha Kumar Kadupitiya.
I think I got the name right, Dr. Harsha Kumar Kadupitiya, is it? Yeah. Yes, okay. We invite you, Doctor, to address this August gathering as the Chairman 2023. Can we have you on stage? Everybody, put your hands together and give him a round of applause.
And thank you very much, DG sir, that keep confident on me to give that opportunity to serve to the agriculture, to the Asda 2023. And I must use this opportunity to thank and congratulate that Asda 2022 Executive Committee and the chairperson, and they have done very marvelous job, and it was very successful in this year. And I know that there are many hard work behind that, many teamwork, many people and many officers work together, and even very long-term research, and many contribute to this research, and that some collaborative work to come up with this type of scientific papers, and that is very hard working, and until to get this form of good research paper. So that even not only the contribution of the scientific aspects of that, even in one Asda event, there are many aspects to look into, and that even that to get the form of this type of papers, there are many, then even many people read in every page to get this type of output by reviewers and also by editors and by page setting people, and there are many people work behind this to get that type of thing. And also there are many other sections to accomplish to get this type of successful Asda. For example, even in invitation, and also in this decoration work, that plant may grow in many months back and look after to up to this level. So there are many people work behind, and all of them ultimately expect after looking at the successfully completed each task. And after looking at the successful completion of this Asda event, only the satisfaction and only the only the happiness by looking at the successful at Asda. And this time also, I cordially invite you all to support as continuous support towards this Asda next time also. And I once again thanking for the DG sir for giving me this opportunity to serve for this Asda. And thank you very much.
While wishing Dr. Kadupitia all the very best, we have a presentation from the outgoing personality, the outgoing chairperson, Mrs. Disna Ratnasinghe, to next year's chairman of Asda 2023. Come on, everybody, a round of applause. Photographic capture. Congratulations on your appointment as the chairman of Asda 2023, Dr. Kadupitia. Well done. Right, and also all good things should come to an end, so it is with closing sessions. Finally, to propose the vote of thanks, I invite the chairperson of Asda 2022, Mrs. Disna Ratnasinghe. All yours.
Good evening. Honorable Mahinda Amaraveera, Minister of Agriculture, Honorable Mohandi Silva, State Minister of Agriculture, Mr. Rohana Pushpakumara, Secretary to the Ministry of Agriculture, Dr. Ajanthi De Silva, Director General of Agriculture, Additional Secretaries of the Ministry of Agriculture, Vice Chancellor, YMB University, other university academics, Director Generals of other departments, Additional Director Generals of the Department of Agriculture, former Director Generals and Asda Chairpersons of the Department of Agriculture, private sector delegates, all the foreign delegates, members of the Directorate, other distinguished guests, ladies and gentlemen, and dear colleagues. Asda 2022 elegantly emerged through many challenges. Executive Committee of Asda 2022 has worked extremely hard, both in the foreground and background, organized this one-day hybrid symposium. The success of Asda 2022 is totally a team effort. First and foremost, I thank our chief guest, Honorable Mahinda Amaraveera, Minister of Agriculture, who managed his time to attend Asda 2022, gracing the occasion and encouraging the DO scientists for further innovations. I express my sincere gratitude and appreciation to the special guest, Honorable Mohandi Silva, the State Minister of Agriculture, for your presence despite the busy schedule. I also like to thank Mr. Rohana Pushpakumara, Secretary to the Ministry of Agriculture, for being with us today and for your inspiring speech with eye-opening remarks. Present Director General of Agriculture, Dr. Ajanthi De Silva, was the driving force behind us to make Asda 2022 successful. Thank you, sir, for showing us the directions towards success of this event. Next, I wish to thank all the distinguished invitees, including foreign delegates, former Director Generals of the Department, former Asda Chairpersons, and all the officers from the Ministry, for accepting our invitation and joining with us today to witness Asda 2022. I am very grateful to the Chief Editor of Annals of the Department of Agriculture, Professor Chalinda Benaragama, for his constructive inputs you made in diligent extra effort to bring all scientific papers of the symposium to the best possible standards in Annals. Thank you, sir. for your unmatched contribution made to the success of Asda 2022. I also offer my sincere gratitude to Dr. Samanthi Vasala for attending editorial board activities despite her busy schedules, and to the editorial board of Asda 2022, namely Dr. Jayanta Senanayaka, Dr. Tushara Wickramarachchi, Dr. Lakmini Priyantha, Dr. Chandi Vellala, Dr. Jeevani Diddugoda, and Mr. Roshan Pereira, who worked very hard from the beginning to the end and made all the arrangements to publish Asda Annals 2022. I especially thank Mr. Roshan Pereira for his dedicated effort towards publishing of Asda Annals and for desktop publishing. Thank you, Roshan, for your marvelous support. It's with great pleasure that I am announcing that Asda 2022 received about 23 papers from the excellent work. We must thank all of them who submitted papers for Asda 2022. I appreciate your hard work, even with the COVID pandemic situation and the difficult situations in the country. Those who have not succeeded this time can submit your manuscripts next time with further improvements. And I take this opportunity to thank all the reviewers, university academics, and officials of other institutions for providing their valuable comments in time and helping towards the improvement of the quality of papers presented in the symposium. I offer our best wishes to all the award winners of the Asda 2022. I am thankful to the FAO, JICA, Copia Sri Lanka, and Sri Lanka for sponsoring the awards and for the various supports given by them. I am highly grateful to IPMP and CIC Agribusiness for sponsoring gold medals and all the other private sector organizations for various types of sponsorships. Thank you for your continuous support to Asda. I take this opportunity to appreciate Dr. BVR Punyawardana for his support on editing the script of the Asda 2022. Then, a special word of thanks must go to Mr. Hasika Kirthiratna, Deputy Director, Agriculture Press, Gannoruwa, and his staff, who made an untiring effort to bring all printed materials of the symposium on time with a very high quality. Director, NAICC, Mr. Hiran Pereira, Deputy Director, Audiovisual Center, Mr. IJK Janaka, Mr. Kapila Wickramasinghe, DD Video Production, Mr. Saraka, Assistant Director, ICT, Mr. Kamis, Assistant Director of Agriculture, and their staff also deserve a special acknowledgement for making this event with such a glamour with modern technology. The beautiful surroundings that we admire in this auditorium are their hard work. They were behind all the audiovisual activities. The whole event was delivered online via Zoom, which increased the number of participants. The official website and the online registration mobile app developed by the NAICC team is highly appreciated. Thank you very much for the NAICC team. Thank you, Mr. A.G. Karunaratne, Deputy Director, In-Service Training Institute, Gannoruwa, and his supportive staff for making arrangements for providing meals. I thank Dr. Jeevani Diddugoda, Ms. Srimati Edir Singha, and supporting staff at PGRC for your dedication and commitments of providing all the support for Asda 2022. I should thank the staff of the Engineering Accounting Division, especially Ms. Vajira Amarsena, who managed all financial matters in a timely and efficient manner to make this event a success. I would like to extend my sincere thanks to the Executive Committee members of the Asda 2022 for your dedication and very untiring efforts to make this event a success. Dr. Samanthi Vasala, Dr. Jayanta Senanayaka, Dr. Tushara Wickramarachchi, Dr. Lakmini Priyantha, Dr. Buddhika Abesinghe, Mr. Prabhat Nishanta, Mr. Kapila Wickramasinghe, Mr. IJK Janaka, Mr. Karunaratne, Mr. Chandana Wickramasinghe Tunga, Mr. Kamis, Mr. Chamila Pereira, Mr. Andika Lokuliyana, Mr. A.S.M. Roshan, Mr. Samantha Tushitakumara, Ms. Samanthi Godigamuwa, Mrs. Dimuthu Vijayaratne, Mr. Roshan Pereira, Dr. Jeevani Diddugoda, Mr. Hasika Kirthiratne, Dr. Chandi Vellala, Mrs. Vajira Amarasena. Thank you for your valuable support in making Asda 2022 a reality. I must give my special thanks to all subcommittee members who worked hard behind the scene to make this event a success. Finally, I must mention the great service rendered by Mr. A.S.M. Roshan, Assistant Director of Agriculture, as the Secretary of this Asda, also Ms. Nishamani, Ms. Ishani, and Ms. Ganga from the Asda Secretariat who completed every task on time. I'm thankful to all of you who have helped us in numerous ways to make this Asda 22 a reality. I beg your apology if I missed any name. And I take this opportunity to congratulate Asda Chairperson 2023 and wish you good luck to make Asda 2023, 25th Asda, a very special, very fruitful and more glamorous one. Thank you very much, ladies and gentlemen. Have a nice evening. Thank you.
And on that wonderful note, we come to the end of the closing session of Asda 2022. Ladies and gentlemen, I'm sure you'll join me in wishing Asda 2023 all success, and you've been a wonderful audience. Goodbye, everybody.